Yep, and so the base of our dive today, I think, is about 830 meters, and the top of this dive site is going to be 760 meters. We have about, I think, about a 900-meter dive planned for you all, and we'll just be hop skipping across the tops of this ridge. I think our maximum slope here that we're expecting is about 30 degrees or so. Let me just check. Yeah, we got about a maximum slope of 30 degrees.
I think we're going to hope to see some Lophelia. Yesterday, we found quite a bit of Lophelia rubble with no major reef formations, but hopefully today we might see some. It looks like maybe the current is going to be coming from the east, and that would be why there's a scour mark on the western side. So if we do see Lophelia, it'll either be on the southern side or on the eastern side of these mounds in the current. Does anyone on shore have any comments or questions? No?
If
We're
no
good?
one has any comments or questions, we're going to go ahead and start the dive once we reach the bottom. So feel free to join throughout the dive at any point, and if you have any input or anything, we welcome that you guys call in or put it in the chat room so we can make sure that your thoughts and ideas are known. And if you have anything that can potentially also help us identify things or even correct our identifications, please do so as well. Thank you.
Yep. And don't forget to log your info in CTube for us. The more data we have, the better the archive.
Especially for you guys later on. Great. We'll see you at the bottom.
All right, we got bottom in sight. At 820 meters down. Looks like we got some rock outcrops.
Yep, we're starting to see more, again, of this calcium carbonate material. We see more, the overall morphology looks to be more like slabs, and they also look to be manganese encrusted. So we're starting to see more of like these boulder-like materials. We're having these sharp edges, so it's not very smooth overall. Although the tops of these look like it has been blanketed by some soft sediments in this area, with just a few of these kind of like cobble rubble stones in the area. And it looks to be like those darker pieces or patches are those also ferromanganese encrusted corals in the area. Now we're just going to do a color correction for the overall video of the dive. And for those of you just tuning in, we're at bottom. We're giving the ROV pilots a minute to catch up with all the videographers to make sure all the color aspects are right, and we'll be on their way in just a few minutes.
Am I seeing coral rubble here in these divots?
Yep, that does look like coral rubble that's been encrusted by ferromanganese.
Got some small gorgonians ahead, possibly whip corals. I think the one at 10 o'clock is a bamboo coral from a distance, but I don't want to commit to that till we get close. It looks like here we have a whip coral. That could possibly be a black coral.
We're coming up on a really nice looking Antipatheria.
Yeah, let's get close. Can we get a zoom in on this whip coral, guys?
Yeah.
Okay. Okay.
Great, thank you.
Yeah, I would definitely call that a black coral. You can see the polyps are singular versus like 8 fingers or 6 fingers. So black corals usually have 1 finger. So this is probably a species of Stichopathes. Yeah, very nice.
And then we have a little yellow guy underneath him. Is that an anemone or a soft coral?
Probably a soft coral. I'm sorry, probably a solitary coral to get closer. Yeah, I believe he's a solitary coral.
Does he have an interior skeletal structure?
Yeah, he should. See how he's attached to the bottom.
Mm-hmm.
And then we've got this round bulbous white sponge right to the right.
Looks really nice.
Mm-hmm.
All those tentacles.
Yep, it's pretty cool.
So most of these polyps look like they're almost two-pronged
Yeah,
sections.
they should be singular. So they just, they grow like that, but they're really, each polyp is only one tentacle.
Okay.
Yeah. It's typical of a black coral, right? The Antipetherians. At one time, they thought they cured disease, so that's where they got their name, Antipeth. Antipeth.
That's interesting.
Mm-hmm.
But now we're actually looking into some sponges that might have some effects towards breast cancer.
That's right. So we have a few species, actually, of deep-sea sponges that help stop the growth of breast cancer in the lab, as well as pancreatic cancer. Our Harbor Branch's Oceanographic has a biomedical research department that has been doing research on that for about, 20 or 30 years. And we go out and collect sponges and give them to the biomedical research department, and they do all kinds of crazy workups on them. So there's at least 3 that I know of that have been found to possibly help or stop the growth of cancer.
Wow.
Yeah. So I think one of them is they're doing lab work on it. I don't think it's quite at human testing, but they've gotten pretty far with the one.
Well, one step closer is one step closer.
Mm-hmm. So we've got
quite a bit of coral rubble in the area. Sorry, didn't mean to interrupt you there.
That's okay. Looks like another Stichopathes ahead of us. And way out there, we might have some crinoids. And I think these little gorgonians might be bamboos. If you want to zoom in on, how about this one right here in dead center screen? That one with the crinoid on it.
And on the edge of it, a nice crinoid.
Mm-hmm. Yep, that's a bamboo coral.
With something adjacent to it, just beyond it, it looks like there's something yellow tinted.
Yeah, it's probably a different species of gorgonian. So these are the crinoids that Chuck saw yesterday, right? He said were usually purple. This one looks a little purple to me.
More purple than that yellow color.
Mm-hmm. Get the name from my notes from yesterday.
Those are really interesting features at the end of each one of those feather tentacles. It looks like it has like a hook at the end where...
Yeah, I'm not sure. I know he said that Chuck Messing from Nova said that those were natural. So it's not like someone came, another animal came by and ripped the pinnules off the tips. They actually grow like this.
Oh, interesting. Yeah, you can kind of see that each one of them the pinnules end at the same place. So I wonder if that has anything to do with stability or swimming or...
Maybe it extends their ability to catch stuff.
Maybe.
In the water column. I honestly don't know. I wonder if Chuck was on the line, he could chime in and tell us, but I don't think he is. Yeah, Xenometra columnaris is the name he gave it yesterday. So, Xenometra.
Whenever you're done, pilot. And then we have this really nice bamboo coral here. You can see by the bandings, this is fairly normal characteristics of bamboo coral.
They look, internodal, right? Where Scott was describing yesterday how the branches come off the main stalk. And if they are on the brown part, they are considered nodal. And if they're between the brown parts, they're considered internodal. And that's a distinguishing characteristic.
Between species.
Mm-hmm.
Nice. The tissue and the polyps look very translucent on this one, very glassy.
Yeah, nice and clear. All right, I'm happy whenever you guys want to move on. I got something in the water. So we're at the base of this ridge in a scour pit. It looks like at 825 meters. We're in east of the Million Mounds, or on the eastern edge of the Million Mounds. This is the seemingly last feature before we hit the eastern edge of the habitat area of particular concern that we are currently in. We're off 127 miles from shore, off of Florida.
That's right, and we're already seeing very different characteristics than from yesterday's dive where we started off, whereas at the base of that ridge that we were climbing, there were a graveyard of coral rubbles, and we didn't see much overall life there. We had very little corals actively growing. We did see occasionally some varieties of fish, but overall we didn't see much, and it's different from this dive here where we're starting off where we're already seeing some live corals even though we're at the base of this scour on the edge of this ridge, and this could have some indication of current systems passing through the area where it's still maybe the currents are actually promoting the erosion at this location, and therefore it's still enough for corals to be actively growing in, or
sorry.
oh, no worries.
Center screen, that looks like there could be a shark right at noon. Sorry, go ahead.
Well, that's interesting.
Is he eating?
He is.
Yes! Deep sea predation, yay!
And what kind of shark is this? This we've seen on Nat Geo.
Right. Oh my gosh. He's got a whole fish head. I don't know what kind of shark this is. He's pretty cool. Go back to the fish head.
Yeah. Finish your lunch or breakfast. Well, he now sees the RV. It looks like a bigger snack to
We might be a little overwhelming.
maybe. But
Cool.
wow. Really great way to start off this dive here. If anyone online happens to know what kind of species this is or what we're actually looking at here, it would be great if you could chime in or type it out in the chat room. We'll definitely give that a shout out. I wonder if it actually is a predator and killed the fish, or if it's more of a scavenger, just came upon the opportunity of a dead fish and started munching.
Yeah.
I wonder if those are specific characteristics of these specific fish or sharks, or I don't know. Seems to be really, really interesting.
My
Maybe
guess
a
is
full
he's
belly
scavenged.
there.
Yeah, right. Maybe he got half of most of it in.
Maybe he took it away from another predator.
Yeah, he could have. Only saw the head. It was pretty big too. It looked too big for him.
Yeah.
So I would imagine he didn't kill that himself. It's probably a scavenging species. Bottom dweller.
Really interesting. Sorry to interrupt your breakfast.
You got another cool fish here at center screen. Not sure what this is. He's got barbels.
On all sides.
Nice.
Wow.
He's pretty cool.
He is.
Possibly a codling of some type. Honestly, I'm not good with fish, so if we have a fish person online, they could help. But
Could we get the laser so maybe we can get a size measurement on him? Okay. Really interesting. He's having these like short bursts of, I guess, maybe pulsing water through his gills.
Yeah, or he could be eating something we're not seeing.
Yeah. But he's these really long whisker-like features. Do you know what they're called?
I think they're barbels, a type of barbel.
Okay.
So they use them to feel around the ground.
Okay. He has one coming up out of his fin as well, on the dorsal fin.
We have to remember that it's pitch black down here normally.
Yeah.
So to figure out how to get around. So you evolve to touch things instead of see things.
Some sort of sensory. And we
Help
also
him find
have
food.
a lovely Stichiopathes here again, where it seems to be swimming around, and some Ophiuroid.
Still the coral
Really
rubble.
cool looking fishies. Seems to be of grayish color and almost 2 white stripes going across his body. He is just showing all sides for the camera.
Right. That's pretty cool.
Really interesting. He looks like he almost has like a scrape along the side of his side.
Oh, up here?
So, yeah, maybe he got snagged on something?
That's
or?
probably his lateral line. So
Okay.
fish have a lateral line which it's a series of pores with a tiny little hair in it, if you will. And it can actually sense pressure change. So that's how fish school. They use their the senses from their lateral line to help them school. So, and then a lot of these deep sea fish also have light creating, like
illuminating
cells. Yeah, illumination. So they make their own bioluminescence. I'm not sure about this guy, but it's pretty common in deep sea fish. You'll see like lighter spots on their face, around their eyes. He's got them. Likely those are bioluminescent organs. But we can't really see it when we got big old bright lights blasting in his face. So.
Thanks, pilot. Well, we're off to a good start on this dive. I'm hoping it only gets more interesting from here.
No.
No?
What kind was it?
I think someone feels left out. Do we know what kind? Ah, okay. Wow.
Oh, there he is. And
We're
the
looking
replay.
at a playback yet.
He's got a really long tail.
Seems to be a pelagic type of shark.
Yeah, pelagic white tip maybe?
Maybe.
I don't really know the sharks that well, so I'm not great with anything that has a vertebrae.
We have people coming in now hearing the word shark. But going back to what we're seeing at hand here, this is more similar to what we've been seeing in yesterday's dive, where we have a lot of carbonate sediments in the area with an overlay of a lot of this coral rubble that's been ferromanganese encrusted. And when I say ferromanganese encrusted, it's a lot of that darker material that you're starting to see being encrusted on these coral fragments, and it has something to do with the concentration of the chemistry of the seawater that reacts with the skeletons of the overall. Oh, look, we have another shark. Is this another shark?
Yes.
Wow, we are quite active today. Guys, if we want to just hang out here, I'm good for that too. I wonder what kind of this shark is.
I
If
don't know this
anyone
one
out
either.
there is a shark expert, please chime in. We would love to hear from you.
You can tweet it. You can tweet it to us. I get tweets at Deep Sea Nerdy. I'll check my Twitter feed if anyone knows what these sharks are, because I certainly don't.
You can also tweet at Ocean Explorer.
And if you want to tweet us, use the hashtag Okeanos, Okeanos.
Or if you don't have Twitter like me, you can go onto Facebook and post any questions or comments at Ocean Exploration Research or at Ocean Exploration-Education. We're also on Instagram at NOAA Exploration. You can also use the hashtag Okeanos Exploration there. Feel free to ask any questions or post any comments. We're available, and we'll try to keep active on those and answer them as they come in. You can also contact me at my Instagram at DeepSeaKim, and at Stephanie's Instagram.
That's at Deep Sea Nerd on Instagram and Facebook.
And maybe tag some shark people in there too, so we can get some commentary from them. I'd love to get to know what we're looking at here. We're looking at a Shark Saturday here.
Hashtag Shark Saturday.
There you go. We're trending. Well, we're trying to. But as I was saying earlier, so the iron, manganese, and crusting that you see on these skeleton materials, it gives them that kind of dark coating look color. And what is that little guy hanging out there? An anemone?
It could be. Could be an anemone. Could be a solitary coral.
Yeah. And the ferromanganese encrusting comes from the ocean, the chemistry of the ocean water mixing with the skeletal material, and it's actually encrusting a lot of this iron manganese coating on top of the coral rubble, and it actually impedes a lot of the overall skeletal structure that we're looking at when we want to look at it under a microscope, or when we want to sample, subsample the coral skeletons for isotope work or age dating. It's quite a degenerating material, and we actually have to clean a lot of that off and kind of mechanically clean it, even with dental tools, and use something called leaching that we're trying to get rid of all of that material so we can actually get down to the actual skeletal bone of the material so we can actually start to work with it.
How do you age these through? Is it like rings on a tree?
So with coral skeletons, you can age them with like rings on a tree. However, a more precise way of measuring them is referred to as absolute age dating, and we use a lot of geochronometry in order to do that. And we can use, depending on the age range, we use radiocarbon dating, which is anything up to about 10 to 12,000 years old. We use uranium thorium dating that can go all the way up to about 500 to 600,000 years old. And then we can also use uranium lead. However, that does not work on this carbonate material very well. And past that, when we're going into the millions of years range, we actually use strontium 8786 dating. And these are all different isotopes that we're looking for inside the skeletal structure so that we can actually measure their different ratios so we can find an accurate date to how old these coral skeletons are. And it's pretty hard to date them actually per slice. So if we were to do a cross-section of one of these, we're actually, there isn't usually enough material to age date them each ring. So we usually end up doing a lot of these bulk analyses so we can get then a rough average of just the overall mean of age of these coral specimens. And right here we have another really cool looking fish. Is he a rat tail?
I would call him a rat tail for sure. I can see how his tail thins out. So this is they're modified like this to create the least amount of movement in the water column, so they're not detected by other species. Because it's dark down here, so the only way they can tell if there's other animals around is if they are close by or if they have light producing organs. So to protect itself, it evolved to have this thin tail that creates very little water movement, so it doesn't get detected.
Interesting. Does it have any way impact on the overall efficiency of the fish of where it can go? Like,
I would imagine it slows it down quite a bit, right? So like if you look at a shark, which has that long dorsal fin, tail fin, that's really a quick animal, right? It can move really fast. This little skinny tail is not very efficient with moving forward, but they don't need to go very far. They hang out near the bottom and look for food on the bottom and avoid getting eaten.
and he has those little sensory,
Yeah, the lateral line running on the side.
yeah, and also under his chin, those were the
barbels.
barbels.
Yeah. Looking to help him find food in the dark. Feel around.
And a lot of the overall white kind of material, fuzzy looking material that you're seeing on the overall coral rubble and whatnot, they're probably hydrozoans that are attaching themselves to these substrates and just kind of going along with the current and whatever you're seeing here. So that's fairly common to see on a lot of the overall substrate that we're going to be looking at. And I think we have another cutthroat eel ahead. Not sure on that.
Yeah.
He's about 20 centimeters long. These two lasers that you see in front of us are used to show measurements, so we can actually then see, because a lot of the time in these deep oceans, things either look a lot larger or a lot smaller than they actually are. So we're using the lasers to actually get an accurate measurement, and they're about 10 centimeters apart. So by having the lasers on, we can measure that this guy's probably about 20 centimeters long. We're seeing a lot of this Lophelia rubble around as well.
You can see this fish kind of has the same modified, thinned out tail, right?
So is this the cusk eel? Cutthroat eel?
I'm not sure. Has a chordate nerve? I have no clue.
I confuse a lot of these. We're not fish people. So again, if you're a shark or fish person, please chime in. Let us know what we're looking at here.
Cutthroat.
Okay. Nice, thanks, pilot. Guess we have our fish person.
Yep, so we're going to be heading up slope. Looks like we're about, an eighth of the way up the slope?
Yeah, I would say.
What do we have here? This white thing sticking up.
Could that be a stalked crinoid?
Oh, maybe.
One of the first ones we're seeing on this expedition.
Can we get a zoom on this guy? There's two of them, so it's either. I feel like we're too shallow for stalks, but let's see. Maybe I'm wrong. But if there's two of them, then they're on this. They're just sharing a dead stick of an old coral. I get close and look at the pinnules on the bottom and see if their feet are actually well, not really feet, but their little root.
I would call them feet.
Feet are touching that, or if they're sticking up. All right, so you can see this one has feet. So that one is not a stalked crinoid, it's a camatulid.
However, the skeletal structure that I'm looking at of the stalk does look like a crinoid stalk. So if we could get a shot of the top, that would be great. Oh. And yeah, it is a
like
stalked
crinoid.
crinoid.
Oh my gosh. That's super cool.
That looks really great.
I just texted Chuck Messing. Hopefully he'll call in. That's pretty cool. So it looks like he's got some kind of zoanthid or hydroid growing on his stalk. We are in the middle of a shift change, so hold.
Yeah, we'll give these guys a chance to change out. But in the meantime, why don't we introduce ourselves? So, I'm Kimberly Galvez. I'm a PhD candidate at the University of Miami, Rosenstiel School of Marine and Atmospheric Sciences. I study marine geology and the aspects of how coral frameworks interrupt and change the morphology of the seafloor and how they grow over time.
I'm Stephanie Farrington. I'm from Harbor Branch Oceanographic Institution at Florida Atlantic University in Fort Pierce. I do a lot of work on the habitats and corresponding invertebrates around deep sea and mesophotic coral reefs. I've been there for about, I think, 9 or 10 years. I have a graduate degree from Nova Southeastern University's Oceanographic Center.
Great, Steph. Why don't we let these guys nestle in really quick? Great, thanks. Do we have a minute to introduce the rest of the control room? One second. So in the front row, we have our ROV pilots and nav. Why don't you go ahead and introduce yourselves?
Sure. Sitting pilot now is Chris Ritter.
Sean Kennison sitting co-pilot.
And sitting next to us, our lovely videographer.
Yeah, back in the back row is Annie Wade on clipping.
Great, thanks guys. And these are all part of a really large team that we're part of with the Okeanos in order to be able to explore the deep sea and get all that great footage that you guys are able to see live, as well as later on. We require a lot of people and manpower in order to actually make this all operational. So a big thank you to everyone that works on this ship.
Okay.
Yeah, there is quite a large team that gets all this to work, and it takes months and months of planning to get everything together and organized and permitted and all kinds of crazy stuff. So it takes a lot of people to get this functioning and beautiful.
And
Yeah,
so,
and Watchley, we should mention, sorry, I didn't mention this earlier, that we're all engineers and video engineers working with the Global Foundation for Ocean Exploration. I left that out earlier, so
Thank you.
thanks, Watchleys.
thank you. And if anyone is logged into the chat room, you can see that the Okeanos nav posts every now and then the overall lat and long depth, temperature, salinity, and dissolved oxygen ratios that the ROV is currently experiencing at this location. And we can already see that there's a temperature change from that average 6.5 on yesterday's dive at that location to on average, we're seeing here about an 8.2, and that is always in degrees Celsius that we're going to be discussing this in. So that is already an over 2 degree change of what we're seeing from the location on yesterday's dive today. And that can impact a lot of the overall biology that we're going to be observing, including these corals, maybe even more of these fish or sharks. Maybe they prefer certain water temperatures or whatnot. We're actually observing something in the water column.
Ctenophore. I got a ctenophore in the center screen. Look at that glow.
Ooh.
Oh man.
And I don't know if you were able to catch an eye of it. He is actually glowing in like a rainbowish color. You can start to see him illuminating. Ooh, that's a
Our
really
thrusters
cool.
might be tossing him around a little bit.
Yeah.
He's moving out.
Yeah. That was really cool to see. I never got to see one illuminating like that before.
Yeah, they call them the comb jelly because they have 8 rows of combs that go along their body, and they flick those little combs up and down, and that's what causes the light.
Wow.
That's where the light comes from. They're quite beautiful. Okay.
Sounds good. There's actually a dark feature straight ahead if we want to go look at that, if we have the time to go look at that. If
The stretch.
Oh, okay. Okay.
Yeah, so
Sounds good.
to explain what the pilot's speaking about, we actually have run 2 ROVs at the same time. So we have the Deep Discover, which is feed 1. That's the video footage you're seeing from feed 1. And then we have Sirius, whose video footage is being broadcast over feed 2. So Sirius actually hangs above Deep Discover, D2, and helps it embrace the impact of the ship to keep that from affecting the large ROV. And I always say I don't think this feed really gives the size of this ROV justice. If you're looking at feed 2, that ROV is 9 feet tall and 10,000 pounds. So you can see how large it is and quite how far away the Sirius is from D2. So when he's talking about he doesn't have the tether to move forward, it means that he doesn't have the distance between D2 and Sirius to move far enough away in order to get to that point.
Yep.
What they actually have to do is move the ship forward, and then that pulls Sirius forward, and then D2 can get closer. So it's this constant dance of the ship's crew piloting the ship, and then we have 2 active pilots, the pilot and the co-pilot, who actively move the ROV forward underneath the ship. So they're always dancing around and working together in this unit to help us move forward through the water column.
And we're coming up, and it looks like that maybe black feature looks to be somewhat like a fish of some
I've got
sort.
a fish dead center, but I'm not sure what the black one is. Maybe it's an octopus. Can we get a snap zoom on that? Oh!
Ooh!
Oh, it's like a crab. It's a shrimp or a lobster?
I
Shrimp.
have no idea what
That's
we're
a
looking
shrimp.
at. That is really cool. It
That
looks
is a
like a
shrimp.
spider vampire shrimp of what we're looking at here. Does anyone know what kind of
Look
shrimp
at those
this could be?
feathers.
Wow.
It might be a royal red. That's my guess. Look how cool.
We're
He's
about
huge. Can we get lasers too if you can put the lasers in view? Oh, there's line there. Oh no, that's his
his
antenna. Oh my gosh.
Wow.
Oh, he's 20 centimeters almost. He's pretty
yeah.
big. Cool.
Wow. A couple
got-
days late on Halloween there, but
Right.
he sure looks creepy. Wow.
So if this is a royal red shrimp, which it looks like it might be, they're actually fished. So it might be a relative of that. He's pretty cool. Maybe another stalk crinoid right in front of him.
Yeah.
Ooh.
Oh, we're seeing him. He's on the move.
And then we have a glass sponge too, the wedding sponge.
Wow.
Oh, no way. He's super cool.
And he has 2 smaller claws and then 2 3 sets of little claws, and then he has some legs that are sticking out, probably more for sensory.
Yeah,
items, but-
likely with the hairs. To identify a lot of these arthropods down as species, you actually have to pick them up and count like the hairs on the chelate fingers, and like they're very specific. So maybe not so much with this guy, but I'm not, some of them are obvious, but some of them you really have to have a specimen in hand and sit down and count count the hairs on the chelate arm and the left leg, and yeah. So a lot of them are pretty hard to ID from video. Sometimes we can get them down to genus.
But this is some great footage we're catching of this guy.
He's pretty cool. He's got swimmerettes on the back. Really long antenna. He's a beautiful bright red color too. So when we're this deep, there's no light, and red is the first color to disappear as you go through the water column. So it actually benefits deep sea animals to be a bright red color because they just disappear. They actually don't reflect color back because there is not enough light for them to reflect the color back. So that's a camouflage in the deep sea, is this deep red color. So a lot of the species down here are this deep red because they're invisible.
He just looks overall really cool.
Mm-hmm.
Whenever you're ready, we're happy. That was a really cool find. Can we get a snap zoom on that white thing that is right at the bottom now of the screen really quick? Oh, it seems to be like we're coming up on another kind of fish here. Can we get a zoom in? Oh, it seems to be another kind of fish. Oh, struggling a little bit from the thrusters. Oh, okay. Then he just seems to be on the move. Maybe we scared him a little bit with the lights. He's really cool using some of his fins to just kind of hang out and rest. Checking us out. He's not sure what he's seeing. Really cool. He has some black spots all along his body and on the tips of his fins. If anyone can give us any more details about him. Oh, and as Stephanie mentioned earlier, those dots that we're seeing all along the bottom edge of his body, those could be illuminating, and they look really interesting. As far as we can see, those little dots could be the fact that he can self-illuminate in certain aspects of his body, and like Stephanie was saying, with the hairline of his overall body, we're seeing at the top that hairline structure, but those illuminating little dots seem to be towards the underside of him. Not sure if his fins are capable of illuminating, but maybe certain aspects of his body are. And it looks like he can just kind of suck in some water to be able to pump water through his gills so he can actually remain stationary, where a lot of other fish are required to constantly keep swimming because they don't have those features. And his dorsal fins, the back dorsal fins, look really interesting in the fact that they look almost frayed on each side of the top and underside of his belly. Are we able to get the lasers out so we can see how big he is? Sounds good, thank you.
He's pretty shiny on the bottom.
Yeah. I was just saying earlier, maybe those dots that we see on his belly could be the ones that are illuminate.
He has photophores. I'm not really sure. I have to look at him
Ah,
a little closer.
he's generally a little guy, maybe about 15 centimeters in all. Again, the overall geology here seems to be a lot of that coral rubble that we're seeing, some small little cobblestones and boulders, but we are seeing some slabs of this carbonate material over the landscape here. And it seems to be quite broken up and eroded in certain areas, maybe more jagged, and in others smoothed out also. Over the tops of these, they seem to be almost nodular, which can be characteristic of a lot of limestones that have been then ferromanganese encrusted.
Looks like we got another Stichopathes here, and I think I saw a stalked crinoid to the left. As we come up the hill.
So now we're starting, just very slowly, we're at the base of this ridge, starting to move upwards along the slope wall. Ooh, yeah, there is. I don't know if he's a stalked crinoid or if he's just hanging out on the edge of a bamboo coral there. And as we move upslope, we're starting to see more and more of this kind of slab-like structure of the substrate here, more than the overall coral rubble. And that's just indicative of just either erosional processes that are occurring in this area. The current is smoothing out some of this rock and leaving some ferromanganese encrusted stones, which are harder then to erode than the underlying carbonate, out exposed.
This looks like it might be a bamboo coral. I can't really commit to it till we get closer, but the formation looks right from a distance.
And just as an overall observation, we're seeing a lot of any sort of biology associated that's stationary is generally picking a lot more of these slab features, the harder substrate that we're looking at overall from the overall sediments surrounding the whole feature.
Yeah, it's pretty common. The soft bottom isn't really a great habitat to stick to.
Yeah, usually they tend to just fly away then in the current system, so...
Usually, yeah, there's usually a lot more biota attached to the rock than necessarily sitting around in the sand. And there are some species that prefer sand, like sea pens or tube anemones. They prefer the sand. They actually grab a clump of sand underneath and bury themselves into the sediment.
And even some crinoids.
Right.
Because they have those long feet and root-like feet structures that can dig then into the sediment and hang out there. Or like we also, like Scott France yesterday said, some species even of coral have this like bulbous feature at the bottom where they can inflate it then and fill it with water so it holds them while they can just stay stationary then in the sediment overall. But we're seeing a lot generally that these, especially the coral features and crinoids, tend to stick to those hard substrates.
This is definitely a bamboo coral. We got to zoom in real quick on the branching. Yes, please, if you can.
And then there's a coral off to the side too. Maybe afterwards we can get a look at it.
So it looks like, what, internodal?
I can't make it out just yet.
Yeah, see how the branching is on the white area between the two brown stripes, right? So that makes it an internodal branching.
You're right.
Look, they've got it. Looks like we have a tiny little worm coming up this side on this one on the right. Maybe.
A worm or a tiny shrimp? Oh, no, I think you're right. Like a little worm. A polychaete?
Polychaete, yeah, you can see his legs moving. He looks like a caterpillar almost.
Ooh.
He caught something.
You can see these polyps retracting when they end up catching some sort of material that's part of the marine snow, and the marine snow is a lot of organic particulate matter that kind of just flows through with the current systems, that what a lot of these stationary organisms are looking for and trying to catch with their little tentacles so that they can pull it in and actually eat, because these corals never see the light of day, so they don't use any photosynthesis in order to actually produce sugar, which then is energy for them to grow. So they rely strictly on current systems, bringing all of that organic material to them. And we have what looks like a little shrimp caught on a tentacle. I don't think he can eat it, but I think he's going to still try.
Looks like a euphalzid or something. A little shrimp.
Oh, it seems to be another one also on the tentacle there.
Nice.
I wonder if they just hang out there trying to steal the food, maybe, if the polyp catches one.
Yeah, he's a little too big, I think, to get eaten by that coral. Nice, robust base. He's pretty sturdy there.
Is it common for some of these corals that we see tissue growth along the base and then spread out onto the substrate, or generally they stick to their own precipitated skeleton?
I think it depends on the species, but most likely most of the time it's stuck to themselves. So there are no, you're fine to move on. We do have we have some
Wetting
glass sponges. There are some encrusting gorgonians that like creep out and encrust on the hard bottom. But most of these stalked ones, the tissue stays on themselves. Oh, Chris, you're on the phone?
Yes, good morning.
Good morning, Chris. How are you?
How are you?
Good. Who else is with you guys?
Well, right now I'm the only one here
You're
in
the only one?
ECC center, and yes, but it's excellent, and looking forward what we will see today.
What'd you say, hon?
And
You're on a delay?
I'm already on time in the right video live stream with
Okay.
you. But I'm refreshing the computers in the ECC room.
Okay.
Okay. How are you guys doing?
Good. We got a nice launch this morning. Everything went smoothly. We're
Saw some really cool fish
Yeah.
earlier.
yeah, 2 sharks, and there's a shark in the water column. That's pretty cool.
Ooh, it looks like we have another bamboo coral.
That's very neat. Okay, so I'm here with you, and can whenever that you find the sponges or any other thing, we can have a connection, a conversation.
All right. Thank you.
Thank you.
Now, this is a different species we haven't seen yet. If we can get a clearer shot of him.
Oh, there was a worm just disappearing off to the right-hand side there. Could this be keratosis?
I don't want to commit to a species ID. It looks almost like Swiftia, but it's not the same. It's not got the right branching. Okay. I don't know, Scott, if you're listening, you can chime in and maybe help us out with this one. I don't really
Ooh,
know.
that looks great. Can we possibly get a tighter zoom on some of the polyps?
It could be like in the same family, Paramyrcia. Primnoids are usually much more fine, I think. I honestly don't know what this guy is. So, maybe your guess is as good as mine. That's pretty cool.
What was the guess, just so the listeners at
Primnoid,
home-
I'm sorry, possible Primnoid.
Yeah, the tentacle structures also look really interesting. They're almost web-like. It almost looks like there's even a membrane on top of it, but it's not. It's just the tentacles overlapping with each other.
And their the tentacles are have little like branches coming
on them.
off. Yeah. If you look real fine at them, nice. That's quite beautiful. I mean, it's definitely an octocoral.
Yeah. I'm not seeing that banding, so.
Well, it's not a bamboo coral.
Nope.
Pretty neat.
Oh, and it looks like he touched laterally up on that rock rather than just straight up at the base with a sponge in the background. Is that a sponge?
That's a sponge back there, yep. I don't know how, oh, these are cool too. These cones over here on the right, on the left. I don't know what those are.
Glassy cone structures. Maybe a former sponge that died off.
Maybe. Pretty cool. And they could be, they could be egg sacs, old egg sacs too. I don't know. I don't know what those are. They're pretty neat. They look like glass though.
Oh yeah, this is a very nice, incredible composition of several sponges.
Chris, can you turn down your TV? I think I'm getting an echo.
Oh, yes. Okay.
There you go.
Yes, sorry.
It's okay.
It is a very nice, like you say, it's either leftover skeleton of 3 cup-shaped small sponges.
These 3 glass sponges, or you think they're glass sponges?
They look like glass sponges, but they're so transparent that it looks like it's not alive anymore.
Yeah.
And then to the right of it, you have a small round white sponge that might be a exact or a calcareous sponge.
A calcareous sponge. Okay. How about the one at the base of the gorgonian, the yellow bulbous one?
Yes, that definitely is a demospongia. So from the class Demospongia, the most diverse group of living sponges, and it seems to be a small specimen, so it's hard to distinguish if it's going to grow into a more clear form. It could be many things. It could be an astrophorid. It could be a rocky sponge. So it could be a hypercalcified sponge. So
Awesome.
hard to say what, but definitely a Demospongia.
Awesome. Thank you. It looks like we have a jellyfish that landed on the ground.
Hanging
he's
out
cool.
there.
You can see again he's got this red stomach, right? Red internal organs. So the jellyfish do that because the food they eat is bioluminescent, and if they were, they actually cover their internal organs with this red, coating, right? So that it makes those bioluminescent animals they just eat disappear. That hides them. So that's kind of neat.
Is he upside down? Ooh.
Oops.
Oh, there he goes. Okay, well, that was
pretty
really
active.
interesting to see. Oh.
I have no idea what this Gorgonian is. It's super nice. Get back to it.
Yeah.
I'll mark him.
Oh, there he is. He's back again, actually. He's swimming right on top on the right-hand side of that coral. Ooh, another cutthroat eel.
Yeah, he did.
He looks really cool. Does he land upside down? Is that what we're seeing? Well,
I don't think so. I think he sits down on the bottom and then takes his tentacles and puts them over his bell.
Oh.
So he's holding them up.
Is that in order to potentially catch some prey, whatever would swim above him?
I'm not sure why he's doing it. It could be just so he doesn't get all that junk on the ground stuck in his sticky tentacles.
Could be,
You know?
too. Oop, there he goes again. All right. And I think we're good here if we want to move on.
We have more of these Heterotella, the wedding sponge. There's one at 3 o'clock. Looks like these slabs are getting darker.
Yeah, so they almost look like basalt, but we know in this region that it's a lot of carbonate material setting on here. So it's unlikely basaltic material. It's still likely that ferromanganese encrusting that we have been seeing up on over the corals and over some of the substrate that's been exposed for quite a while.
Again, we've got these glass wedding sponges. There's 3 of them in view right now. We have probably a bamboo coral at 3. We got a fish at 10, the cutthroat eel maybe.
With what could be a yellowish sponge underneath that bamboo coral.
Yeah, it could be
Or
yellow
some-
encrusting or...
Something yellow.
Yeah, I
somewhere.
can zoom in real quick.
We do see some potentially stuck crinoids in the back too. This is very different from what we've seen on the previous dive. And another solitary coral off to the bottom right-hand side, that orange looking.
Yeah. This is probably an encrusting demosponge of some kind. Yeah. And there's a big old worm on there. And then we've got maybe possibly an egg sac. You can see the soft, clear, gelatinous at 6 o'clock.
Under that white sponge
here's
with
a- Go ahead.
No, that under, just notifying, just it's under that white sponge in case people can't really see it.
And we have this same Gorgonian species we saw a few minutes ago.
Yep. And we have a lot of these cup corals nestling themselves on either these sponges or I think there's another one that we saw on the coral. Oh, there's quite a few of these worms on there. Oh,
Hey, Chris, you just call this a Dema sponge? Any names for it?
Yes, this cream tan sponge that you can see with connulos, little like spike surface. I'm almost positive will be Dendroceratidae. So it's again in the group of the Demospongia, but it's a group that only has fibers and dendritic fibers in their skeleton.
Oh, as opposed to spicules?
And I would say it's a Dendroceratid because of the...
Dendroceratid?
Yes, Dendroceratida.
Awesome.
and what's really interesting...
then these small white blobs that are probably exactinelids that we saw yesterday.
Glass sponges, yeah.
Sporadically.
Awesome. Thank you. Ooh, nice heterotella. This is the wedding sponge. They give these away as wedding gifts in Japan, I believe. There's two tiny little shrimps that live inside them, and they live in there their entire lives. There's a nice shrimp.
Ooh.
Oh, yay! Failed attempt.
He lives to fight another day.
Yeah. Nice try, buddy.
And we can see a lot of these solitary cup corals growing around in various colors. We've seen a lot of them in like this orange pinkish and now some in yellow that you can see right above where that shrimp was. So it is quite active in this region. Despite not being able to see all that much along this substrate, as soon as you zoom in, you can actually, it is quite covered in a lot of the biology that we're seeing around here. We are just under halfway up scaling this wall here. And we can see that there's a lot more jagged edges towards this carbonate features. And the soft sediments are starting to infill in this area. The overall morphology looks really interesting. We're seeing a lot of, again, that ferromanganese crusting. Any exposed rock really in this area is subject to it. And if it was possible to potentially dig underneath, if there were some soft sediment on top, we would be able to see that just a few centimeters below where that sediment would be laying we'll probably have some fresh carbonates in that area. What do you think that stalked thing on that right-hand side of the screen is?
Looks dead to me. I'm not sure. I have to get a closer zoom to it.
Maybe...
Ooh! Siphonophore? Look at him go. So siphonophores are actually a colonial animal. Ooh, look! Oh, fun.
Really cool. So when you say colonial animal...
Yeah, so there's actually, like, if you were to break up each little bell, they would be able to survive on their own. They're actually independent animals that live as a unit. So one part of it will act as the bell, will help move. One part will be the reproductive organs. One part will act as the food. And they all just live, together as a unit. But if they were to separate, they'd be able to reform and live independently.
Oh,
So
nice.
like
Would they then, are they clones of each other, or
I don't know how they reproduce. I would imagine that there's some kind of clone, like that, where they bud and then grow that way. Can we look at this yellow stalk at,
just about noon?
yeah, I think it's dead, but looks like we've got some spindly bamboo corals too. And a Stichopathes, the black coral.
Yep. And Chuck Messing says on the chat room that slender stalk crinoids with 5 arms are likely Democrinus.
Oh, nice.
And the species in the Atlantic is not very well understood, so maybe if we do
Specimen.
see someone, we'll collect.
Okay. We saw a couple, so
We
maybe just
did.
grab one. We'll grab one next time we see it.
Ooh.
I have no idea what's going on here. It looks like a dead stalk.
I think a dead sponge of at some point maybe, with a whole bunch of bryozoans and hydrozoans and ophioids and other sponges. And what is that guy hanging off the side of it on the lower end of the screen
That
there?
looks like...
It's not a crinoid, is it?
No, it might be hydroid.
Just a really large hydroid.
He's weird looking.
Yeah, so even when you're dead, you're useful.
Right? Helping them get up off the ground so they can filter feed.
Maybe those, that conglomerate of spongy-looking material at the base could be from what was previously part of the sponge and now fell off
Yeah, it could be.
and
Get a
just
little
hanging out
closer.
at the base.
Yes, I'm looking at the image, and I'm not sure that it's a dead sponge.
You're not sure it's dead sponge? Maybe it's a stalk of a dead black coral?
Or even could be a live sponge.
Oh, you think it's alive?
Oh.
It could be, I mean, something to remain, so erect and solidly having a community growing on it. That, I don't know how old is this, but by the growth of all these organisms, I'd say that it's been there for a while.
Mm-hmm.
So I'm not sure it will be a dead sponge.
Okay.
Definitely, I cannot tell what a sponge that will be, but yes, I'm not positive about that it's dead organism.
And what about some of the material that was at the base of the stalk here? We're about to zoom out so you can see it. Do you think that has anything in relation to the actual sponge itself, or is that a different kind of sponge? Just
What a cool observation. Let me think about what the size. I cannot see the
Lasers?
laser.
Can you guys turn the lasers on?
Could you tell me more or less what is the size of
There you go.
of this tube that is coming off the sediment? Because what we could be looking at is a siphon or a oscular projection of a sponge that actually live in the sediment.
Oh, okay. Kind of like the tubes that come...
oceanapia
Yeah, that ocean app, yeah.
That has a tube that either is excurrent, so it has an osc, or there are cases in which these tubes contain incurrent pores that bring water to the sediment area, to the main body of the sponge that is under the sediment. So it would be both cases.
Awesome.
interesting. And
Thank
Chuck wanted to state that on the stalk, it's likely the fuzzy trees that we were kind of looking at are a mixture between some hydroids, zoanthids, and even some
Forams, yeah. Foraminiferans. And guys, pilots, if we see another stalked crinoid, we're probably going to collect it. So just be prepared for that.
These are some nice corals looking along here.
Another heterotella. These wedding sponges.
Now, do they get the name specifically because they've been used culturally as a wedding gift in Japan?
Yep. And the little shrimp inside are married.
Oh.
Right?
Can we actually get a look on that left-hand side, whatever that dark coral looks like to be? If it's alive or not. So it's interesting to note that we have not seen a scleractinian coral yet. Looks to be some sort of black coral, potentially, some Antipatheria.
Oh, yeah, definitely. Let's get a good zoom on the polyps. That sure looks like a black coral to me. Has a nice black stalk. These are, I don't know about this species in particular, but black corals are commonly poached for jewelry. So they are actually a protected species under the CITES laws. So if, for instance, we collect these for specimens and I want to ship them to literally anywhere, I have to have permits because they're endangered.
Oh, we got a little squat lobster hanging out on him too. And this... Oh, we have that jellyfish that's back in the background there. You can see that red dot over some, with some of the branches there.
At 9 o'clock, yeah.
And here we have...
I don't saw any little guys on here. Little shrimps or something.
I don't know, it looks like schmutz.
Schmutz. Some schmutz. Sea snot. It looks like maybe.
Oh, there's a little tiny one off to the side there. Oh, and what's dangling?
I don't know. Maybe a clam? Stuck to him?
Maybe. And is that specific thicker area of the branch there could be housing a worm?
It
likely?
could be, yeah. It could be housing a worm. It could be damaged and recovered. Oh, here we got a squat lobster or something on the right.
Really nice look at the polyps. If we can get even a tighter zoom on some of the polyps, just so we can make sure later on we can have a good identification.
Yep, you can see they're single again, right? They're little single stalks. Right, that's indicative of antipatharians or black corals. As opposed to having 8 tentacles a polyp or 6 tentacles a polyp. There's only one tentacle per polyp. I think that's a clam. I don't know what kind of clam. It could be an ostracod.
I was thinking ostracod, but
I don't think
I'm
they
not
get,
sure.
unless he's stuck to it. He might be stuck to it.
He might be.
He
looks like
actively
he's
stuck.
just
Hanging
out in the middle of nowhere. Not sure how he got there.
Scott said,
looks good.
oh my gosh, I'm not going to be able to do that. Ascothoracid parasite. Did I get that right?
Oh, is that larger black section?
Yeah, the clammy looking thing.
Oh, the clammy looking thing. Ah, okay. So it is a parasite.
It's parasitic.
Okay.
Nice.
So does it feed on the tentacles and the tissue?
And Scott, if you know that can identify this antipatharian, I don't know which one this is.
Maybe he's trying to dial in at the moment.
Yeah, he said the thick part is probably modified for worm tube.
Oh, okay.
And he's standing by on the ID.
Okay. Really nice looking squat lobster there. He is more pinkish than some of the other ones we've seen on the last dive.
We've got a brittle star here at the base. Look at the rock, it's like pockmarked.
Yeah.
See that?
And that's very characteristic of limestone. When overall, the evolution of it starts to create a lot of these hole strictures, really porous. And that's due to the overall formation of how it forms, and then also sometimes when it interacts with a different current system that maybe is more acidic, it starts to erode and deepen those pores. A lot of biota tends to also bore into the limestone, creating some of those holes, attributing to the features and digging further, and then even using some of the previous pores there to create their own network system through the entire limestone feature.
Nice. Scott said possible hexapathes, but he wasn't 100% on it. We got a nice slope on the, you can see it towards the left. Looks like maybe 45 degrees we're coming uphill. Yeah. So I estimate slope using, maybe more than 45. Using the corners of the screen, right? So if it makes a square, perfect diagonal, that's 45 degrees. So that's about 45 degrees. I don't know if that made any sense, but my brain works that way.
No, and that's a perfect way to kind of estimate on what kind of slope angle we're looking at. From the bathymetry, the slope was indicated about 30 degrees, but since it's somewhat a coarse resolution at 25 meters, we can always see some slight different variations on what we were expecting then from the actual bathymetry.
We are on the steepest part, though, are coming up on the steepest part that was in our multi-beam. Here's another one of those possible ocean apia that Chris said. Can we look at the 9 o'clock, the stalk at 9 o'clock?
So cool.
Do you think that's ocean apia, maybe?
It looks, yes, it could be. Let's get close.
It's got that same single stalk with the tissue around the base. And actually, most of ocean apia, right, grows under the sediment, right? It grows down into the sediment.
Yes. Yes, Stephanie. So that's something that made me curious. That it seems such a compact substrate. How is the sponge? How much can it put its body under the sediment? But apparently, it might be very old organism that started growing and has accumulated rubble around it.
Nice.
Definitely.
You want a piece of it? Would it be worth grabbing for the community?
Yes.
It would be worth grabbing?
I think it would be a very interesting sponge to identify.
I'm going to collect this guy.
This is the second one you observe.
Yeah.
And so it's usually, if I'm understanding this correctly, it's pretty uncharacteristic of it to be on such a hard substrate rather than around more sediment where it can bury under?
Yes, this is a genus that usually grows, the main body grows under rubble or sediment. But I can see that this sponge, again, might be old, and it has accumulated rubble and material around it. And we can see how also it might be an important community builder because it's a lot of things grow.
We might collect
around
this.
So this looks very interesting. If you see one of the projections, the larger one has a hole inside, and then you have other projections that are blind at the end. Those are called fistulas. So there's usually a different function of each of these type of projections. Some of them might be in current for getting water inside of the body, and the large central one with the central hole on the top might be the oscular projection where the water is being shot out of the main body. So this definitely, if we continue finding it, it might be an important, participant or component of this.
Do you think it would be better to wait to see if there's another one before we commit to taking it?
I will collect this one.
You would collect it?
It's a nice specimen. And if it's not complicated for the pilots, for the positioning of the ROV, I think it will be a good opportunity to collect it.
Okay, let me check. Hey, pilots, do you think you could grab a piece of this guy? Okay. Hey, Chris, is it better to get the fistule that's poking out of the top, the X-current siphon, or a piece of this, bulbousy part of the bottom?
Yes.
Or both?
So if you can grab it at the most basal part and just grab a piece, and we'll have the photographic material representing the overall shape, but it could allow us to get a little bit, it will be good to get a little bit of the base of the species.
The base.
So
the base.
At the base of this large tube-like projection,
Okay.
it will be the best.
So grab. Basically, we're going to grab the tube and maybe a chunk of that weird shaped stuff if we can. Okay.
Yes, and there's a lot of things growing on it, so there might be interesting organisms for other people to look about this small, niche or this small community that grows associated to this sponge.
Okay. Let me communicate to the pilots and let them know. So, pilots, can you hear me? Okay. So we're aiming for the base of that large tube, and ideally grab the fistral. That's called a fistral. Or ideally grab that whole thing. Yeah. So at 6 o'clock, see 6 o'clock on the large stalked part. So you grab the base of that, and if you can, some of the bulbousy material that's surrounding it. Does that make sense? Yeah. Yes. That would be good. And I think it's probably going to be crispy, but let me check with Chris real quick. So hang on a sec. Hey, Chris, do you know the texture of this? If we're expecting rubbery or crispy or broken or, fragile?
Yeah, it will be the tubes will be fragile, but they have enough sponging that it won't crumble on them.
It'll be fragile, but not crumbly.
Super hard. Yes.
Okay.
Not crumbly. It will be, but it won't be elastic, so it will be easy to break a piece off.
It should be easy to break. It's not going to be rubbery.
Yes.
Okay.
Yes.
Perfect.
It won't be rubbery.
Thank you.
Now, could you give me an estimate of the size of this?
Yeah. Can we flick the lasers on before we? All right. So I'm looking at 10 to 15 centimeters tall, and probably about 10 to 15 centimeters wide. Maybe 20 wide.
Okay. Okay.
Yeah, closer to 20 wide. Yeah. Yeah, you're good. Thank you. Yeah, that's good. It just crumbles apart, yeah. I got exudate. So that's either slime coming directly from the sponge or just dirt.
Excellent.
Yeah. Yeah, that's fantastic. Chris, would you say this could be a new species? Or
I would say yes.
Thank
I would
you.
say yes. Let me check how many records. Well, deep water oceanapid, there's not too many records. So I would say there's high probability that this is a species that has not been described.
Okay.
So it will be a very interesting sponge to identify and to look a little bit about what grows on it.
Okay.
Abundant it is in this life. Yes.
Is one DNA sample enough for both tissue types, or would you like me to do a DNA sample for the fistule and the bulbousy part?
So get as much as you can, get tissue from the bulb part
Uh-huh.
and internal. So in this way, it's not as much as contaminated as the surface tissue.
Okay.
So just get a piece of the internal material, mushy material of the sponge inside, and are you collecting a separate sample for DNA?
Okay.
I'm asking.
Okay. So we're port middle.
Port inner, yep. Looks like we have a successful sample collection. Way to go, guys. This is our first sample collection of dive 2.
Yep. So we're at EX1907 1907 D02 01B, and it's Oceanapia. Possible new species. It is undescribed. And we're going to be collecting DNA from both the fistules and the bulbousy area. And it is in the port inner box. Thank you, pilots.
We're at approximately 798 meters water depth, just for reference as to where we are and where that sample has been collected. That looks like another shrimp. We have some plated sponges with some fringes around there too. Can we also get a quick zoom on the shrimp and then also that purple feature that's just off to the right of it, which is dead center? Yeah, sorry, left. The one that's dead center, it looks interesting.
Yay for shrimps. He's pretty neat. I don't think this is the same species. He didn't have the swimmerettes like the other one does.
No, and he looks a lot smaller in comparison.
Mm-hmm. Nice reflective eyes, though. It's neat. You can see the little feelers he's got, or these sensitive hairs along his antenna. Help him detect food and stuff passing by. Cold? Ooh, there's a nice bamboo coral above.
Yep. That
Oh, we've got a
purple
sponge, I think.
I'm just not sure what it was.
And
so
then like a dying or dead
solitary
cup coral. Yeah. What's that yellow thing?
Yeah, looks like a
I have no idea.
something also
It
dying.
looks like a sponge to me.
The yellow one?
The yellow one, yes.
What about the
Get
blue?
slightly closer. I will tell you that it's either a calcareous or a verongid type. It will be
Verongid.
very different.
The yellow one you're
Yes,
seeing.
it could be
Yep.
these bright colors, bright yellow colors. You can find in calcareous sponges, which are usually very small, like this sponge is.
Oh, this little
So it
yellow
might be
one.
a calcareous.
Yeah. What about, Chris, what if you look at like 2 o'clock from there's like a yellow strip. You see that?
Yeah.
I don't know what that is. How about the blue one?
The blue one also makes me think of a thin encrusting Poecilosclerida type of
Poecilosclerida.
sponge. Poecilosclerida, yes. Beautiful combination there we have with that cup coral.
Looks interesting. It has almost like a burrow structure in it. Is that characteristic of that?
Yes, I'm not completely sure if those tubes that we see
Hey,
are,
pilots, can you swing 3
are
o'clock?
tubes from worms and the sponge is growing
Yeah.
over these tubes, or if they represent canals of the sponge. So,
And then down a little bit.
It could be both cases.
Yay.
I mean, I don't see it clear enough to decide if it's a very thin veil encrusting sponge that is growing over leftover tubes from other type of organisms, serpulids or other organisms, or if it's part of the structure of the sponge. Oh, there's a really nice cookie color sea star.
Yeah, we found a goniaster, right?
Very brightly.
Nice, thank you. So we're about to zoom in on, is this a starfish?
Yeah, so he's so cute. Yeah, the cookie cutter sea star or biscuit sea star, it's a goniaster or goniasteridae is the family. I don't know the genus and species of this guy. If anyone out there does, please chime in. Sure. Uh-huh. We have a Facebook question. Let's see.
And that question is from an Anthony Nairhood on Facebook. Why do animals you encounter hold so still most of the time? Is that they're scared or curious? I would think seeing the sub come down with lights and lasers would be enough to scare anything away.
Well, the modal, we usually do scare a lot of the modal fish away, so we don't actually see them, but they're gone way before we get here because the most ROVs are quite loud. If they're sessile like this, they actually can't move. They can't physically, well, this one can, but it moves very slowly. But most of the gorgonians, or all the gorgonians, the corals are actually stuck to the bottom. So they're considered sessile. So they are, once they find a place to hang on, they are glued there forever until they topple over or die. But these small starfish and stuff don't move very quickly. They also can't really sense us. I mean, they can probably sense our presence, but they don't have eyeballs, so they can't see us.
But even for some of the features, and correct me if I'm wrong, Stephanie, a lot of the animals and stuff that maybe the shrimps and whatnot, they can't really see all that well.
Correct.
And so a lot of the light presence that comes from the ROV and the lasers, they're actually not really detecting. And it's something that
Can we zoom center
we can also
screen on
take
the
into account
starfish?
for. Maybe that's why they're not being scared off so fast. But it's something that we're also keeping in mind about. So a lot of the fish and whatnot, the ones that do stay stationary, generally they don't. Well, like some of the fish we saw earlier, they don't really see the ROV maybe as a predator, considering that they haven't seen anything like that, or simply they just can't see it. Period.
And then Scott suggests possibly they're doing it to conserve energy, so they're not going to pick up and run until they're absolutely sure they're endangered. And we're actually quite far away from these critters. It looks like we're super close with the camera because we have an excellent zoom, but we're pretty far away. There we go. That's a nice starfish.
Nice starfish. Yeah, and you can see, based off what Scott said, we saw that earlier. So as we were zooming in on not the shrimp that we saw last shrimp, but the shrimp before that, he didn't move until the cutthroat eel almost had him and grabbed him. So he was conserving all of his energy until the last possible second to try to make that getaway. So unless we're actually physically trying to touch some of these specimens or animals, they would likely try to consume a lot of their energy, given that a lot of their food source maybe is not as regular as they would like to see, so they're conserving a lot of that energy.
Yes, it's hard to eat down here when there's no light and there's very few sparse specimens or sparse species.
Things to eat, yeah.
Nice, thank you.
Very interesting question on Facebook from Anthony. I would like to add for you guys that when we dive in areas that are protected, for example, in shallow waters, the fish don't even care about a diver, which normally in areas that are heavily fished by spearfishing, they're really afraid.
All right.
They seem to have amazing eyes, this shrimp that was still. So probably there's not any fear, there's not any experience to have fear to the presence of the ROV.
Right. They don't know. We don't look like a danger to them.
Yeah, life hasn't taught them that there's something to be scared of.
Like the Galapagos, right? I know all those animals they don't even care about people being there because they don't have any natural predators.
Yep.
Now this looks like a zoanthid covered dead octocoral of some kind. Yeah.
Yeah.
Looks like it might have been a black coral at one point, based on the base. Yep.
Even some sponges hanging out on there.
Oh, nice. Looks like
hydroids.
a living black coral in the background.
We'll go there next.
Possibly Liopathes, maybe?
With maybe an anemone hanging out on it. This looks like also there seems to be a barnacle attached to this guy.
And yeah, we've got Aphrocalistes on the base. Yeah, look at that barnacle.
Oh,
ooh, and a hermit.
A hermit crab.
So these are zoanthids. They're just taking advantage of this dead stalk.
Each little lump is its own animal.
Yep.
And you can see on the bottom end of the screen, some are happy and they're looking for some food with their tentacles out.
And some are retracted. Maybe they caught something already and are eating it.
Maybe. Or they're still sleeping.
The base, if you could look at the base a little closer. I saw Aphrocalistes down there. We got a worm right there, too.
Yep.
There we go, Aphrocalistes.
with a squat lobster.
Yep, so this is one of our target species for the Aspire campaign. Thank you. So we could, in theory, collect this
Afrocalistes.
if it's convenient, but it's not really in a great place because it's attached to this really stable base. So it might be difficult to get our arm in there to pull it. So, and there's also, I believe, a Lyopathes in the background, which is also a target. And I would think it'd be better to grab a piece of that.
Yeah. We might come across the Afrocalistes later.
Mm-hmm.
Sounds good, and I think this is indeed a black coral. We know that based off the black skeleton structure.
Mm-hmm. Was a black coral, right?
Yes. Giving it, in fact, its name. And we're about halfway up the slope of this ridge.
So this black coral is a target specimen. So you might want to hunker down and try and collect him. He's going to be really tough. Their skeleton's going to be pretty difficult. So you're going to want to get out those cutters and get a good snip of him. You don't need much. We really just want him for DNA.
Yep.
We know the species, or we know at least the genus. So yeah.
He looks to be quite robust itself, and he has an anemone attached to him.
So that's a fly trap anemone, and if I would call it Actinoscyphia, but I will get yelled at by the anemone people because they're impossible to tell. There's could be a couple of different species. So.
And what gives it its name?
So when it's normal, right now it looks a little, sad.
Yeah.
But normally they're opened up, and they're actually like this big round central disc, and they kind of look like they're folded, like a Venus flytrap, So it gets it's a flytrap anemone.
Interesting. Similar to the one I have at home.
The flytrap anemone?
Yeah. Well, the flytrap.
How are you keeping that alive? It's very cold here. Guys, there's also one in the distance. If you look up, whatever you want to do is fine with us, whichever one you want to grab. But if you pan up, there's going to be another more robust one in the background. Whatever's easier for you guys to get. That one in the background is quite large. If you're good, I'm good. Yeah, go for it then. Grab a piece of that.
And these look quite bushy overall, where they're growing different branches in all directions. There doesn't seem to be a favorable direction towards any of them. And I think, Scott, correct me if I'm wrong, that is typical of the Lyopathes.
Mm-hmm.
And
yeah, they're not planar. They're not like a planar shape, right, where you see a lot of that fan, the typical fan, right? This is more of a bush.
Or polyps growing in any particular direction.
And you can actually age them based, if you know how wide the base is, you can age them. And when they get large like that, they could be thousands of years old. They're really old animals. Pretty neat.
Really nice.
Mm-hmm. No, I just need a small snip because I don't need shape. I just need enough for DNA. So just snip me a piece and we'll be happy. And it's going to be pretty tough. So you're really going to have to, squeeze your cutters. And then it needs to be in its own bucket. So, yeah, we can't mix it with another specimen. I don't know if you guys usually do that or not, but okay.
And Scott, I was saying earlier that these branches look quite bushy and they're not growing in any particular direction or anything. So just wanted to verify if that was characteristic of Leopathes or not. Let these guys do their thing. Oh, Shrimpy.
How big is he? If you can flick your lasers on at some point, just so I can get his size roughly, but don't
Do
interrupt
your
what you're doing.
thing.
Just when you're free. Yeah, you're
That
good.
looks good.
Yeah, you're going to want to snip it with your cutters. Nice.
Well, it looks like you got a bit of the other branch.
Yeah, if you just pull away, it should just release. Or, yeah, twist your arm. That's fine, too.
Wow, these guys are pretty robust.
They are really hard. Yeah, they're really hard to... That's why they make jewelry out of them, right? Because they're really a nice, like, rock-solid skeletal structure. Yep, twist it up. Twist and pull. There you go.
Well done.
It looks like we upset the anemone.
Yeah. Do we know which box we're aiming for? Starboard inner, thank you.
It's going to be really light, so there's a possibility it might try to float away if there's any kind of current. So you might want to shove it all the way down into the bottom. To get it pretty deep into that bucket, and then open your claw, as it can just drift off.
A light but robust skeleton.
And then if we collect anything on that side, just be aware that they
Could
can
flutter out.
Yeah, it can fly out. We don't seem to have too much of a current today, so hopefully it's a non-issue, but I've lost many specimens that way. They just fly away.
And for those of you watching at home, on your main screen you're seeing the coral specimen, but the ROV pilots.
All right.
Are successful. Good job, guys.
All All right. Thank you. And before we speed off, can we get those lasers popped on real quick? Okay. So about 40 or 50 centimeters. Oh, maybe bigger than that.
Yeah.
Almost 60 or 70 centimeters wide.
Looks like we also have some dead bamboo coral at the base, a little bit of rubble.
And a starfish.
Starfish, yeah. This guy has some zoanthids piling on what looked like a dead stalk off to it as well.
Looks like we got another bamboo coral over here.
And more stichiopathes off to the, just off screen now.
There's a nice
Oh,
layout.
there's even another one. Oh, that's the one we saw before.
Mm-hmm.
Ah, okay.
There's a big guy, yeah.
Wow.
Nice. He is huge. Jeez.
Almost as tall as the ROV.
Right? You put your lasers on for me? That's it.
I think they are on,
There they
yeah.
are, yeah. So he's
Ooh,
a couple
well
meters
over,
tall.
yeah.
Nice.
Wait, can we get a quick zoom in on that white piece too? Maybe it's another coral. Oh,
I don't know what that is. Hydro. It could be.
looks like a hydroid.
That's that pink stuff.
Yeah, that's what I'm wondering too. If it's the coral trying to eat the hydroid or
Or
maybe
it's
take
sloughing
back.
off.
Yeah, maybe it's decaying coral tissue.
Could be. We zoom in a little more if I can. Yeah, it looks like it's sloughing off.
Yeah. Well, we know some shallow water corals try to release some digestive juices to try to, as a way to deflect any other corals encroaching on their space. I'm not sure if that's any characteristic of any deep water corals, but maybe, or maybe it is just dying tissue. And Scott France on the chat says the Leopathes colonies off Hawaii have been aged to over 4,000 years
nice.
old. And this one, based off the size, we might have a competitor for that kind of age structure too.
If you guys can just put lasers around the base, I know that that's how they age them. I don't know this. I don't know how big they get, but someone else does. So for the future, if someone else comes and looks at this, they can definitely tell how old it is. Just to give it a quick laser. Yeah, just to give it a liquid lasering. Okay, that's good. About 10 centimeters on the wide area, and probably 2 or 3 centimeters on the
Stalk
on the
itself.
stalk. Yeah. Thank you, guys.
And we have some bamboo corals potentially sitting around it. This guy is beautiful.
That is super cool.
So Scott also chimed in on that tentacle before that we were looking at that seems to be have the hydroid at the end of it. And it's a form of a sweeper tentacle, Scott says. Basically, the colony has been damaged where the hydroid is growing, and the adjacent polyps are developing defensive tentacles that are likely heavily studded with stinging cells. So they actually are quite similar then to the shallow water corals in that they create these tentacles and form these tentacles that are stinging and try to use them as a defense mechanism for any other biology or other corals encroaching then on their space. So this is really interesting to see down in the deep water. I didn't know that was part of, that could happen also. Generally, with so much space down here, you think that wouldn't really be a problem.
Right? There's plenty of places to live, right?
But
A
I
lot of
guess
barren
trying to be at the
spots.
highest space, at the highest point, has its
Yep.
pros and cons. Thank you, Scott, for that information.
So yeah, we're looking at about a 45-degree slope here. Maybe a little bit higher.
With some possible cutthroat eels in the background. Another one of those wedding glass sponges. Those are
Heterotella.
quite common in this area. We've seen quite a few.
Bamboo corals, too. A lot of these white ones are bamboos.
And despite the fact that we are seeing coral rubble in this area, it doesn't seem to be nearly as pronounced as it was at the base or even at the last dive. That's just an observation. Maybe they're just tumbling down the slope. Maybe they just can't really hold it. And it seems to be, look, we even have another Leopathes in this area, excuse me. And
No part of it's dead and encrusted with zoanthids.
half of the base seems to be dead.
We have a different colored pink coral here at the base.
Yeah.
Not sure what that is.
We do have some paramecia on the list. I don't know if that would be anything related to a paramecia.
I don't think that's the same kind.
Well, when we're done looking at the Leopathes, ROV, if you want to get a quick zoom of the pink coral.
Yeah, it might be bubblegum coral.
Which is also on our collection species list. Paragorgia arborea is that bubblegum coral, and correct me if I'm wrong, the coral that had the oldest coral that has been ever collected was Paragorgia arborea.
Oh, I didn't know that. That's pretty cool. How old was it?
Like
Remember?
Scott said, well over 4,000
4,000.
years old. Again, correct me if I'm wrong, but that was sort of a tidbit that of a while ago, but I don't know if that still holds accurate if we found a coral that has been dated to be even older. But the base was the size of a tree trunk, and it was taken off the coast of Alaska.
Scott, would you call this Paragorgia arborea? Because it's a. I don't want to collect something if we're not sure.
Yep. It does seem to have the knobby texture of it, but I can't really tell with the tentacles out.
I might confirm
Seems
the
to be
ID
happy.
before we, you know.
This is Paragorgia, but not arborea.
Okay, so same genus, wrong species. So we are not going to collect him.
You live for another day, coral.
And then we've got a cool spaghetti-armed ophiuroid.
With quite a large hole at the base of it. Is that
Yeah, I don't know what that
typical?
is. No,
I
I've
don't
never
know.
seen that before. Oh, it's the gap between his legs, right? Is that what we're looking at?
Yes.
Yeah. I'm not sure of the species.
So Scott said he doesn't think that Paragorgia arborea does go this far south. But if we do happen to see it, that would maybe be a first for this area, and we'll definitely then aim to collect it.
Scott, you know how far south it does get? Because then I can keep an eye out for it on the next few days.
And for those of you watching at home, so we've had 2 biological collections, and we only have 2 spaces left for any biological collection. So we're definitely going to keep an eye out for whatever is necessary to collect, but just to keep that in mind, we still have quite a few hours left of this dive, so we might come across something really cool later on that we might not be able to collect if we use all the space up. So just to keep that in mind. But as we move further upslope here, we're starting to see a more dense network of coral systems in this area, and we have come across quite a few variety of species in this area, and quite large. So generally speaking, usually the larger the coral is, the generally older it can be. And we're coming across a lot of black corals and bamboo corals and possibly some primnoids. And this is overall really nice to see. There's possibly another larger black coral off in the distance there, about 11 o'clock. And we see at this black coral here, there seems to be, it's mostly dead. It's still somewhat alive, but it seems to be kind of coalescing, some zoanthids and maybe some other of those fuzzy trees that Chuck Messing talked about earlier, which are foraminiferous. And we can see all of these individual colonies here, but it seems to be quite a bit of a coral forest. Even though we have seen quite a few of these isolated cup corals, which are of the scleractinian species, we haven't really seen any framework-forming scleractinian species yet on this dive, as opposed to a lot of the other maybe soft corals that were, one was left unidentified, but compared to the leopathes, the black corals, as well as the bamboo corals in this area. And here, speaking about earlier when I was talking about the overall substrate of the area, the morphology, here we're starting to see a lot more of that coral rubble in that area, where possibly at a more downgrading, higher degree slope area, we started to see more of that slabs and the sediment textures that are all around. But here at this relatively looking kind of almost plateau terracey area right in the middle of our dive as we're going up the ridge here, we're starting to see a lot more of that coral rubble accumulation in this area, which means that it is likely the slope, the degree of the slope that is not keeping the coral rubble in place, that it's likely tumbling down and going somewhere else. Can we actually zoom in on that little yellow coral off to the left-hand side of the screen? It seems to be like something we haven't seen before yet. And we have some more sponges around that area. Yes, that was. Thank you. And I'm not quite sure on the species here, but if we just take a look at the overall morphology, we can see that the skeletal structure does look black. So I'm not the biologist here, but if someone were to chime in and to tell me what kind of species we're looking at here, just based off the overall of my basic knowledge, it looks to be like a black skeleton. And Scott Franks is chiming in. It's actually an octocoral, Acanthogorgia octocoral, and it looks really nice with its solid yellow structure, and each one of those polyps seem to have a little light-colored bulb at the end of it before you see all the tentacles.
So I think those little light spots are probably its mouth, its oral cavity, oral opening.
And the smaller one off that came right off screen, Scott is identifying it as a Primnoidae coral. Not sure of what kind it is yet.
Oh, the small white one? Oh,
Yep.
over here. And again, we're seeing this cone-shaped glass structure that we saw before.
Yep. That
This
is
like
a
cluster.
rhythm. Yep.
My guess would be that's probably an egg sac, the empty egg sacs.
Maybe.
But I'm not sure.
But there seems to be more coalesced here than there was before. And then there's this little guy here. Also very translucent in color.
A single stalk.
Well, it has one little branch
Yes.
sprouting near
Sparsely
the base.
branched.
Maybe it's a juvenile.
Plexurity, maybe unicella. Thank you, Scott.
So another fun fact that I'm also learning now, that not all black skeletons mean that it's Antipatharia. So these acanthogorgids also have some dark skeletons. So that's another characteristic that we have to watch out for, that just because it has that black skeleton doesn't automatically mean that's Antipatharia. So thank you for that, Scott. I've learned something now today again.
Right, so it's easier to tell by that solitary polyp that sticks
Yeah.
out, right? So, or the one that sticks out.
So that's a good way to keep an eye out on things and to help with identifications for you at home.
I think there's probably, I think we got a nice bamboo coral at 9. 9. There's the Leiopathes at 3. I think the bamboo coral at 9 is probably a better, because we saw a couple Leiopathes already.
9 o'clock.
Oh, sorry, swing to 9 o'clock, so swing left. And then keep going left.
Yeah, we passed it earlier.
Oh, you already went past it?
Well, we didn't get a zoom in on it,
Oh,
but
okay.
it was something that caught
There we
my
go.
eye.
That
Yeah.
big white one right there. So we have a planar. So it's fan-shaped.
And generally, these are very flexible, so they can bend and sway with the current systems, and it looks pretty stationary. Almost all of the corals here look pretty stationary, so that just gives another indication of how the current system in this area is behaving.
Yeah, it's just as close as you guys can get. Don't fuss too much about it. All right, I do not know what this is. Oh, bamboo coral. You can tell by the base. You can see the
You can start to see some banding.
Yep.
And even a lot of these wispy corals in the background here, Scott is identifying that they could be a lot of different kinds of species of bamboo coral just in one area. Seems to be doing quite well. Look at all those tentacles and those polyps. So this is similar to what we were seeing actually in another coral that we zoomed in earlier on, that even the tentacles have their own little pinnacles and on them.
Tentacles have tentacles.
Tentacles have tentacles. There you go. Can't just have one.
Great way to catch food, to have multiple tentacles on your tentacles.
Yep.
Awesome, guys. Thanks. There's another Leiopathes in the background.
So the tentacle on tentacle features, Scott's saying that it's also highly characteristic of octocorals. So that's something that we can also help to identify certain coral species if we can appropriately get that zoom in on them. So that's how a lot of corals now are trying to be identified via their DNA. But before, it was really hard to identify things actually on camera. So we can't just point at a coral and say, hey, that's that from that far away. A lot of these corals are then identified based off their polyp structures, off the corallite structures, how many septa they have.
Branching.
And the branching overall, the color of the skeletons, how the branching is forming, the direction of the polyps and where they're facing. All of these are kind of a combination that are needed in order to get an exact species identification of these corals. And I know from being at home on shore as the geologist, I'm not entirely familiar with all of these. So when I would log into the chat room and say, like, why can't you identify these corals? We actually, now I'm understanding how much effort it takes to actually go in and say definitively an identification of them.
All right, so a lot of times we'll see these repeating what we think they're the same species, right? So I'm under here underwater watching this footage, and we go over large swaths of area, and I try to identify as much as I can via video. And there's just some you just can't ID from video. So if we can, we collect them. We collect small specimens of them, and we actually send the physical specimen out to a taxonomic expert who will sit down and pick through the details of each specimen and give us an accurate ID, and then give us the information back so we can update our data.
Yep.
But without a specimen in hand, it can be pretty difficult to determine the difference between a lot of these, crabs, lobsters, corals. A lot of them, they're difficult to, all kinds of phyla are difficult. Sponges especially are difficult to identify unless you can actually physically look at them underneath a microscope.
Yep. I mean, it's likely we can maybe get down to the genus level on video, but per actual species, there's a lot that goes into actually identifying them.
Right, so like this one, Liopathes, right?
Yeah.
This is a Liopathes. It's an obvious one. I don't know the species, though, so none of that comes with the in-hand sample. So yeah, black corals do have 6 tentacles. It's just that you can only really see one at a time.
Yeah.
The way that they grow. So it just looks like one little tentacle poking out. Versus the other octocorals, or the other soft corals, which you can see all the tentacles sticking out. So that's one of the characteristics.
And it's characteristic of octocorals to have 8 of those pinnacles of the tentacles, which gives, generally, its octocoral name, octo being 8.
And hexacoral being 6, so the hard corals are in the hexacoral. It looks like we have a fish at 3 o'clock. I don't. Sure, whatever you guys need to do, we're good.
Where's that fish?
Right here underneath this rock.
Oh, good ID. Slightly above 2 o'clock now.
I only got it to fish, so let's not give me too much credit. There's another fish. It looks like cutthroat eel maybe at dead center.
Yep.
These heterotella, these wetting sponges are really common.
Yeah, in this whole area.
It looks like we're getting some white, either coral or some white, sorry, I'm pushing you, I'm sorry. White gorgonians on the left.
And even though we're not at the ridge crest of this whole feature, we're starting to get more and more dense populations of these corals, and not just of of a whole variety of species that we're seeing in this area.
This one's an Anipatharian right here at 7. At 7 o'clock we've got an Anipatharian. Yeah, sure. I mean, I'm not sure what it is. Tenacetipathes, maybe. Guessing. A lot of these are tough. But it's a different type.
Yes, that's another type of black coral.
And these polyps look very fine, more brush-like than what we've seen previously, and especially what we also saw in the Leopathes.
Yeah, so you can see them really separated and fine and thin. We call this the Australian pine tree coral, right? Because it looks like an Australian pine tree. If you're from Florida, you know, or Australia, you know what that means. It looks like a pine tree. You know, pine branches. You can
Well, we
see
only get most of the pines in, well, the larger, yeah, you're right, the actual Australian pines, but you're starting to see more of those small little pinnacles on the actual tentacles themselves, or are those the individual polyps?
So those are the individual. So if you look along the thread, you can see the individual polyps. They're very tiny, right, sticking out each direction. So each one of those is a critter. Yeah, and Scott said Tennessee is reasonable for this guy. I think here's a crab.
Oh,
A
yeah.
crab or a squat lobster with his arms out, waiting for
Trying
some
to
food.
make as much space as possible.
Pretty cool.
And wow, he has polyps going all the way down almost to the base, just a few centimeters off the base, and you can already see it start to branch out and form these polyps. Now, just in comparison to the other corals that we've seen in this area, I mean, these polyps are almost microscopic in view compared to some of the larger ones where you can actually see the individual polyp on the corals themselves.
Yes, they're just teeny tiny. Scott said you can try to count the 6 little knob tentacles ringing in the mouth, but I think we're too far away.
That to you, Scott.
We're too far away now. If you guys can swing up, I think there's a crinoid above us. That purple one. Yep. He's stuck on the Leopathes.
Oh, yeah. Oh. Cutthroat eel.
Hey, buddy.
Seems to be having a good time.
Right at 3. There you go. This purple. Yeah.
So that's the one Chuck Messing was referring to earlier, that they're usually characteristic in purple, and
He was the yellow one. He was wanting us to collect. So.
yeah. So if we see one that has 10
arms.
10 arms, then give us a shout out over any of our social medias or on the chat room, and if we don't see it, maybe you guys will. Definitely point it out if it seems to be any of the colors that are not purple. It's definitely something we'll want to see and try to possibly collect.
And we see our first stylasters here. Those white small bushes down on the real tight to the ground. They're small and short. Next to Stichopathes, which is the orange whip coral. There's another one. The Stichopathes is a black coral.
Black whip lace coral, if I think it's its common name.
Lace coral.
Lace coral. And an observation that I've made in previous dives relative to, say, this one that we've mentioned now, stylasterids, that usually you start to see a lot of stylasterids be some of the first colonies of the somewhat coral family in the area until maybe some other framework systems start to overgrow it, and it tends to die off or be just overgrown by the other framework systems. Is that something that's quite common?
Like the stylasters, like the first type of coral in the succession order, like when the stuff starts settling out. Yeah, that's not uncommon. That's not uncommon, I don't think.
Okay.
Where we actually see more. I think it also has a wider range of tolerance for habitat. So, like in an actual shallow water coral reef, they usually grow underneath the rocks hanging down. And they range, like they're all the way up in the shallow water reefs, right? The stylaster, not necessarily the species, but the stylaster family. It ranges all the way from the shallow water all the way to the deep, and it grows in habitats that aren't necessarily conducive to our more like reef builders, or like our I don't want to say reef builders because we don't really have reef builder
That's what
corals
I'm
down in the
saying.
deep. We have a hermatypic coral, which is a non-reef building coral, but they grow those big bushes like of I'm blanking.
Like a
Come on.
stony coral framework
Yeah.
system?
Yeah.
Okay. Well, I mean, generally you can still call those framework forming corals because they do have those intricate complex branches that spread out, and like Lophelia pertusa or Madrepora oculata or even oculina reefs, like they form these large, robust reefs that are usually the foundation for a lot of these mounds that I study that we start to see sprout over and grow over thousands of years.
Right. Oh, nice.
Sounds good. Not sure what you saw, but go for it.
I think it's just this hole.
Okay. Looks like to be a just slightly steeper area. Yeah, they were still slightly pinkish. I'm not sure if that qualifies under the really of what we're trying to collect. I think we're going more for those really yellow intense crinoids.
We have Lophelia. We have a little bit of Lophelia. And then it looks like
Lophelia sightings.
we also have this really large mound up here, which might be some type of tilefish mound. But here's our first Lophelia sighting.
If we can get a snap zoom on that. Straight to that Lophelia reef right at the base, the center base of the frame. And then we'll scale up to maybe that live one that's up on that little slab. But these were that framework structures that we were just talking about now, that these coral reefs, I would still call them framework forming systems. Maybe ahermatypic is the better terminology for it, but in the geology world, these are still very much the framework forming systems that then trap and baffle the sediments to, over thousands of years, grow these large carbonate mound features that we see kind of populate a lot of the Atlantic, closer to the coastal regions rather than maybe at the ridge. So pretty.
So this brown is dead. Is dead standing. There's a piece of
Bamboo
bamboo
coral
corals
attached to it.
attached to it.
Tried to grow on it, didn't make it. And it's being already recolonized by a lot of different corals. If we could maybe pan to the right to get a view of that live coral.
And we've got a glass sponge, a Hexactinellida. Possibly Feria, but I'm not 100% on that guy.
We have some soft coral also at the bottom there.
Yep, so look at the yellow.
Ooh.
Nice yellow. Yeah, and primnoids on the right, on the left. So we got...
Is that yellow normal or?
I've seen it before, but I don't know why it's different color. It's definitely alive. You can see the polyps.
Yeah, we see more
Usually
of the
white.
polyp structures on the yellow sections than any of the white. I think the white sections are, yeah, there just seems to be this coral is kind of in surpassed its prime. Let's put it that way.
The yellow part looks healthy, though. I'm not sure why it's yellow, though. Scott, do you see a lot of yellow Lophelia? Oh, the yellow is the only part living right now.
But usually a lot of Lophelia is characterized by maybe slight pink at most or white. I haven't really seen it this robust yellow in a while. And we have a crinoid stalk sticking out of the dead part of the Lophelia.
Oh, he says no, but I do see yellow Inalapsamia, so it might be Inalapsamia, not Lophelia.
Oh. Yes, please, if that's possible. I know it's kind of in the center of a lot of that coral. maybe coming from the other side. Maybe it is an elopsomnia rather than Lophelia. Just to go and show you again, folks, that at home that it is incredibly difficult to identify a lot of these corals right at hand. Oh yeah. We'll get a closer look after to see if that's the guy we're after. This actually looks like Lophelia. This guy is white. He has tentacles all throughout with some elenopsomnia in the background there of that yellow coral. But this does look more characteristic of Lophelia, I would say.
Lots of gorgonians around him too. Yep, you can see the polyps sticking out. So he looks pretty healthy.
Looks like also maybe some sort of gastropod, or no, that's a barnacle maybe on the side of it.
I'm not sure. We have to focus a little better on that. And then the hydroid.
Looks really good.
Cool.
His polyps look very kind of web structured like. Is that normal for that, or are they relatively smooth?
No, they have those little like dots, right? It's probably its stinging cells. Is nidae. That's how they catch food.
Yep. And I'm seeing over and over again off to the right-hand side that little stalked anemone of some sort looking.
Oh, this guy. Yeah, I think that's a hydroid.
That's
a type
hydroid,
yeah.
Okay.
No, it was the hermit crab that we were, who's right here.
Potential barnacle.
Yeah, I think it's a hermit, though. I can see some
The shell.
articulate legs. Yeah.
Okay. That's another one of those yellow corals that we were seeing earlier. I think we're good, thank you.
Yeah, we have a yellow. Maybe an alapsamia on this one. And we have that stylaster underneath this outcrop here. It's really lace-like. There's some brown cones. Oh, if we're moving on, don't worry about it. No, go for it. No, you can move. That's fine. Let's look at this rubble pile up here if we're heading that direction. That's okay. That's the wrong species. So it looks like we're at the top of this feature, but I think according to the multi-beam, we have quite a bit to go. So we might just be on a little piece of, like a little bit of the shelf. But this big pile of coral rubble is possibly fish created. My guess is a tilefish of some type. Yeah, likely the fish dug it out. Yeah, if we can get a good look down in the hole. So a lot of these tilefish mounds will have like a front door and a back door. If it is tilefish, it may not be. Yep, it's a burrow. Tilefish tends to be very shy, so the likelihood that we'll see one is slim to none. Usually you catch them in the distance and they disappear into their little hole. So that could possibly be a front and back door to the same mound, or it could be two mounds. But this Lophelia rubble probably came from down when he, oh neat, when he buried him, when he dug it out. So likely this is a giant pile of Lophelia, which would make this a lithoherm or bioherm. At least a couple hundred years of, a couple thousands of years of piled up Lophelia rubble. Nice. My guess is this is the front door, and that other hole on the left, on the right, is the back door. Cool. Kim went to lunch, so otherwise I'd tell her to tell me about these rocks, what this is made out of. We have a nice starfish at 2. Getting a little more outcropping up here. Yeah, so Mike asked on the thing. I believe they pick it up with their mouths and, carry it forward and spit it out to make those mounds the tilefish do. Chris asked where else have we seen these mounds before, and we see the mounding where the tilefish. There's a lot of sand tilefish off the coast of Georgia. We also see them in the soft sediments heading north of, so between, off the coast of Florida, I guess. And then the hard substrate ones we see in the mesophotic areas, mesophotic MPAs that we go to sometimes. Ooh, look, we have a sea pen. We have a sea pen dead center. We also see sand tile on Pulley Ridge, too. Oh, maybe this is Antipathes bipinnata. Yep, that urchin is called a pancake urchin, and it's extremely toxic. Do not touch it. I think Antipathes bipinnata is one of our targets. Oh, sorry, Bathypathes alternata. This is a type of black coral. Hey, Scott, what'd you call this alternata? Is it worth collecting? It's one of my target specimens, if it is Bathypathes. So cool. Oh, we've got a cool shell underneath them, too. It's so pretty. Yeah. Look how shiny he is. I think, guys, we're going to grab a snippet of this, or at least prepare to do that. Let me just get confirmation from Scott. And again, this is just a DNA sample. We only need a tiny snippet. Okay. So in order for us to identify this to species, we need to get a really good shot of the main axis. and how the branches come off the center axis. So if you can kind of line up a little better to see the branching there, that would be helpful. Whichever way you need to do that. So yeah, this the branches alternate, right? As opposed to coming off of the same exact spot, they alternate left and right. I think we're gonna grab this guy. I only need a tiny piece. He's probably gonna be really stiff, just like the last one we got. So you're gonna want to snip it with your coral cutter, and he's also gonna be very light once you put him in the box. Thank you. Just a tiny piece. We don't need the whole thing. I mean, if you grab the whole thing, we'll preserve it, but we don't need it. We know the species. I just need a tiny piece for DNA. Yes, and you're going to have to snip it.
We're good?
Awesome. Thank you. Port outer box. All right. Thank you. Before you fly off, can you flick those lasers on for me just so I can get a quick size? So about 30. Okay. Thank you. Okay.
So, folks on the chat room here, now that we've kind of driven around this area a little bit and quite a large coral coming in the foreground there, was this something we would call a dense and diverse deep sea community? Is that a box you think we can check on this dive here? Thanks, Ken, for helping us identify that shark we saw a little bit earlier in the dive. Super appreciate that.
This looks like a really robust coral over here.
Chris wanted to know what the question was again. If you could repeat it, she was.
Yeah, I think one of the things we look out for on these ROV dives is if we get into an area we would determine as diverse and dense, and that comes with certain quantifying descriptors, so many species in such an area. So just wanted to see if we were there. I mean, this has been a really exciting dive so far. A lot of different types of coral, lots of hard bottom. I think in the shots we've had many species. So just wanted to chime in there. It's just something we closely track here for our partners out in the wide world.
And thanks for that. And sorry, is anyone chiming in?
Yes, I just wanted to ask if you were interested on us giving an estimate of how many different sponges, for example, we have observed up to the moment. And so I could mention about the soft corals. So if you believe that we give you some input about that.
So the different variety of them, there's definitely a high biodiversity in the area. I couldn't put a number on how many different types of species we've seen. Definitely quite a few different kinds of bamboo coral, different kinds of scleractinian isolated cup corals. We have seen quite a few of the sponges, which I'm sure you've seen. One that potentially could be a new species. We've seen a variety of fish, including those different sharks. We saw spotted fish as we were nearing the bottom, as well as later on gray and white spotted fish. It's hard to put a number on how much of a diversity we've seen, but definitely a lot more diversity than we saw yesterday, and hopefully we can continue that.
So we have Ken Sulak weighing in on the chat room here. He was correcting one of our earlier observations as we identified here on the ship as a cutthroat eel. But Ken's actually saying, no, it was actually a congrid, but many of the other eels that we saw today were indeed the Synoptobranchus cutthroat eels.
And now here we can see a lot of this more coral rubble features encrusted with that ferromanganese crusting, which has a lot to do with the overall ocean chemistry in the area, reacting with the coral skeletons and depositing this iron manganese crust that gives it that dark looking feature. And it's, speaking scientifically, it's quite a pain to try to remove in order to get to the actual sample of the coral skeletons that we want to see. If we could possibly then age date them or use them for specific isotopes or mineralogy, we have to get a lot of that crusting off it before we can actually do the work that we need to do in order to find out a lot of the characteristics that go along with how the coral once lived and how it formed. Now here we have some blank spots of some maybe not so abundant coral species, but right up ahead we're starting to see, I don't know if we're backtracking, but we're starting to see a lot more diversity again.
So what was interesting from yesterday, Kim, and you've spoken a lot to this so far leading up to this cruise, is what's driving the coral colonization here, right? Is that the coral larvae are finding hard substrates to settle on. That hard substrate, maybe in this case, would be the relic Lophelia, or what mostly seems to be Lophelia skeletons, or is Lophelia settling on some hard substrate or the underlying sand grains, and then the Lophelia dies and it gets recolonized, right? Really sort of an interesting evolution of the benthic ecology itself over time, one would presume.
Yeah, absolutely, Mike. And there is a lot that goes into that. And part of what I study is that evolution period of how these mounds actually form over time. And hopefully we'll get to see more of these isolated mound features in future dives for this expedition. So make sure you tune in when we get to go to those dives. But for the overall purpose, we're starting to see that it doesn't take very much for a coral to start to colonize in a certain area. It does need specific ocean chemistry as far as temperature, salinities, and oxygen levels go, but really just that small hard substrate that the initial larvae needs to start to settle on can start to promote and have these corals then proliferate in a certain area. And overall, when we see a lot of this area that starts to die off and a lot of the coral rubble, that forms a new hard substrate for new corals to be part of. So that's just an overall life cycle of it. And some corals do get to grow over thousands of years. Like Scott said earlier, some of the Leopathes that have been discovered are well over 4,000 years old and still living. So these creatures can grow very old, but a lot of what we're talking about with coral rubble and these life cycles of evolution happen over thousands, hundreds of thousands of years, and we're talking over geologic time where these mounds need time to form and to grow, and that's given that nothing else impedes them from actually developing over this time.
I mean, there's just some really stunning examples today of how this secondary colonization, right? So originally we were targeting some of these, live Lophelia mounds. Well, we're seeing a lot of, Lophelia skeletons and being, recolonized by these black corals, marinoid corals, and octocorals and hexacorals, and of course we have all sorts of associates living on these corals. I think definitely a crinoid up to the left here, hanging out on this black coral.
Yep. It looks to be that pinkish color that Chuck was speaking to yesterday, that the majority of the specific maybe species of crinoids that we're looking at here, these 10-armed species of crinoids, do have that purple pinkish color primarily, but we're going to possibly, if we do see any of them in a bright yellow, that is a sample that is of very high interest to collect. So hopefully we'll get to see one of those, just like we saw last at yesterday's dive. However, it was inside of a drift ghost net, so we didn't want to mess with that, especially with the ROVs for safety reasons.
And just to to take a step back here, contextualizing where we are today, we're on this about 10 to 15 long kilometer ridge. It seems to mark the eastern extent of what is known out there sort of colloquially as a Million Mounds area. There's this area in this habitat area of particular concern, the Miami Steson Terrace here. Excuse me, the Steson Miami Terrace. And to the east of it is, if you look at the mapping data or the transit data that we have, it's relatively flat. It's relatively featureless until you got to the middle of Blake Plateau. You have some isolated mounds. And so one of the dives we're planning on in the future of this cruise, down the line here, dive 15, is we're actually going to try and do an ROV transect from where we're on this to the east of here, some flat bottom, and then head into these areas which have all these mounds and features, and really look at how that biodiversity or the benthic communities, how they change laterally over that extent.
That's right, Mike. And you need, excuse me. And even though Shirley Pomponi wrote in the message in the chat room saying that there has been an evolution of what we've been seeing of the overall substrate in comparison to also yesterday's dive, where before we were seeing a lot of that rubble features with maybe a few outcrops here and there seen of the karstified slabs, and then also a lot of sometimes those exposed sediment grains which created those ripples from the current. But today we're actually seeing not only a lot of coral rubble, but more of those karstified slab substrate features, that hard ground of which these corals likely desire, and it's excuse me, and it desire as well as used to promote the foundation and grow up on.
Scott, could you help us in the chat room where you saw that pinkish primnoid coral? Was that down to our bottom right there? We're just in the middle of a shift change here, so didn't want to pass anything if you felt we'll sort of stop and look at.
Yeah. And just to reiterate, in the beginning of the dive, we did see a couple sharks when we reached the bottom, and it was extremely exciting to see. And Ken Sulak chimed in the chat room, and he was able to identify some of those sharks as Squalus cubensis, the Cuban dogfish shark from the family Squalidae. I'm sorry if I end up saying these names wrong. Biology, the scientific names are pretty tough these days, but that was one of the more interesting sharks that we definitely saw swim across the bottom, and we were able to get a quick view of it, and hopefully we'll get to see maybe more along these dives in the future. And here we're coming up on that pinkish coral again.
So Ken, super appreciate you continuing to chime in here. So he's detailing on the chat here some other fishes we've encountered today so far. First up, the gray cod-like fish with very long white pelvic fins. That's Phyces chesteri. The otherwise common name is a long-finned hake. Typically rests on the bottom, semi-curled, often with a shallow pit that it digs or hiding under a ledge. The next fish we saw, the small rat tail, was family Microridae, Nizumia bairdae, and again, I'm doing my best on the pronunciations here, in typical oblique posture, nose to the substrate. And then the next small black-spotted whitefish sitting on the substrate in typical fashion, again, is the Acropomatidae, Synagrops bella, or otherwise known commonly as the blackmouth bass.
And we'll try to get back to that pinkish color, but there was a shift change with the ROV dives, and we may have drifted a little further, so we'll try to get back to that pink coral. But if anything, the goal of this dive is to reach the ridge crest and potentially follow along some of those top mound features that we've been seeing before. Oh, there's that pinkish coral off to the right-hand side of the screen.
So with these dive plans, because we are exploring, it's often difficult to determine if what we're on right now is going to be some of the what one might describe the more rewarding parts of the dive, or if we make it to the top, then we're going to see some different colonies, some different benthic communities. So you always have to be careful about how many chickens or eggs you count in the basket before you actually have them in there. So, Pilot, we have noted in the chat room here, we think this is just another pink paragorgid coral with an ophiuroid typically associated around it there. I think we're good on this one as soon as we're ready to continue here. The one we were looking at was a bit taller and definitely pinkish in color. If we see it again, we'll give you a heads up, but thank you, Pilot.
Thank you. And we seem to have come upon a lot of this coral, almost forest-like, although not exactly framework forming. We do have a lot of these overall isolated coral colonies in this area, and we do what seems like we're starting to come upon some scleractinian bushes. If we can get a quick snap zoom on one of those white corals. Maybe a little tighter if we can go in. Thank you. And this might be either Enelopsomnia or Lophelia pertusa. I'm not entirely certain on the ID. But we do also have some of those isolated cup corals. We see some skeletal features right underneath that seems pretty definitive on the solitary coral. We have a few squat lobsters and another gastropod hanging out on this coral skeleton here. Oh yeah, thank you. And we're starting to see a lot of these corallite structures that are used to identify a lot of these coral species here. So each one of these polyps, when we have a scleractinian coral, and what the polyp comes out of, this specific center, is called a corallite, and that's actually the feature of where the individual animal of the polyp lives. And we can see that the larger little thin discs on the top are actually called septa, and those we can also count, see how many are large and small, depending on, then it can help us further ID the actual species of the coral itself. And we can see a little bit of the underside of a squat lobster right at the top of the screen there, and there's a little snail off to the back. Not seeing too many polyp structures here, but it does still look like it is living.
And just want to give a shout out to our video team here today. We have a nice compliment in the chat room, getting those images of the fishes earlier, particularly kind of the broadside still shots. It super helps being able to identify those. So well done to our video team on board here.
Yeah, filming those fish is not an easy task. Thank you, pilot. We're good here. There's a request to put on some lasers so we could see the overall size of the corals. Thank you. And we do see quite abundant of different species of corals, all in just one shot alone on this plateau of a ridge that we're currently slowly climbing. We have everything from crinoids to black corals to octocorals, a lot of bamboo corals. We have seen some scleractinian, Analopsomnia, as well as Lophelia pertusa. We've seen quite a few of crinoids, some being stocked, which is the first time that we saw them, compared to yesterday where we didn't see any of those stocked crinoids. But today we have quite a few. They tend to stay relatively small compared to the other crinoids that we've seen, especially the ones that are not stocked. We have again another Leopathes here with a pink crinoid on it. And a starfish underneath. It's an overall great shot of all the different crinoids. I think I'm counting 2 or 3 at most, with the starfish then at the base there. And it looks like these corals are actually finding their way on the hard substrate below some of the coral rubble that's there, maybe directly on the karst itself. And just 2 guys. Thanks, pilot. Ready when you are. Yeah, sure, while we're here. And there is another one of those sponges again. Afrocalistus beatrix. I know it's on the species list, but I don't think we're still going to collect these guys yet, considering we only have one bio box left, and we still have quite a bit of dive left, so we might come up something maybe more pertinent later on. But that's a great view of this overall, the starfish here. And not sure if he's currently trying to feed on something or not, but he seems quite bent. Maybe just hanging out. But what's also interesting is near the sponges there, we have what looks like kind of like a fan-tailed cup if we can get a tighter zoom in on that, that looks actually quite interesting. If anyone on the chat or anything wants to chime in on what this particular thing is, if it is its own cup coral, or if this is something associated directly with the sponge, I'm not entirely sure what we're seeing here. But it does look to have some skeletal structure, so maybe it is a cup coral in the end. Not sure. Thank you, ROV. And we're getting from the chat room that they think it was also a cup coral, not necessarily associated with the sponges. But the sea star did look like it was feeding. There was a small little branch protruding from underneath it that was maybe a leftover of a bamboo coral that it decided to stop by and have for a snack. They are one of the main predators of a lot of these corals. So usually the ophiuroids or the brittle sea stars kind of hang around it and are more symbiotic with the coral species, but these sea stars are actually a predator of them and eat the live tissue. Oh, good. Just again, we're seeing a wide variety of a diversity of corals just along the slope here. And maybe at the ridge we'll get to see even more. We're close to the top now at one of these small mound features. If you want to look on your third screen, on the camera screen, we have a quad view of some different screens that you're able to see, and one of them includes a bathymetry map of the overall region. And we're close to the ridge and the crest at one of these ridges. So we've been going upslope from the basin all the way to the top now, and we're nearing the top of one of these features. And we'll be down tracking further south and kind of scaling over a few of these more ridge-like crests that we'll be seeing more in the future.
Yeah, we've got a lot more coral here than before lunch.
We did.
Looks like we have another Afrocalistus sponge. Okay.
Okay, sounds good.
Can you...
Maybe more on the port side. But what is that bulbous looking sponge on the end of that stalk there with the crinoid on it?
I don't know. We'd have to ask Chris that one. We've got some kind of ribbon fish too at 9. You see it?
Oh, just slightly in the light that was there. A slight glimmer.
There he is behind us.
Oh, yeah.
There he is. Look. Ooh.
Ooh. Now, is that bioluminescence or is that a reflection?
I think it's reflecting. We're too bright to see the bioluminescence, right? Because we've got these big bright lights on it. So it would wash any bioluminescent light out. So it's probably just a reflection. I think this is not a sponge. I think this is a retracted anemone.
Oh, okay.
Likely one of those fly trap anemones.
Interesting. And it's just one, or is it two?
I think it's just one.
Oh, they can grow quite large then.
Yeah, they get pretty big.
And then the rest of this coral seems to be colonized by some zoanthids and whatnot.
We do have some cool glass sponges, though, on the right at 3 and 4 o'clock.
Yep. We're seeing a higher diversity of sponges scaling upwards towards of these slopes, and especially compared to yesterday's dive. We have a lot of those wetting glass sponges. We saw possibly a new species of them. A lot more of the demo sponges, the diversity of the demo sponges we're actually seeing in this dive.
So this is a retracted anemone.
Oh, wow. I'm trying to look at the surface of this organism, and yeah, it could be perfectly a retracted anemone, like you are saying. Now, it has a very small attachment to this octocoral, the skeleton of this octocoral.
Yep. And Scott says in the chat room that it's also on the left-hand side of that coral, we see a stoloniferous octocoral overgrowing on that side. So it's taking advantage of the already dead skeletal structure and kind of planting itself on there and growing. No, that sounds good. We do want to get a few of these pinnacles in there in the dive. We have more Antipatheria coming on screen, intermixed with a lot of this bamboo coral and other octocorals.
If we can look, there's a white sponge at 3 o'clock. Which I have seen before. Now it's at noon.
Yeah,
Yep.
the background.
Yep, the back.
The more column. Next to possibly an Analopsomnia coral, given that we know about its... Oh, look, we have maybe another shark here.
Oh, nice.
Or fish.
Yep.
Ken, if you're still on the line, feel free to call in or chime in the chat room.
So maybe a chain dogfish?
He did identify a dogfish earlier. Cuban dogfish.
Cuban dogfish.
So maybe this is of the same species, although it doesn't look similar to what we really saw before. It looks maybe different, but it could have been just different in juvenile or whatnot. So Ken, if you're around, please chime in to let us know what we're looking at here. It's quite long. Do we have lasers? Oh, they're there. It seems to be about 40 centimeters long, with a mouth on the underside and some pretty wide finned, and it seems like it's able to stay stationary, sucking in water. A little brown with some black, I don't want to say spots, they're more squares, it's almost checkered.
I would just go with dogfish for now. I don't know much else.
I think we're at dogfish.
Yeah. Oh, we got a cool shrimp underneath that rock too.
Yeah. And a few little more shrimps peeking out.
Some Afrocalyptes.
Maybe he's on the hunt. Ooh, is he going for it? I don't know.
Eat the shrimp, buddy.
We're getting a really good look now at the lateral side of the shark. A really large shrimp in that burrow there. Oh, that's a better view actually with the lasers. Thank you. Maybe closer 30 to 40 centimeters. Just hanging out. Not sure if he's resting on something or if he's
Yeah.
on purpose putting that tail up.
I think he's resting. It looks so weird. It's a very odd posture to
Yeah.
be sitting in. It can't be comfortable.
Up close to one of those Afrocalistus sponges. Wow, really cool find. It is indeed Shark Saturday. Post that on your Twitter.
Right? Shark Saturday, that's right. Let's take a picture of him.
It's a really cool sponge also that it's hanging out by.
Yeah, so it looks like that there's two Afrocalistes over here, and one of them is actually dead.
Dead. Yeah.
Yeah, that brown one is dead.
Oh, very cool.
Yep.
Looks like he's just exploring like we are.
Yeah, sponge was back to the left. When you're done
Port
with the
side.
shark.
Oh, right there. Almost dead center now. Cool. Bye, shark.
Yep, so this is the sponge we were after at 6 o'clock. And it looks like it has those fistulas like Oceanapia. It looks like it could be Oceanapia. So most of the sponge is actually under the sediment. I'm sure Chris can chime in and let me know if I'm right.
Yes, I am. I'm in awe. This is a beautiful Oceanapia. I'm not positive. It might be a young specimen of the same species we found before. You can see the bulb base with fistulas that are these shorter projections that are blind at the top, and then a main central oscular projection where the water comes out of the sponge. So if you can see in the image I'm looking right now, you can see lots of little pores. The water goes through inside of the sponge body through all these small pores, and then it comes out in that central main opening. I think I agree with Scott wrote before that this might be a clean, it's just probably a young specimen of the species we collected before. It's
A younger version.
beautiful.
But you think it's the same?
Yes, yes.
Than the bulbous one we
Baby
collected
version
earlier?
that hasn't been like the one that we saw before. Probably this bulb base was already covered with lots of coral rubble and other organisms. It will be
Cool.
a younger version.
Awesome.
Beautiful.
It is quite nice, huh?
Oh, and what does that hydroid have on it?
I
on
don't
its
know.
side? There's something like kind of, it almost looks like a horse, a seahorse,
Right.
but like a wormy seahorse.
Probably a worm. My gosh, that's so
Some
weird.
sort of worm.
Thanks,
Don't
Chris.
think any seahorses live this deep.
My pleasure.
Maybe more of a parasite to the hydroid.
Scott's looking for, can we get a closer look at the yellow solitary hydroid behind the sponge on the left? Yeah, I guess that's what we're looking at now, right? This guy.
The one with the worm on it.
He's pretty cool. Looks
A
like he's
little
got
barnacle.
an amphipod or something. Yeah, maybe
oh,
a barnacle.
maybe. Oh, no. Yeah.
Gotta get
I
closer.
think you're right. What is that?
I know, that's the craziest thing I've ever seen. What is it?
Yeah, it looks like it's growing into something, like maybe a larval state of it. Maybe the top is some sort of head, or...
I have no idea. It's super weird.
Is there any way we can get a tighter zoom on it?
That's it. It's as tight
Okay.
as we can get. I think that's a tunicate on a hydroid, so maybe a tunicate stuck to it.
Maybe. On the top there, or the ugly thing?
The weird wormy looking thing.
Okay. Yeah, Scott agrees that it looks like it could be a tunicate.
There's a pink solitary coral.
Yeah. Oh yeah, that thing was maybe a centimeter at most long based off the lasers.
The tiny little thing. Lots of Elizellid, maybe these white, thin, single-branched gorgonians.
Some soft coral, that reddish soft coral that was there. And Ken Sulek again in the chat room was discussing some of the sharks that we saw earlier in the dive again. Near the beginning, the one that was munching on that leftover hatchet fish is another squalidae, likely Eptomopterus hilianus. I'm sorry, again, if I say that wrong. A lanternfish and one of the deep water sharks that can actually glow. And this type of shark is usually really fast and a highly active predator, so it's not surprising at all to see it munching on or attacking a hatchet fish. And maybe the RV even helped it out by shining some light and pointing it directly where it was, and we got some great footage at that. So thanks again, Ken. That was really great information. If you have any more information about any of the other fish or animals that we've seen here on this dive, feel free to call in and we can further discuss it, or let us know in the chat room.
Scott's pointing out that the hydride we were looking at had very developed gonophores, which are the berry-like structures around the mouth, and essentially become budding jellyfish.
Interesting. So that's not part of the hydroid, that's just been attached to the hydroid itself?
No, it's reproducing.
Oh, okay.
Yeah. If we can look at this white coral at 2 o'clock, the real bright white one, looks like the other two are probably, the two larger ones are probably bamboo corals. Oh, blotched cat shark is what we were looking at.
Blotched cat shark. Oh, nice.
Octopus. Yay! I'm so excited. They're my favorite. Oh, cool.
Really cool looking guy. He's like orangish, reddish, pinkish.
Nice. Oh, look how cool his eye is. Oh my gosh. He's just chilling.
So is he able to change his color?
Probably. Most of them do. I don't know of any that don't. They also can change the texture of their skin, which is pretty cool. We might be blinding him right now. So, yeah, that looks like he's really, squished his eyes shut. It is moving around, though.
Nice.
Look at that.
He is pretty small, though.
Get up and do something, buddy.
Yeah, right?
Yeah. Either that or he's got his tentacles down in those holes.
Maybe looking for something, yep. Being stealthy.
So cool. Yeah, he's only about 10 centimeters. He's pretty small.
Yeah. Very cool find. Looking over above, his tentacles do look like to be sifting almost through the coral rubble.
So they actually can use their tentacles, their suckers, as a way to taste. So if you ever touch an octopus, it's actually tasting you with its tentacles.
Now, do all octopi have beaks?
To the best of my
As in?
knowledge, they do. So that's the only hard part of their body. So they can actually collapse and squish themselves through any hole that's, as long as their beak can actually get through it. So any hole that's bigger than their beak, they can get through.
Yeah, he's turning away from us now. Probably telling us he doesn't like us.
He hasn't bolted yet, so he might think he's invisible. Not used to seeing so much bright light around. Being a red-orange color would make him disappear. He's a little confused there, buddy.
Sounds good.
Awesome. You can see his siphon right here moving around right there. That's
Yep.
where he would blow water through to help him move forward in the water column. They also use that to fan their eggs when they've laid eggs. They'll blow water over the eggs.
And that's just under the eye there, that kind of
Yeah,
muscle
you can
part that's
kinda see
moving.
it. Yeah, you can kind of see it moving around under there. So he's pushing water through that.
Yeah.
He's almost clear. The tissue around his eye is pretty see-through.
Yeah.
You can see the shape of his eyeball under there.
His eyes are closed, but he's definitely probably keeping an eye on us. We're
like
happy
his
if you want to
iris.
move on. Thanks. Awesome find. And Ken just also identified that shark that we were looking at earlier is, not sure if we said it out loud yet or not, but it's a blotched cat shark. No offense, I'm not even going to pronounce the scientific name. It's usually around the bottom, among the coral. It's usually strongly associated with Lophelia. It usually doesn't need to swim constantly. Not all sharks do need to swim continuously. Especially some of these cat sharks are typically sedentary, where they can just hang out and pump water then through their gills. And the checkered marking that we saw earlier are a good representation of disruptive camo used to break up the body outline, and so it's also stealthy towards its own predators. So thank you, Ken. That was really helpful what we're looking at here. And this looks interesting. It looks like a stylastrid, if I'm not mistaken.
So.
Quite a large stylastrid.
Pretty robust at the base there.
Yeah. The biggest that we've seen on this dive. With, is that a tube worm or an anemone at the base of them, the purple ones?
Those are cereanthids, which are tube worms.
Oh, okay.
So they actually find some soft sediment to settle in, and then they can, if you get too close to them, they'll pop down into their own tube.
Oh.
Cereanthididae is the
Can we
family.
poke it?
We can. I'm fine with poking. I don't know if anyone else is. There's also an Ophiothrix, brittle star. At 3 o'clock, you can see he's really spiny.
And he's very similar in color to the stylastrid.
This may not be a stylaster.
Oh. Can we get a tight zoom in on some of the polyps for better identification? Thank you.
No, that's probably fine.
I think we can determine it from that.
I'm thinking this is probably a gorgonian, based on the polyps. Yeah, do you see 8?
2, 4, 6. I do
Corallium.
see 8, but usually Octocoralia, do they have such a stony frame?
I don't know if it's stony. I think it just looks stony. Scott's saying corallium. So my guess is that it only looks that, stiff, right? Like a Starlaster's rock hard.
Yeah.
This probably has a little more give to it. It's just very robust. Pretty cool.
Looks good. Thank you, pilot.
There's a bamboo coral right next to it, too.
Yep. And maybe some primnoids also, the finer skeletons. Oh no, that is still bamboo core. We're looking at the bands.
There's a solitary cup in the background.
Oh, nice. There's that sea anemone, maybe that Venus, not Venus, but flytrap anemone we're looking at.
Oh yeah, he's different. So the
Cool looking
corallidae
fella.
do have an almost stony skeleton, like think precious pink corals used in jewelry for that white corallium coral. So they're almost stony-like.
Interesting. Do we know if, again, the skeletal structure is aragonite? I don't know, Scott, if you're aware of the mineralogy, but it would be interesting to know if they're indeed that similar to the stony corals that most of them are aragonite producing. Now, what is that blue coral off to slightly 3 o'clock? 3 o'clock.
Probably
7
a
o'clock, 8 o'clock?
sponge.
That does look spongy, but...
So Hymodesmia can be blue. Has this blue, and it's pretty common on the Lophelia reefs. So if I had to guess, I'd say Hymodesmia, but I defer to Chris on that one.
Oh, okay. And just to point out earlier, Scott said that the coral is actually not composed of aragonite. It's more like a cemented together sclerite forming coral. So not a true skeleton overall. And this sponge does look really interesting. Did you see something?
I thought maybe it was a tunicate based on the holes, but no, I think it's a sponge.
It's a beautiful sponge, and I cannot go farther than Demospongia that we saw it yesterday. It was a smaller specimen. We have two of these large oscules that we can see, these larger specimens, to have many of these round oscules with a thin white border around them. So the same specimen we saw yesterday once, but much larger organism. Really cool.
Pretty cool.
Interesting to collect if there is.
And see, even this deep...
time.
Okay.
Yeah.
Thank you.
Even this deep down, we start to have also a variety of colors. And we can see also to the back and to the right-hand side of this, we have some pretty robust cup corals there. And then on the bottom, would you say now that's primnoid coral, or not sure what that is?
The small gorgonian on the left.
Yeah.
These two pink things are definitely cup corals on the right. So possibly it could be primnoid.
Beautiful.
And I would like to add, Steph, that yes, Hymedesmias tend to have a species with this blue color, but they tend to be thin crust.
Right, they're like a thin crusting one, right? They have that blue-gray, though, right?
It's very interesting.
Yeah.
Is that blue-gray like calcifying that crust, or is it just somewhat crusty overall?
No, it's more of like a soft, like spread out thin layer. The Hymodesmia. It's a blue. We'll probably see some. It's pretty, not uncommon here, and it's just like blue, really bright blue.
And just to correct myself earlier, Scott says that it's not a primnoid, but we couldn't really tell what kind of coral that small coral was below the sponge there. So if anyone else can chime in and look through the video and tell us what it is, we would greatly appreciate it.
Are we almost at the top? How far up?
I think,
Almost.
yeah. Really close. Wow, and we're starting to see a lot denser coral forest in this area, with also underneath the substrate, you're starting to see a lot, little to no sediment whatsoever, but we're seeing a lot of this coral rubble, but maintaining a lot of that framework structure, along with the karst features in this area that all of the corals seem to be really loving at the moment. And we're seeing a lot of different variety of octocorals and bamboo corals. We're having another one of those Afrocalistis.
Yeah, for
sponges. What is that floating by the screen just now? I don't know what that is. Some
Schmutz?
organic schmutz. There you go.
When in doubt, it's schmutz. Scott said there was a yellow fan to the left. I guess we went past it. You guys, can you swing back to the left a little bit?
There seems to be one yellow fan up front as well, so if we missed that one. Oh, yeah, that one.
Yeah. Paramuricea, maybe? Or Muricea?
Primnoa. Some of those yellow-like colors and fuzzy.
This looks a lot like Muricea. It's yellow, though. Usually you see them purple, more shallower. What do you think, Scott?
Can we get the lasers on? Thank you. Oh, it's quite a large coral.
And we got some Lophelia behind it. A tiny little colony. And some, we got corallimorph on the right.
Some soft coral also there.
Maybe Paramuricea. Oh, Acanthogorgia.
Can we get a tighter look on the polyps, please?
I see the Lophelia polyps are out. That's pretty cool.
I am.
Okay.
No problem. That looks good. I do count 8 tentacles on some of these. Scott says it's pretty hard to say exactly what this is, but he's leaning towards plexurity. Thank you. And then we have an ophiuroid hanging out in the middle of this guy.
Would a sample be possible? He wants to sample it. Do you think it's a new species? Do you think it's... Ooh, look at the little euphalsid shrimp. Look how cool he is.
Yeah.
Could Amanda say if this is something familiar? Is Amanda on? Yes, she is. Hey, Amanda. Do you know what species this is? At least genus.
Yeah, just to keep in mind that this would be our last biosample if we were to take a sample of this. Not sure. We're not really getting a lot of feedback.
Yeah.
So we'll maybe if we come across it in the
future
later on, and if we get a response from anyone else on the chat room, we'll come back maybe to collect it, or if we see another one that looks like this. Oh, maybe she's back now in the chat room. Could help us be a little more definitive on if it's a sample worthy or not. Is that a little
Isopod.
isopod hanging out on him?
Maybe a shrimp or something down there at 6. She says collect if you can for confirmation.
So just to repeat, if we do collect part of the sample, this would be the last biological sample we can collect, filling in all of the bio boxes that we do have. So if we can agree that this is definitely worth collecting to potentially identify a new species, we'll go ahead and do it. But just wanted to let you guys know that
we're down to...
This is the last one if we do it.
We still have 4 or 5 hours left. Or 3 hours left, sorry. She said I'll leave it to you to decide.
It can go in this. Oh, we can slurp it. I don't think it will it break up apart to be in the tank enough to be just sucked up for a little bit.
I think we'd have to snip it and then pick it
Suck it up.
Which is kind of a difficult maneuver, right?
Yeah.
All right, can you do that? A tiny piece?
Yeah. If
just suck it up.
Sounds good if you can do it like that.
As long as we can get a good wide shot of the formation of the branches from far away with a laser, then we're cool, because the branching might be a determining factor. So, yeah, if we get a full specimen shot, and then just a tiny clip is good.
Yeah, so this guy is definitely at least 50 to 60 centimeters tall. Would we be preserving this more for DNA structures, or would you like to potentially keep the structure also? This will be our 4th biological sample collection.
Again, rough size. Did you see the laser paint?
We did about 50 centimeters, I would say.
Yeah, you're going to have to snip that, I think.
Well, if you end up dropping part of that sample anyways and sucking it up, I maybe just let go. Ooh, awesome.
Good job.
No, you can just collect it. It sounds good. We got some good zoom ins. Way to go. Good job, guys. Good collection.
All right, we're moving on. That little crab that jumped off was a type of squat lobster. Here he goes
Oh,
again.
Look, he's swimming about, looking for the next coral.
Probably running from us, I would imagine. It's probably pretty difficult to see where you're going when you swim rear end first.
He's not sure what to expect from us.
Oh, what a nice spot. We got lots of outcrops here. Is it possible geodia sponge maybe at dead center? No, it's okay. You can keep going.
Oh, we're just getting full live structures here of all of these coral specimens. This looks amazing. And something floating by really quick on the left-hand side of the screen,
clear.
a jellyfish.
It's hard to see.
Yep.
What is that? A ctenophore,
Ooh.
maybe? A jelly. Oh, sweet.
It looks pretty cool.
Look how amazing he is. I would bet money that he glows. The tips of his tentacles probably light up, yeah. He's probably bioluminescent.
Really cool looking. All right, buddy. Whew. Looks like we won't really see him anymore.
Are we at the top of this mound or no? Here's another probably sandtile fish burrow or some other type of burrow. You can see all the dead Lophelia has been piled up.
Yep. And we've gotten a request again for the lasers. If you could just keep them on so they can look at the overall landscape, that would be great. Thank you. Thank you. And there is some large piles of coral rubble in this area still, so we do know that there once was at least a lot of stony corals in this area. Now we're seeing predominantly a lot of octocorals and black corals, which do sometimes produce skeletons, not all the time, but definitely not usually as robust as the rubble we're seeing around here. So at some point we do know that there were scleractinian corals in this area that probably were taking up the majority of the substrate.
Can you guys swing to 3 o'clock? I saw a pencil urchin that we haven't seen yet on this rock. There he is. So likely Stylosideris, this pencil urchin.
And I also want to take the opportunity to point out the overall geology. So if we're looking at this boulder right now, it looks like it is part of the substrate, that it's not an isolated boulder that was accidentally moved here, pushed here by some force. But we're starting to see that there's different layers, and some layers look more eroded. So we're having the bottom structure, then a little bit eroded away, and then a top structure, a sandwich in between that looks to be eroded away, and then the top structure. And this is partly potentially due to maybe the inner section, that middle sandwich section that is now looking like a hole. Maybe it was of a softer sediment or a different kind of carbonate sediment that was not karstified as quickly, so it actually got dissolved or eroded away, leaving in that cavern-like feature. Or maybe it was never formed to begin with. Maybe it is an internal cavern that maybe the side of it got broken off, now revealing it to us in the overall. We do know a lot in these carbonate environments that in the subsurface they do produce a lot of these caverns, and since they're highly porous, a lot of the water that goes through can end up dissolving these huge portions of these caverns. So we can go back to the biology, but I just thought I needed to throw something in there. So
I've got enough biology here. It's fine. There we go. Probably Stylosideris. And there's some really cool tiny urchins, white urchins on the right, at the top of the rock too, and some solitary cup corals,
some soft corals.
Yeah, we got maybe a possible hexactinellid sponge. I think we collected something that looks similar yesterday at 9 o'clock. It's real small.
Yep. A little tiny lobster or shrimp at the 7 o'clock range. Just within a few centimeters of each other, we got quite a huge diversity, a hot spot. More of those hydroids are seeing, a sponge also in this area. And is there anything particular about this specific species of urchin, or is this very similar to the shallow water ones that we see a lot tucked in away into rocks?
Yeah, we get these pencil urchins a lot in the deep sea. I think they're just more of an ancient species. We do have some pencil urchins in the shallow waters, but they're different. They're different species.
Okay.
I think I see another Geodia sponge at 3 o'clock. Getting a dial tone.
We're getting the dial tone over the loud audio here. Technical difficulties, but we're looking again at maybe a possible one of those
Welcome to
yellow
the conferencing
corals.
center. At any time during this message, please enter your passcode followed by the pound sign.
Hey, video, we can hear the phone dialing out. Are you supposed to hear that? Okay, we're supposed to be hearing that. Okay. I'm gonna.
Can
we turn the lasers on?
They are on.
Okay.
I needed.
Your passcode has been confirmed. If you need technical assistance during your call, press *0. After the tone, please state your name followed by the pound sign. Thank you. There are 4 parties in conference, including you.
Wow, so
Hi,
we're
everybody.
Can everyone hear us again? I hope we're able to get into that now, fix that problem.
Now I can hear you.
Yay!
Here,
Oh,
Christina
yay!
from Harbor Branch.
Hi.
Is that better? Thanks.
Sounds good. And we can see, like, right off this ridge here, as soon as it starts to increase in slope, I mean, based off just our visibility. Oh, wait.
We got a. Can you look at the Gorgonian at 4 o'clock? Swing 4 o'clock. This pink one, that's new.
Yeah, so Scott said this looks similar to what Scott had pointed out earlier that we may have missed in our dives, or that we missed to get a close-up view on, if we could then get a close-up on it. Oh, that's that hemicorallian. Maybe that primnoid paracalyptrophora. Did I say that right?
Paracalyptrophora. I think so.
Good,
This
that
one's
looks
different.
very similar. Scott, if you want to confirm, that's what we're looking at here.
Primnoidy.
Sounds good. Can we get a quick zoom in on some of the polyps?
There you go.
Oh, some of it seems to have some mucusy attachments. Not sure if that's from the coral itself or maybe another organism left it there. The polyps itself look very translucent. The tissue looks quite pink, but if you look at the tentacles, you can really barely see them. They're so small. If it's possible, if we can get even closer. So Scott said that we need to note the polyp heads. They're actually pointing in the downward direction, which is actually a distinctive characteristic for some of these corals. So when we get that closer zoom in, we'll double check on that to make sure we're seeing that downward characteristic. And he said to look at the number of polyps that come from that same place, which is known as the whorl, also is a defining characteristic of it. And he was saying that maybe there's 2 to 3 at each whorl site.
I think there's more. Let's get closer. Got to settle out.
We have a nice gold coral in the back. This is a beautiful little niche right here. We have so much diversity. Well, these are pointing generally down. They look a little sagging, if you will, towards that downward feature. So that's what Scott was pointing out earlier.
I think there might be 5 in a whorl. Yeah.
So that's going around the entire branch is we're referring to that whorl, correct?
Mm-hmm.
So I'm counting 1, 2, 3, 4. I can't make out what's really behind it, but maybe 5.
You can probably assume there's one face in the opposite direction because it does go all the way around. So.
And this thicker branch here, they don't seem to be very well aligned with each other. That the most front pinkish branch, they don't seem to be so linear like they are in the other branches. I don't know if that's because it's maybe a primary branch where these corals are just so robust they don't need to grow linear anymore, but also another interesting point of view.
Okay, we can move on whenever you guys are ready.
And we just got notified that because this is such an amazing place that we've come upon that previously no one has even discovered, especially this ridge from the new bathymetry that was acquired only a few weeks ago, we've been able now to extend the dive by 2 hours. So now our off-bottom time will be at 6 p.m. local. And a big thank you to the ship crew and all the coordinators that are involved in that, because it is that they are taking up a brunt of the work on that, and we really appreciate it because it's not too often that we come across such a beautiful coral garden and forest like this, like we're seeing now today. So big thank you on that, and thanks to the entire crew and the team for everyone sticking it out for us for another 2 hours.
Right here, there's some Lophelia at dead center at the base of this Gorgonian.
And then what's that also stonier structure that's sitting on top of it on the Leopathes? If we want to get a zoom in on that too, I'm not entirely familiar with that one.
With the pink?
Yeah.
Yeah, we can look close. It might be dying or dead or some other species. Who knows from this far away, right?
We see some little fish hanging out underneath the Leopathes. And I thought I saw a black shrimp swim by earlier, but
nice.
I couldn't quite make it out.
So down at the base, there's some Lophelia. I just want to see if it's a living.
And then right underneath it, there's a bulbous round sponge. If we can then also get a close-up on that afterwards, there's a request for that.
I think that round sponge is probably Geodia. Yeah, it looks like most of this is dead already. So we've got a strawberry coral living on it, and some anemones, some yellow gorgonians.
And a cup coral looks like also that purplish feature. And then some of that, is that a platy sponge in the background with the fringes?
Yeah, I think that's a hexactinella, glass sponge. So if you pan down a little bit, there you go to the Geodia, which is this round ball sponge.
Right next to a shrimp. So what is that center column, pore column used for? That
So that's usually the X current. So the sponges will, you know, they have the tiny little holes around the outside or smaller holes around the outside that they bring water in, and then they shoot the water out of that large hole at the top.
Okay. Is that for feeding purposes that they draw
Yeah.
in the water and then
Right.
try to filter out?
Exactly, they're filter feeders. So I'm sure Chris can give you a much more detailed explanation into it and how important they are on the reef.
If you want to call in.
I think she's on the line.
I'm here.
Yeah.
Hi, I'm hearing you guys. So I can join in this moment. This is a beautiful round sponge. It might be a geodia, but it also could be, Steph, one of these petrosids.
Petrosid?
So a petrosid, it could be a petroside ovata, for example, that is seen before around 300 meters depth. So if there is any chance to collect this sponge, if we find it more frequently in the rest of the dive,
Okay.
it could be interesting.
Okay.
And yes, this, as Stephanie told you, the large hole will be the excurrent opening that is usually called the oscule. And these organisms, although they seem to be doing nothing, if you could see the water movement through their body that we've been able to see by putting fluorescent on the surrounding of the sponge, you could see that through the whole surface of the sponge, there's very tiny pores, around 50 microns in diameter, where the water is coming in, and then it's leaving continuously through that large excurrent hole. Now, the amount of water these organisms process is incredible. It's been measured up to 1 liter of water per hour per cubic centimeter of the sponge. So this sponge, if it's around 10 centimeters, it might be a kilo. If it would be a kilo, it will be filtering 24 tons of water a day.
20? How many? How many?
Filtering 24 tons of water.
24
You
tons.
extrapolate to the number of 1 liter per hour per cubic centimeter.
Per cubic centimeter.
Tissue. Yes, so I'm not sure exactly the size of the sponge. This is kind of the highest, on the highest end of the rate of water filtration. And when I say filtration, it's truly a transformation. Around 99% of the bacteria in the incoming water gets taken by the sponge and all its associated microbes, and it's only 1% of bacteria that comes through the excurrent water.
Awesome.
Besides taking up the bacteria, there are many types of microbes that are symbionts and live inside of the sponge that might do ammonium oxidation, sulfate oxidation, sulfur oxidation, and there's a big transformation of the water that comes out through this, through the body of the sponge. So it's really fascinating once you get in that aspect of their function, of their feeding.
Right, so this little tiny 10 centimeter sponge is extremely important to this habitat. And it's funny because it's just like, looks just like a little white ball. What could it possibly be doing? But it's extremely important to help maintain the health of the reef.
Oh, thank you for that.
Yes, hopefully we'll be talking more about this during the cruise. I will let you know other interesting aspects.
Yeah, chime in whenever you want.
Like I said, if you look,
Absolutely.
Petrosia ovata looks a lot like this sponge.
Okay.
I will write it, but we need to check because we know how there's several sponges that look exactly alike and they might belong to different orders of sponges.
Great, thank you.
We can move on, guys, when you're ready.
You ladies.
Thank you, Chris.
And earlier in the chat room, Ken mentioned that a lot of these animals, especially the fish and sharks and octopus that we've seen in the past, they're not used to this bright light that's coming from the ROV. And despite some of them maybe being blind and not being able to perceive light, a lot of them still are, and they're very sensitive to these lights. So sometimes when the ROV passes by them and that light, they get that first waft of light, they tend to actually shake their head because they're quite confused as to what is going on. But they do fully recover. It's not damaging at all to them. It's just a momentary point of confusion.
6 o'clock,
Ooh.
please.
What is this grumpy looking thing?
Some kind of goosefish, I think.
Ooh.
Let's get a good shot of him.
This guy looks really cool.
You can see all the cool barbels. He's definitely going to be a sit and wait predator. Has a big mouth, lays on the bottom. I would bet that he has a lure, but we'll see.
He does. It's kind of wafted off to the left-hand side.
So you can see as he's modified back fins, he uses look more like feet to crawl around the bottom.
Or just stand incredibly still.
See, we might freak him out enough to get him to move.
So are all those little things that are around his face for sensory purposes or more for camouflage, decorative? I belong here.
I'm not entirely sure. It could go either way. It could be both. He's pretty fun. He is very grumpy. You can see his teeth. Oh, how cool.
Those look scary. Would not want to be in the jaws of that. No coming back.
Yep. I would imagine he can get a pretty big piece of prey out of that, too. And that big smiling mouth.
If we have anyone in the chat room or online that can help us identify what kind of fish this is, that would be really great. We think for now it's a
goosefish. I think it's a goosefish, but again, it's got a, you know,
the dorsal
nerve,
fins also look like they have some spikes on them, some spines, where potentially if someone were to come up above them, they would get, I don't know if stung is the right word, but maybe stabbed.
Yeah.
This looks really interesting overall.
I've seen these goosefish on the Lophites. It's an angler. Right, so it has this, when they say it's an anglerfish, that means it has a lure on its, between its eyes that usually uses to sit and wait predator, and actually like acts like a little fishing lure to attract little fish.
Like Finding Nemo taught us. And it's from the genus Lophoides. Thanks, Ken. And he says that there are 3 similar species closely associated with Lophelia habitats, so good to know. And actually, to answer my question from earlier about those fringes around his jawline, they're actually meant as camouflage to help him associate better with the overall substrate. So as we can see all around these corals, there's a lot of that fuzzy tree material that Chuck had mentioned earlier, which are a lot of these forams and hydroids and zoanthids. And it looks like he's developed these overall fringes on the sides to help camouflage himself to be part of it. So thank you a lot, Ken, for that input.
Lophoides Barrio, maybe.
We're good here if you want to move on anytime. And he has really blue, crystal, greenish eyes. I wonder if that's something to better see in the dark lights, or if he's actually capable of seeing really at all. Maybe some of the bioluminescence quick to pick up. I don't know if any of his internal color structure of his eyes has anything to do with that, but they are quite distinctive.
A lot of the times, I know it happens in sharks. The back of the eye is reflective, and it's called tapedae lucidum. You can actually see it in cats too. And what that does is, as light comes into the eye, it's actually reflected twice, so they get more more bright light than we could see, right? So like it increases the light that it reaches the back of the eye.
Really interesting. And to be noted, he has not moved a single centimeter while we've been discussing all of this. So he's in serious stealth mode at the
or he
moment.
thinks he is, at least. We can see you, buddy.
And Ken also wanted to add in that with this angler, because we're looking at the little fishing pole, the lure that he uses is folded backwards. They usually are lightning fast when they want to strike, and they're noted to be one of the fastest among vertebrates of how fast they can actually move. So if we were able to see it being able to catch its prey, we probably, if we blinked, we'd miss it. So definitely a very, very interesting find during this dive.
Looks like more of this Corellium, possibly, this white one.
You tell me.
I don't remember. I'm judge. I don't know. I just work here, right? That's Liopathes, the orange one. That's another black coral.
Could look similar to something Scott mentioned earlier about Corallidae hemicorallium in white. Maybe. Not sure on the identification there.
Yeah. There's another bamboo coral on the upper left, excuse me. I guess we can recap where we are, since it's been quite some time. This is dive 2 of this cruise. We are on the Million Mounds East location. We are actually in the furthest east feature that we've found on the multi-beam within the habitat area of particular concern, or what they call a HAPSI. This is a protected area established by the South Atlantic Fisheries Management Council in 2010. We had our deepest depth for this dive was about 830 meters, and we should be about at our shallowest point, or near our shallowest point, at the top of this ridge at 750, but it might get, 750 meters. I think the highest slope in the multi-beam was about 30, but I'm pretty sure when we ground truthed that we were closer, about 45 or 50 degrees on the slope coming up this feature. There is a large scour mark to the east of it, so likely the current's coming either from the south or from the west. And as we got closer to the top, we had an increase in gorgonians and life in general. So we, by the graciousness of our ship's crew, have extended this dive until 6 p.m. Because of that. This entire mount is entirely covered in Lophelia rubble, with very little living Lophelia on it. So that's about a sum up of the recap. We are a couple hours in. We've collected 4 specimens so far, most for DNA. 1 possible new species of Oceanapia. We will find out when it gets studied.
I think you hit it right on the money, Steph. And as she was saying earlier, as we've been progressing up the ridge of this mound feature upslope, we're starting to get more and more dense framework systems. And we were just in a patch where you could not look a few centimeters without seeing some form, shape, or form of coral in the area, as well as some associated biology along with it. And you can see that off in the distance there, that there were some fairly large frameworks of coral systems there. Maybe not frameworks is the wrong word, because that's usually associated with scleractinian, but quite large corals overall in this region. And that's really what we expected, maybe not expected to see, but is generally characteristic of a top of a mound, especially when there's good current quality, good temperatures, salinities, and dissolved oxygen, which are all a lot of the basic requirements for these coral features to even be present.
Can you turn to 9 o'clock, 8 o'clock? There's a ball on a stick.
A spongy looking material.
Possibly Hylanema. Yeah. Now, yep. Yeah, that's a pretty good one. At 6 o'clock, we've got a Hylanema. Yeah, right there, almost off screen.
And Ken Sulak also chimed in again, and when we were discussing those blue-green eyes of the goosefish that we had seen earlier, he says that's an indication of the typical blue bioluminescence produced in most of the midwater fishes. Like Steph said, is reflected off the back, and it overall. I'm not sure what defeating ventral camo bioluminescence really means, but it enables the fish to see a lot of that red or yellow light that comes through from certain organs that are seen in other midwater fish. So it's a way for it to it's evolved in a way that it can actually see a lot of those red and orange yellow organs inside what would be an otherwise clear fish. So
just. Can you guys sit here for a minute? Is that okay? Okay, so ventral chemobioluminescence is when the fish is midwater or above. They actually will bioluminesce their chest or their bottom half of their body, so that when a fish looks up from a predator looks up from the underneath, it actually disappears in the light above. So that's what he means by defeating ventral chemobioluminescence.
Wow.
This is a Hylanema, which is actually a type of hexactinellate or glass sponge. We have a few samples of these at the lab. They actually grow really tall, like a couple feet tall. And if you look at the stalk to the right at the bottom, those are actually siliceous silicate spicules that grow. They're all, each one of those strands is a single spicule. They can grow a couple feet tall, and they're so fine you can actually use them for, was that when they put the light into it? Like that thing. Fiber optics. Thanks, guys. Fiber optics. They're so fine and clear, you can use them for fiber optics. I mean, obviously it would be a pretty expensive fiber optics, but yeah, that's pretty neat. They're pretty cool. So the actual sponge is sitting on top, and it's on this spicule stalk, and to hold itself up above the bottom of the water so it can filter feed better. So that's Hylanema.
Really interesting. And aren't there a few coral species that also use the silicate spicules as the form of their base structure?
In the gorgonians?
Yes.
Yeah, I think so. Silicate's pretty common. Yeah, but I don't recommend that. I feel like it costs an awful lot of money to get out here to see one of these sponges.
And it's using a lot of these sponge spicules and whatnot to attach itself to the substrate underneath. So it's not necessarily looking for the most stable substrate, but a lot of this coral framework is stable enough for it to attach itself to. And just as Shirley indicated in the chat room, and she also said that the sponge-like material at the top of this is actually very much similar to the consistency of cotton. So very, very soft to the touch. Those are those potential fiber optics silica spicules that we're referring to. And is it normal that they end up twisting with each other so they can form stronger?
I think
bonds?
so. The one we have in the lab does the same thing. It's got that twisted formation. Granted, it gets handled a lot because it's one of our sharing specimens, but yeah, it has that same kind of twisted feeling to it.
Interesting. Thank you. We're good whenever you are. Oh, it's next to a fallen down bamboo coral. And there are some other bulbous sponges around it, but it doesn't seem like they have such as long as a stalk, or if a stalk at all. So
yeah, they look like Hylanemon, the same species,
Yeah.
or at least the same genera.
So it's not necessary that the stalk always grows, or is it just because they're juvenile?
They're probably just smaller.
Okay.
They can get pretty big. They're not always that tall. It also could be that they have found a nice spot in the water column, not to, they're getting enough energy. They don't need to grow up taller. Here's another one right here. It's real short.
Yeah.
It looks like we've got Lophelia dead center. And more Africalices sponges.
And just overall to point out, look at now the substrate, how much it has changed at the crest of this ridge. So we're not really seeing a lot of sediment deposition overall, but we're seeing a lot of this limestone and karst features that have created these huge tunnel systems, either by being dissolved or just not filling in that area or forming. And it creates these large intricate networks, which then the biology can take advantage of and either use it to hide like fish or crustaceans. And a lot of that hard substrate is really just really great for coral proliferation, as we can see in this area. It's totally taking advantage of the overall area. And as for the current systems, there still seems to be a current flowing, but it's not as strong, promoting corals that it's just strong enough to carry the food without having to carry all the larval away, or even that the corals have to fight maybe against the coral overall. And what is that sponge-looking glassy thing? Is that another wetting glass sponge?
Mm-hmm.
It seems to almost grow downwards.
I think it's growing, down and then up like that underneath a rock.
Really interesting.
Yeah. Pretty cool. There's a little teeny tiny baby one in the middle.
Yeah.
Heterotella.
Thank you, pilots. And
some stylaster and some more gorgonians. We've got some
soft corals.
Yeah. Some
the strawberry
corals.
coral.
Mm-hmm.
And a lot of solitary corals. Highly porous. And a lot of those
you might be able to, if you zoom in real close to the base of it, to the lower half, we might be able to see the shrimp that live inside. I think I saw something move. There he is. You can see the little shrimps inside.
Oh, yeah.
Moving around. So there's a mated pair. I don't know how they get in there. I assume they come in when they're planktonic form. But nobody really knows how they get in there and how they know there's only, who goes in first, whether the male goes in first and then attracts a female, or we really have no idea how they get in there because they can't get out. So they're in there their whole lives. You can see them, they're moving around right in the middle of the sponge.
Okay.
Maria says, Chris says as babies, they come in as babies.
And Shirley added on that when they come in small, they then start to grow, and eventually they get too big where they can't crawl out any of the porous areas. They get just too big for it. So,
and there's always a mated pair. So they're trapped for life.
Do they form larval, or do they have eggs that then potentially are small enough to then flow out through the sponge walls?
Yeah, I would imagine that they, when they lay their eggs, they go through the excurrent siphon is probably the best guess I have.
And there's only this singular species.
I don't know about that.
The sponge?
I'd ask Shirley that. I think it's one specific species, but Shirley would be better at giving me a. She says she's not sure about the species, so I don't know if it's one singular species or multiple species, or if like this heterotella, this specific heterotella has a specific species of shrimp that lives in it, and whereas like maybe there's more heterotella, a different species and a different species of ship, like in the Pacific.
Interesting. Yeah. Thank you, pilot. We're ready whenever you are.
I think there's a coral-listed sponge at 2. That's a white flat pancake up there.
And there's another one of those large shrimp features right on the bottom
Yeah.
right-hand corner.
At
What was it
4.
called? A royal red
Oh,
shrimp?
I thought maybe a royal red, but I wasn't 100% sure on them.
Interesting.
This shrimp is good. That's at 4.
Just
off
off screen.
screen. In that rock.
And I just want to reiterate to those that are involved in the chat room or anything, you can see that the Okeanos nav posts our precise location with the ROV as far as lat and longitude goes, along with additional information. And just to go over some of that information, right now we're currently at 753 meters below the sea surface. The temperature is approximately 8.3 degrees Celsius, and if you want to note from yesterday's dive to today, yesterday's dive was about 6.5 degrees, so we're just about a 2-degree difference, and not sure if it was the substrate or the current that anything has to do with it, but the temperature-wise, we're seeing a roughly 2-degree temperature difference, which may or may not make the difference with how much coral we're actually observing and the overall biology. There are a lot of multiple factors that play into that, but that's something definitely to keep in mind of when we're looking at different dives throughout this month that we'll be looking at this along this cruise. And also the salinity is roughly about the same, about 35 PSUs, and the dissolved oxygen is about 4.4 milligrams per liter. So that's well within the margins of coral proliferation, and we're actually very well seeing how happy these corals are just by the sheer abundance and also the diversity that we're seeing in them, and also then their associated biology. And just like this crab that we're seeing, which was maybe identified as a royal red shrimp.
Possibly a royal red shrimp, but I'm not sure.
He seems to has made his home in one of those porous features, those kind of little caverns that we're seeing that's in the limestone.
Yeah, he's really armored, right? His rostrum is really spiky.
There is a request to see if there can be a momentary shutdown of the lights to see if there's any interaction with it. I don't know if ROV, if we can really do that, or if you would rather have constant light. Well, I guess all of D2's lights. I think that's a good compromise. Potentially, it wasn't said entirely exactly the purpose, but
Oh, you won't pick up anything?
okay. Not sure. We can give it a go if you want to maybe pan out a little bit. I don't know if we're looking specifically at the shrimp for bioluminescence. I don't know if it does any of that.
Shirley said it would be interesting to put a UV light and filter on the ROV. Don't shoot her, GFOE guys.
Just another idea in the long run, I guess.
Yeah, if you're not going to pick up anything, if you guys can't see bioluminescence with your cameras because they're not strong enough or aren't made for that, then yeah, it would just not be
Maybe it's not worth it.
worth it. Yeah, not worth the risk of them not popping back on.
I wonder if that would be a possibility in the future to add a latch so we can kind of cover the UV camera or the camera with a UV or not. Thank you very much, pilots. Oh, we are able to turn off the lights. Okay. So we are seeing now everything in the dark. Sirius. Maybe momentarily to see if there's any flicker of light that we could possibly see. Not sure if... Oh, no. Either the biology is just not there for it or the camera can't pick it up, but we're not really seeing anything besides the blank screens. So, oh good, the lights come back on. Okay. It's always a momentary question. If we can get it all back on. Thank you, that was really great. We can... Maybe if we do potentially see a jelly or something in the future. It did look creepy. Maybe we'll use that shot later for Halloween again. And we're seeing some more Antipatharia here and a little more squat lobsters, some cup corals. The diversity along this entire area is just so big. It's a hot spot for all of this, for all of these different biological materials and animals and anything we're looking at. Every centimeter is covered in some sort of biology. We're getting a special thank you for the pilots from the chat room, so thank you very much for experimenting with that. Once again, we're seeing some lovely bamboo corals, and this is, there you can characteristically tell they're bamboo corals by the banding seen on their skeleton, which you can see through some of the translucent tissue that's overlaying it, and even on this dead one that's lying on its side, we're starting to really see that striped banding that's indicative of a bamboo coral. And Shirley is saying from the chat that there's a lot of spherical lithistid sponges and some large sponges, which are called pacastrelid. So that's some really good information, not just the corals, but also the diversity of sponges that we're seeing in the area, plus one that was previously collected that may be indicative of a new species that we're seeing through this area. So a lot of these corals are very large in size, which could potentially mean they're also very old. And very old is a relative term because some of these corals, despite usually being more slow growing than the shallow water corals, vary in growth rates overall to each other. So different species might be of the same size but of different ages overall. And there are some more stony corals in the background. Not sure if those are Lophelia or not. Can we get a quick zoom in on that white stony coral off to the right-hand side? Yep. This is in fact Lophelia pertusa. And where you can see at the top there are some polyps that are extruded out and looking for some food. We have some associated zoanthids, and it seems like it is growing up on a older degrading bamboo coral skeleton, which I guess that was its hard substrate that it chose to start growing on, and we have maybe a squat lobster hiding out there in the center of the framework system. And these stony corals are actually really robust and strong. As far as overall the corals goes, thus the name from Scalartinian, stony corals, they produce a really strong framework system that's aragonite based as far as mineralogy goes of what their skeletal structure is shaped as. This is a calcium carbonate mineral that you either in coral skeletons we see a lot of aragonite, which is predominantly a lot of what we see in shallow water corals. As well as the stony corals. They are aragonite precipitating. But the other mineral that we're actually seeing of calcium carbonate is high magnesium chloride that's in that area. And I think we just saw a fish. Right above us. Okay. And I won. Oh, all right. Bye, little fish. And so what we also have, the other one, as I was saying, is magnesium calcium. High magnesium calcium is also precipitated and seen in coral skeleton structures. And within the water column, we have different levels and different densities of where corals are usually typically seen, and a lot of the chemistry that's involved in the water column affects how these corals grow. So at certain depths we have what are called horizons and saturation zones. So towards the more shallow end we have a lot of aragonite saturation horizons. A aragonite saturation horizon where aragonite, the saturation level is quite abundant, so it's easily precipitated from the water column, whereas below that aragonite saturation level, which in the Atlantic we usually do not surpass that level, so we are able to see aragonite precipitating organisms all the way down to the seafloor. But in the Pacific, because it goes so much deeper than the Atlantic, we do pass that threshold, and it's projected to be around 2,000 meters in the equatorial to southern Pacific and slightly shallower at the northern end. And usually below that we do not see a lot of aragonite precipitating material. But we do still see some high magnesium calcite. However, that does have a similar solubility level to the aragonite saturation horizon. So we actually are able to tell the difference between the two chemistry levels in the water column and see which skeletal, by indicating what kind of skeletal material the overall coral's in, we can likely estimate what the overall horizons are and the solubility levels of the coral structures themselves.
Yeah, lots of outcrops, slabbiness up ahead.
Yep, and we also have a lot of that coral rubble and more of the limestone features, and these look more like large boulders that have been eroded at the top from maybe some of the current systems or whatnot, but it's quite bumpy. It's not necessarily all flat unless we get more to the sediment regions that we're looking at right now, going down slope. And then coming up to the top of the ridge here, we're looking more at those nobular, bumpy, more features of the limestone, which the corals seem to really prefer overall, this hard substrate that's really great for them to latch onto and to grow, despite any sort of current surges or anything that might come through.
Looks like we're getting more barren here, a lot less corals on this side. Where exactly are we?
We are
The eastern edge?
I think on the northern edge.
Northern side? Okay.
Of that ridge.
Or southern side?
Well, that's north.
Okay.
Going that direction there, so
Okay.
yeah, we're a little bit on the northern side of it.
Yeah, it's quite steep here.
Very steep.
Almost 60 degrees, maybe? It's
Potentially very steep in this location.
even more coral rubble piled up.
Falling along the edges. A little Antipatharia hanging out there. And again, there seems to be a stony coral, some stony coral growing on top of the black coral there.
The
Which
Leiopathes.
usually it's the other way around. As Scott pointed off, there's usually either a bamboo or something growing off the stony coral.
You have a gooseneck barnacle on them too, one that Leiopathes.
Yep.
And another geodia around 3 o'clock. And a crinoid.
So Ken has pointed out on the chat room that in some previous submersible and ROV dives, we have occasionally turned off the light and sat in the dark for a minute or two. And when we turn the lights back on, a lot of the fish that was otherwise avoiding kind of the big bright lights of being seen by the ROV start to actually come back. And then when we turn the lights on, we can actually see them, if even for just a moment. So maybe in one of these dives, maybe we can do that. We'll have to coordinate with Nav.
It looks like another one of these oceanapias that we collected already at 9 o'clock. It's kind of blending into the background. It's a browny color with this fistule that sticks straight up.
And another one of those
Afroclistes. There's another nice one. There's a nicely formed one right here on the rock. Noon. Apparently my mother is watching, so say hi to my mom.
Hi, Mom. I'll give a shout out to my mom, too. I know she's watching. Thanks for raising me. And while we're giving out shout outs, I would like to also dedicate some time to all of my friends and faculty members at the University of Miami at Erasmus. They're probably enjoying some sun-filled time now.
It is Saturday.
Never mind. They're not at work then. They're not watching this. That's fine. We can move on.
Yeah, lots of Lophelia up here. A couple of coral heads. So I think Lophelia, maybe Edalopsamia are the only two scleractinian corals we've seen so far. There's no Madrepora, right? This dive?
Not that I have really seen or would know.
And this one is also growing off the Leiopathes. Isn't that odd?
Yeah.
It's settled on there.
I don't know. We've seen quite a few of those bushy ones coming about in that area. And I just want to say in the shout-outs, I don't also want to forget my dad, who helped also raise me. So just in case he's listening too, don't want to leave anyone out. But looking at, this is really interesting as far as the whole limestone system works. Like, there are all these little caverns that are coming on, and do you see something there?
Possibly a pinnate hydroid if you look at 6 o'clock. Looks like a feather.
Almost in the dead center, just below dead center, if we can get a quick zoom on there.
Yeah, the feather. That might be another Bathypathes alternata.
Oh, it looks like it has an exterior branch kind of.
Yeah,
poking
that's
out.
odd. And then there's also another Gorgonian behind it.
Yeah.
Looks more bushy.
So really quick, looking at the crinoid now. This is a white crinoid. How many arms does it have? Is this something that Chuck was mentioning earlier?
Yeah, let's see if it's the same. I think you said Xenometra.
Something along.
1, 2, 3, 4, 5, 6. I see 7, 8 arms.
Can we get a possible zoom in on that crinoid?
One, 2, 3, 4, 5, 6. Yeah, I see.
Yeah.
I see 8.
I see 8. Okay. Oh.
We're looking for a 10-armed.
Yep.
No, we're good. Thank you.
I think we're good. And then we can see some sediment accumulations within the actual limestone. That could be the limestone itself eroding and breaking down, forming the sediments as you see, or it's also an accumulation of a lot of foraminifera that are in the overall water column that when they die, they form and become part of the sediments that are there. And we see them that the current generally brings them throughout the internal cavern system and pores that are seen all throughout this dive, that the overall fish and crustaceans do tend to take complete advantage of with in terms of safety and finding maybe new food sources. Christy says, what are those white standing sponges? I'm not sure which ones you are referring to. 7 p.m. 7 p.m.
7
Is
o'clock?
that
the white, yeah, Africalistes beatrix.
Oh, another ribbonfish just passed by.
These Africalistes sponges have been found by our biomedical research department at Harbor Branch to have anti-cancer agents in them. Which one?
That rock almost looks like a face over there, so looks really
It
weird.
could be.
I don't think it's really anything. It just takes the shape of it.
They kind of have a skinny stalk. You might be talking about Hylanema, which would be about 3 o'clock right now. So there's two sponges here. Both of them are hexactinellids. The one in the dead center is Africalistes beatrix, and that's really common on the Ophelia reefs. And that's the one that has, I think, anti-breast cancer agents in it. They're secondary metabolites. And then the one on the right, which is about at 3 o'clock, which is also sort of stalked, is Hylanema, which has siliceous or silicate spicules that it sits on that grow out of the bottom of it that pick it up off the reef or out of the sand so it can filter feed better. So I'm not sure which sponge you're talking about on the chat room, but it was one of those two, I think. You can zoom into Africalistes real quick, which is the one at either noon or 9 o'clock. Yeah, the white stalk. Yeah. I think Chris is looking at this one, so she just couldn't tell what it was from far away. Yeah.
Yes, thank you, Steph.
You good?
I have noticed that these two specimens have kind of a thinner
base.
kind of stalk, or the basal part. So I was curious, but thank you, and you're right. It's the same Afrocalistes. And look how it has this kind of looks like mangrove roots coming out and helping sponge.
Keeping it sturdy, right?
It's very interesting. Thank you.
All right, pilots, we're good whenever you want to move on. There's a solitary cup corals in the background and some bamboo corals to the right, excuse me. wispy orange colony on the right. Before you guys move off, there is a wispy colony. There's an orange colony to the right of the sponge we were just looking at 5. 4, really. The light orange brown one, yeah. Yeah, now dead center screen right there. Let's give a quick snap zoom on that guy and see what it is. Yeah, that is probably a black coral. Maybe a baby Leiopathes? Any ideas, Scott? Yeah, young Leiopathes. Scott France on the chat room says probably young Leiopathes, maybe. Okay, we're good. Thank you. There's a really big Leiopathes in the background. We look up a little bit. There is that what might be a same species, maybe. Just fully grown. Can we get lasers on that? Can you paint the orange Gorgonian for me? There we go, I see it. So he's probably almost 2 meters tall. This is a really, really old black coral. These are commonly poached, and they are considered an endangered species and protected. There's a really large white coral at 9 off screen. If you guys could swing 9. There's a giant white coral back there. He's huge. There you go. There's 2 of them. Either one is fine. Yeah, there's actually quite a few of these. Okay, I'm good. You can keep going. This is a nice Gorgonian. Yeah, I just wanted to take a peek at it. Oh, that is a bamboo coral. Jeez, it's huge. This looks like internodal. You can see its polyps. Keratosis is the genus. Keratosis. Very nice, thank you very much. Keratosis. Beautiful. You can see the individual polyps growing. Their tentacles are out waiting for food. And the tissue on the bamboo coral are along the margin, well, the stalk is actually pretty clear, so you can see the banding of the skeletal structure underneath, which is what makes it pretty characteristic of what we call it a bamboo coral. Yeah, we're good. That's really big. So it looks like we have maybe a orange ruffy, or what used to be called a Darwin slimehead until we started fishing them, and nobody wants to eat anything named Darwin slimehead. So they changed its name to orange ruffy. Oh, there's a sponge at 6 o'clock. It's a tube. Now it's more at 5 on the edge if we could get a zoom on that guy. Yeah, it's about center screen now. Yeah, that guy right there. This might be Visalia. If it is, it's one of our target species. You think this is Visalia portalisi, Chris? Yep. Let me double check. I'm pretty sure that's one of my targets. If we can get a good close-up of this guy, that would be helpful. We might be collecting this. Let me just double check. So this is Visalia portalisi, I think. Yes, which is one of our target specimens. So maybe we should grab this guy.
You want to try to suck him up? Or put him
Yeah,
in the bio box?
we only need DNA. We know the genus and species, so we don't really need a whole form, but likely if they pick up any of it, it's the whole thing's going to come.
Okay.
Hey, John. I'm glad you're on the chat.
Ooh, all those spicules.
Mm-hmm.
Are they stinging or?
They don't sting, but they certainly get buried in your skin. So you have to wear really thick gloves when we process this one.
That's going to be fun.
Mm-hmm. It is.
Hi, John. Thanks for joining the chat room. So how do we want to approach this? We're going to use the last bio box. What do you think's the best way to approach this pilot?
Mm-hmm. Mm-hmm. Okay.
I think that's a good call.
We only need a small piece of it for DNA. We know the genus and species, so having the whole form isn't necessary, but you're likely just to suck up the whole thing. It'll just pop right off. So. If you guys can pop the lasers on real quick. It's before, because this whole thing's going to go, so. So he's only about 10 centimeters. Yeah, thanks. That's perfect.
Like 13, I would say, if we're being precise. Ish. It's not that big. Why don't you collect the whole thing? Well, it depends more on the size of the suction thing. So we rather it not have, maybe it gets stuck within the hose or something. So maybe better to take a snippet of it.
I think we're gonna end up. Collecting the whole thing.
Okay.
Just because I think that's what's gonna. As soon as I touch it, it's just gonna pop right off. So. I don't. I've never heard it called the Russian hat sponge, but maybe. Shirley, have you ever heard this called the Russian hat sponge? It is not. She said it is not. Yeah. Oh, Russian hat sponge is a geode, yeah. We collected one of these once, and found a whole bunch of squid eggs in the bottom of it.
Oh, nice. Is there biota likely to be living inside some of these sponges?
Yeah, that's pretty common for them to have associates.
Oh.
Yeah, we just need it for DNA. It's okay. Well, it's pretty rubbery on there, huh? I thought it'd be much crumblier.
Seems to also be pretty well attached to that rock.
There we go. We got a big chunk.
Good job. It looked like you also just collected some hydroid. Something just helped itself right in there. Very nicely done. Was this in tank 2? Thank you. Very well done.
Thank you.
And we've been notified by Shirley that the sponge can heal, so it will potentially grow back. Good to know.
Nice. We are good if you guys want to move on. Is the plan to go to the south or to the north? South. Okay. Did we by any chance get over to the western side of this mound? We're on the western side. Okay. 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 Okay, great. Thank you.
Thank you, co-pilot. And then also what we're looking at again is that steeper slope area. Although it does have some coral framework on it, it's less sparse than at the top. And just to have another point of view, if you want to look at camera 2, you can see also just how steep this side is. So D2 is trying to be stabilized at neutral, and the surrounding sloping, you can actually see it goes quite steep. I would say also like 30 degrees probably.
Yeah, maybe even more. Get a good look at the look down the hill.
Yeah, maybe, yeah, close to 40. We're seeing a lot of that dead coral rubble again, but some are still maintaining the reef structure, the overall framework structure that we're seeing. So indicative that maybe it's fresher than a lot of the coral rubble that we were seeing at the base.
There's a different kind of crinoid at dead center. We can snap zoom under that rock. That would be, oh, maybe not a crinoid. Hold on a minute. Yep, crinoid.
With all of his arms tucked in.
He's got tons of arms. More than 10.
Yeah. Chuck, if you're around and can give us an ID on what this guy potentially is. Not sure. Probably at least 12 arms. Usually it's an even number, right?
Yeah, I think so. It's usually even. I mean, they lose them, but naturally, right?
Yep.
So, like the septipod from Finding Dory. Right. He's missing an arm. But usually it's 8. Starfish are really commonly 5, though. And these are related to starfish, so I don't think it's totally unheard of, but I think that's a Chuck question.
Okay. Pilot, we're good if you are.
That's pretty cool.
That is. And all of the little
Endocrinus. Might be an undescribed species. We've called it E. minimus, abundant in the lithoherm to the south, but no specimens for DNA. So you want a snippet of it? This is a suction sample. Yeah, I would say so, but let's see. Hey, Chuck, you want a suction sample? Is this worth getting?
The pinnacles are slightly curled in at the edges. It's really interesting.
Yeah. Shirley thinks she sees a carnivorous sponge. And a sponge to the right of the crinoid. So we might just suck these both up if there's a carnivorous sponge here. So Shirley, if you could, can you call in and tell me where your carnivorous sponge is? I'm not sure what she was talking about. That white one, is that white ball? Is that what you're looking at?
The greenish yellow stalk to the left of the crinoid. Oh, I thought that was more of a hydroid.
Stalk to the left of the crinoid. Okay. We got to get in a little closer here.
The entire specimen might detach with a suction. Likely.
Yeah, the crinoid, right? Endocrinus.
We've seen quite a few of those little guys on corals and whatnot, so maybe if we get a closer view, we can then determine what it is. To the left, yeah,
The green hydroid?
Yeah, I think that's a hydroid. Sorry to disappoint Shirley. But,
Yeah, if we can get this crinoid in suction, that would make for a good find for a DNA sample. We do not have any. We have no DNA specimens for this. So this is going to be sample 6.
yep.
Sample 6B. Endocrinus minimus. It's probably close to 5 centimeters. Possibly undescribed species.
Oh, I think you're getting that coral also atop.
Yep.
Oh.
They can hang on pretty tight.
It's a strong little fella.
Might want to scrape its feet.
Did you just get it? No.
He had a death grip on that rock.
Yeah.
Literally, I guess. Poor thing.
Did we get him? Oh.
Oh, he looks so sad. Yeah, your best bet's probably just to take the tip of your suction hose and just scrape the rock behind him, and he'll eventually release himself. Chuck, is this a stalked crinoid?
I don't think so. I think it's just trying really hard to grab.
Let's see, it looks like this is all part of its stalk.
Maybe.
For this? No. This one. Chuck said try scraping sideways. He also said you could grab it with a claw. They're pretty tough. Yeah.
I think we're finding out how tough exactly. Maybe, and then you can suck it up after.
Yes, it is a stalked crinoid, so it's not going to swim off. It's actually sessile.
Or it's sitting on a previous stock of one.
No, Chuck said it is it's stalked. Another thing he said that's. He's just got his stalk shoved under a rock. Oh, they can let go and crawl slowly, but they're not gonna. He's not gonna swim away.
Oh, he looks pretty defeated at the moment.
Poor thing. If it's way too difficult, you guys, we can give up. If it's impossible. He said to get as much of the white crown as you can. Oh man, this is a tough one, huh?
He's starting to open back up again.
Chris wants to know if it's playing dead. Yeah, if you guys can't do it, we understand. So we'll let you make the call. I know we have a squall coming in. You can do it.
Looks like he's holding on pretty tight.
Yeah. Yeah, I think he's got his stalk all the way back in that hole. And he's got a death grip on it. Which might actually end up saving him if we decide to give up. Did he go?
I don't think so.
Oh my gosh. Jeez. Oh, yeah!
There you go! Yay!
He's in! All right. Good job. Thank you for your persistence.
Oh.
That's all right. A big deal.
We'll sort through it. Got some coral rubble. Good. We have sample for me too. Or rather, the geologists.
Thank you, guys.
Yes, thank you very much. And that was in can 3, correct? Thank you. We're going through a shift change really quick with the ROV, so just hang on a moment. Can take a moment to...
Steph and Kim, since we are right here, I wonder if we can get close up of those round, that round kind of elliptical globe white sponge.
At 3 o'clock?
To the right of the crime. Yeah, just to look at it because I think it's different from the white geodia, like Petrofia, like that we saw before. And I just would like to, since we're right there, we could have a closer look.
Sure. Let me get the shift change settled out, and then I'll tell them to zoom in.
Excellent, thank you.
Thanks, Lars.
That's my husband.
That was.
Yeah.
Get it done. Yeah. Well,
Thank
you.
you. This is what I said.
We got a request before we're moving on if we could actually look at that sponge that's off to the right-hand side about 3 o'clock.
Oh, this was a good idea, huh? Mm-hmm. Yes, please. Hmm? Yep. Here, Chris. Like a Petrosid or Geodia? He looks like he has a crust. Looks like it's cracked at the bottom. Which would make it a Geodia, I think.
Actually, that's why it seems to me that it will be kind of more of a hollow species.
Hollow.
It doesn't. It's not like a geodia, like the one we saw before. If you notice, this oscule is very small, and that crack like that makes me think it might be a calcareous sponge.
Oh,
Which
calcareous.
can be more hollow. Yes, but I just wanted to have a closer look at it.
Okay, you're good?
Make note of it. It's a different one. Thank you.
Yep, okay, we're ready, pilots, whenever you are. No, I think I'm good. We got something swimming in front of you.
Oh, I think that was a ribbon fish from the top view, potentially. Or no, maybe not.
Yeah, some kind of fish. There he goes. We have another mound, don't we, coming up
Yeah, we
above
have
that?
a smaller crest coming up if we keep heading south. So
Are you comfortable heading that way? Okay. Yeah, we should have 2 more mounds to cover, at least, but there's a whole series here, so yeah.
we're starting to see overall less coral. It might be smaller, just we can't really see it, but overall the less of the larger corals. And just again to notify on the substrate, we're starting to see more and more of the Lophelia rubble, or maybe not Lophelia, but overall stony coral rubble that's in this here. We're seeing less slab texture from the overall limestone, a lot more of the softer sediments. We see a few isolated colonies off in the distance. A little fish making its way across the screen now.
Yeah, he is pretty tiny.
Bye, little buddy.
Looks confused. Probably blinded him.
Took him by surprise.
Yeah, hopefully when we get to this second mound, we'll have more coral cover between here and there, although I would imagine most of it's probably going to look like this with the thick layer of Lophelia rubble. Or Analopsamia rubble. It's probably Lophelia, but...
Possibly, but they do seem to be fairly well formed, so maybe we'll get lucky.
Yeah. We're putting in a ship move, and what this means is, well, the ship on the surface, and then we have 2 ROVs. We have the Sirius ROV, which floats above us, and then we have the D2 ROV. And the D2 ROV feed is coming in through feed 1, and the Sirius feed is coming in through feed 2, so you can actually see the ROV from above using Sirius. So Sirius sits above us and then acts like what we call a clunk weight. So it actually takes the ship movement away from the ROV, so the ROV below it, D2, can actually move around freely without being yanked. So in order to make a large move, we have to actually move the ship, physically move the ship forward, and then the Sirius will follow behind it, and then D2 can continue to look around underneath it. So it's quite a dance they've got worked out here. So we've got 2 a main pilot and a co-pilot who pilot the 2 ROVs, and then we also have a crew up on the bridge who are navigating the ship. So it's a serious teamwork down here.
And overall, to begin with, we have then a navigator talking to the bridge and the co-pilots, making sure everyone is satisfied and with the overall coordinates and directions we're going in. We have a wonderful videographer team, making sure we're getting all the best shots for you guys at home live, as well as images, still images, and more video that are then being recorded. And for those of you just joining us,
can we zoom in on this pink coral? Sorry.
Actually, no, that looks really interesting.
Go ahead.
For those of you just joining us, we're on dive 2 of this current cruise. We're on a ridge that is the easternmost ridge seen in the Million Miles Eastern Zone that recently has been mapped by bathymetry, so no one even really knew that this ridge was even here for the longest time. Just a few weeks ago, this whole region was mapped, and we're starting to get a lot of new data, and this is the first time anyone has seen anything that was potentially down here, as well as looking over the general morphology as well as the associated biology in this area here.
This is a bamboo coral we've zoomed in on. At the base of it, there's also a black coral, likely Tennisidopathes.
Looks to be internodal.
We're good with this whenever you guys are comfortable. Yeah, there's also a pink coral to the right of it. Oh no, it's okay. This is cool too.
Yeah.
Sweet. Small geodias, maybe. Those are sponges. We have a solitary cup corals at the base. You can see this one, the tissue goes all the way down to the bottom.
Yep.
And then we have this pink coral, possibly a black coral, I think.
This one looks like a black coral.
Maybe Bathopathes.
And a stylastrid in the back.
Mm-hmm.
And if we keep moving, I can...
Oh, it could be...
Oh, it looks like a little branching guy off one of those main nodes.
Yep, could be Tenosidopathes. It's very small, so...
And then if we keep moving off to the right, we'll get to that pinkish coral.
Yep, pink coral. It had a maybe Astroshima, a ophiuroid in it. Yeah, that guy. Is there another stalked crinoid right next to it?
Shh. We're not saying anything.
Yeah, so that's probably Astroshima maybe. That's a spaghetti ophiuroid on possibly bumblegum coral maybe. All right, Shirley said she's taken off, so have a good day, Shirley. Thanks for joining us.
This looks similar to the Paragorgia that we saw earlier. Not sure, though, it's Arborea.
Yeah, they're usually not this far south, right, is what Scott said.
Yep. But this is a highly large and intricate ophiuroid wrapping itself, holding on quite tight.
This association is pretty common where you have the ophiuroids associated with the corals.
Yep.
They can get higher up off the ground, and they can filter feed while they're attached to the coral.
It's a pretty big one, though.
Mm-hmm. He's pretty hefty. You can see the really distinct central disc. So these are actually related to starfish, or type of echinoderm. Also related to crinoids, which we just sucked up. So they're kind of the close relatives. Same phylum.
The skeletal structure here is quite bumpy. It's not as smooth as a lot of the other places, and I'm wondering if that's a direct relation to this tissue or if that's on the skeleton itself.
I don't know if it's... I've never dissected one of these, so... I'm not sure if it's tissue or... Chuck might know. He's good with the echinoderms.
I was talking about the coral, though, itself.
Oh, okay.
But Chuck did request to see the small crinoid that's just adjacent to the coral itself, so...
likely those lumps are the actual skeleton,
okay.
but maybe not. I'd have to pick it off with a...
Looks kind of like it a little bit, though. I think he said off to the to the right, sorry. Yep, the crinoid to the right. Another solitary cup coral.
Yeah, Astroshima is what I was guessing, Chuck. He said Astroshima or Ophiocrius, but they're both in the same family, so... Yeah, there's another crinoid, a white crinoid at 9. That Chuck wanted to look at.
This one. He wanted to look at this one.
Oh, the yellow one?
Yeah. So this one does have 10 arms, and it's yellow.
Okay, so the skeletal arm plates are covered with tissue. So that would be why the lumping on the...
on the coral skeleton?
On the... no, I'm sorry.
Paragorgia?
No, the
crinoid?
Yes, the ophiuroid.
Oh, the ophiuroid. Oh.
There's too many scientific terms running by me. So here's this yellow one. 1, 2, 3, 4, 5, 6, 7, 8, 9. That's the 10-armed yellow one, isn't it, Chuck?
Yep.
Isn't that what you were after?
If you want to dial in and talk a little bit about this guy.
This would be easier to collect than yesterday. We do have some weather coming in though, so I might have to cut you off. We might have to pull off the bottom. I'm not sure if it got worse or not.
The base of the arms lack the pinnule side branches.
Atelocrinidae? Is that the same one you were after yesterday, Chuck? Or no?
Seems he's having something protrude from the center. I guess, is that part of his mouth he's protruding?
I'm not sure.
Nope, not needed.
Okay, we can move on from this guy. I don't know, it looks exactly like the one we saw yesterday, so I have no idea.
Thank you, pilots. So we are coming up over the southern end of this main mound. We're going to be heading... Oh, can we zoom in on that dark feature there? Maybe it's another
Fish, maybe?
fish.
Orange roughy?
No, that we saw earlier, actually. Is this the same? He looks
think so.
darker.
I think he's the same. They can change colors.
They do? Wow.
Lots of fish change colors. The groupers have like, I don't know, 4 or 5 different color morphs. They're pretty cool. Yep, I would call this an orange roughy. Or a Darwin slime head.
Is that the common name?
Yes, that's the old common name, till they started fishing it. And no one wants to eat anything called slime head, so they changed it to orange roughy. He's pretty cool.
He is.
He's got lots of plates on his face. Almost looks mechanical.
Yeah, almost like all the gears are connected together.
He's
Bit
steampunk.
of armor.
Steampunk fish. I think we probably scared him.
Maybe.
Doesn't know where to look.
Down seems to be a safe bet. It looks like, I wonder if that's the average shape of his mouth, but there seems to be maybe some parasite or something on it.
That's possible.
Oh, just got out of view.
You can see the shininess. It's almost like he's covered in plastic, right? That like clear, this clear coating over shiny scales. He's really neat.
He is. Do they have teeth?
I think so. Most fish have teeth. I know it doesn't have teeth like the goosefish, but... Oh yeah, you're fine. Whenever you're ready.
Oh, he's a decent size, a little over 10 centimeters. And Ken Sulak on the chat room is saying, a few moments ago when we were seeing that long silvery item that kind of looked vertical to the water column, it was in fact a fish. It's a... Oh dear.
You want to try?
Paralepididae or a barracuda. Maybe though, Notolepsis risoi. This intense little silverfish typically orients vertically and drifts down into the substrate and can bounce up with vigorous vibrations at the end of the tail. And it kind of almost resembles more of a pogo stick along the substrate as it travels vertically up and down in the water column. We're starting to see a little more biodiversity here than in some more of the barren areas again. Oh, look, we have another guy. What was his name?
Which one? Where am I looking? Oh, Chimera. This is an ancient shark. So they're related to sharks, if I'm remembering right. Then they have a cartilaginous skeleton, like a shark does.
They look ancient.
Mm-hmm.
They can swim backwards, apparently.
Yeah. Most sharks can't move their pectoral fins. They're fixed.
Yeah.
So he's pretty remarkable. Oops.
Scared him a little.
Looks kind of like a kite.
Yeah. Almost like between a shark and a flying fish.
Mm-hmm.
And Ken Sulak calls it a Chimera monstrosa.
Chimera monstrosa. Good to know. Thank you.
We're seeing some little sprouts of some octocoral around. Amongst now a lot of coral rubble. We're not seeing that same framework structure being kept as we did near the top or at some other slope areas.
Are we coming up on the second slope? Or is
Not
this still the
just yet.
first one? Okay. I am going to shout out to my friend Amy, who just had a baby. And she is on the sofa watching us in our live feed while she nurses.
Oh,
So hi,
congrats.
Amy.
Oh, we just passed another little shrimp in the area. We're seeing some smaller octocorals around. So Ken Sulak just chimed in again on the chat room. So that chimera that we just saw a few moments ago has shark-like teeth. It's fused into what looks like a beak. It's more like parrotfish-like, and it's used to crush any hard-shelled prey, such as mollusks, so it can eat it. And it uses also electroreception to find any prey buried beneath some coral rubble or some sediments. Thank you for that, Ken. And he also chimed in that the fish we saw before that bronze-ish and reddish little fish is called a berry siform fish. It's Hoplosteithus. Did I say that right?
Hoplostithius. Hoplostithius.
There are two species. Sorry, I'm not going to say that now. I butchered the first one already. They're associated with the type of habitats that we're generally seeing, so among some corals and some frameworks in the overall vicinity. So they move slowly among the corals, looking out for a potential prey.
So we're looking upslope here. We got a few gorgonians scattered about, mostly coral rubble bottom. And we are heading south, trying to make our way to the second mound in a row here. We got about a 45-degree slope, no rugosity, which means no hidey holes for fish to live in. It's mostly some small gorgonians. There's one rubble ball, one rubble rock.
Probably roll down from the top.
Sure, Kim can find some way to find it, make it interesting. You can see the top of the ledge here, right above the screen on the left.
Yep. That was a little terrace-like feature. Seeing quite a few sprouts of some octocorals. Quite a few sponges also. A few little sprouts of, I think, are stylasterids.
Oh, okay, so that fish is not technically an orange roughy, but it is related. So that's what Ken's letting me know. It looks like we've got a field of tiny gorgonians, maybe Unicella? Small, white, sparsely branched. There is just a field of them on top of here. Maybe another Astrosema, or Ariosoma, excuse me, at 3 o'clock, which is the pancake urchin.
Was that an urchin?
I think so, down at,
okay.
we blew past it. And then looks like maybe a sponge up here on the left at 9. This
on
white,
the far side.
Yeah, this white mounding sponge. But otherwise, it's pretty barren. There's not a lot of, not too much. I'll get some more maybe on the next mound. See if we could just take a quick peek at this sponge at 9. I'm just gonna poke in real quick and Pacastrellid, or could be Hexactinellid. Ooh, he's neat. That's probably Hexactinellid. You can see all the really fine hairs sticking off it. No, that's good. If Chris knows what this is, if she's still on the line.
Yes. No, I'm here. Yes, on the line. Definitely an exact name, but I don't know.
Don't know what kind.
The gene for the family, no.
It looks like Vazella does that, right? With his like really fine spicule hairs coming out of it, but it's definitely not formed that way. I wonder if they're the same family, maybe.
Family. It could be. It could be the same family. So.
Yeah, and you can see there's a Faria. It looks like Faria at the far, around, 7 o'clock, the far lower left corner. There's a small Faria, different species of Faria sponge.
With a squat lobster.
Yes.
Oh, you have squat lobster on it.
And we return like a similar geomorphology than yesterday. What do you think?
I think we're good pilot.
The sponges we picked up yesterday?
No, the geomorphology of substrate, geomorphology, mostly coral rubble with black color to it.
The lack of substrate yesterday?
Yes, the substrate yesterday that's mostly rubble, coral rubble
Yeah.
that was covered by this manganese, iron manganese material.
Yeah, so yesterday it looked like it was a lot darker, right? There was a lot more darkening of the coral rubble than there is today. It looks a lot lighter. Maybe this is newer.
Well, you can't really say that to be certain, and it's more relative than anything because we did have a lot of the white patchy sand areas, which made the coral rubble look also very dark in comparison to the white patches. So we still see a lot of that iron manganese encrusting on the corals themselves. There are coral skeletons that don't look like they have a lot of that encrusting, if any at all, but we can't say for sure if those are younger, if those are older, without some absolute age dating on that. It just maybe so happens the crust formed around them or some sort of biologic impact turned them over, or something of the sorts happened. Maybe the current swept through too strong and turned some of these corals over that weren't officially crusted, but we do see a fair bit amount of it. Maybe a little less because at the top of that ridge there was some really high intense limestone, but that was also fairly encrusted by a lot of that iron manganese coating.
Okay. Thank you. Now, that's quite impressive how different one mound from the other mound. And
I think we haven't gotten to Mound 2 yet. I think we're still on the slope of Mound 1. So the top of that, where all that life was actually the top of this same mound, and we're still just transecting across the top.
Okay.
Yeah, we're on the downslope on the way to Mound 2. So hopefully when we get to Mound 2, we'll have a lot more of that life, a lot more rugosity and places for coral to grow on Mound 2.
Okay.
Yeah.
And just to note here, I mean, it doesn't look like there is a high intense current passing through here. All the corals still look fairly stable. No branches are really caving over on any particular side, indicating higher intensities of current systems. We have, if you look on the Sirios view, there is a current coming through from the top to the bottom. So in the direction the ROV is facing, due south, I think, or
Yeah, 160, yeah.
okay, so the current system is probably coming in that same direction there.
We're also traveling that way too, so maybe it's just us moving.
Maybe it is just us moving. Can't say for sure, but based off the corals themselves, we're not seeing a lot of them wave or flex in the direction of the current system, so it still seems to be quite adequate for coral growth in this area, but maybe there just isn't enough of that exposed substrate for the more dense corals to really colonize and start to grow stronger.
There are places that are in the Gulf Stream we've been driving where the current is so strong that Gorgonians are literally laid almost flat to the ground.
Which makes the case, I mean, if they can survive in that kind of environment, then how did they originally colonize on it?
Right. Yeah, because I think it varies, right? So it goes from really, really fast one day and then really slow the next.
Potentially. I mean, it also has a tidal regime in that area where it even changes every 6 hours, just like it does on the shallow ends.
Right.
So maybe that's just enough time to colonize, stabilize, and
get settled and stick.
hold on. While we're here, I'd like to, since we had the shift change, I'd like to introduce the control room with our new shifted ROV pilot. So if you want to go ahead and introduce yourselves.
Hi everyone, this is the front row. My name is Sean Kennison. I'm currently piloting D2. We're all from the Global Foundation for Ocean Exploration. We're made up of engineers and videographers, and we're responsible for the ROVs and all the video products you see today. And then sitting co-pilot to my right.
Fernando Aragon.
Then sitting nav to my left. And doing video right now.
And in video is Caitlin Bailey.
In the back row, clipping is Tara Smithy. She's headphones in. And again, we're all from the GFOE.
Great, thank you so much. And these guys are responsible for navigating a lot, navigating, period, the ROVs and making sure everything is stable so we can make sure that we at home and on the ship can see the most and get the most out of these explorations. And we are very grateful that they're on board and can navigate the ROV and make it look really easy. But let me tell you, it's a lot more complex than it makes out to be. And the ROV itself is a massive piece of machinery standing at over 9 feet tall and over 10,000 pounds. And if you want to give a few of the specs, that would be great.
So, yes, comes up a lot. D2 is much larger than most people think. D2 is about 10,000 pounds, 9 feet tall, 12 feet long, 8 feet wide. A large piece of machinery. But D2, even though D2 weighs 10,000 pounds, he's actually buoyant in water. And that's accomplished by, on the top portion of D2, it's made up of syntactic foam, which is the large white blocks you'll see on top of D2, especially when looking from the Sirius view. And syntactic foam, in air it's still very heavy. All the blocks together are still thousands of pounds. But in water they're buoyant. And that's what provides the buoyancy force that makes D2 actually a couple 100 pounds buoyant. So D2 actually floats in water due to that. And then in the front we have 2 manipulator arms that we use for sampling. 7 functions each, hydraulically driven. And then we have various tools and such that we can use to assist in sampling and collections. And then we also have installed a suction sampler, which can be grabbed by the manipulator arms, and then that provides a siphoning force that can inhale different samples. And then we have 6 jars that we can use to collect and separate samples as we go. And then we have 4 separated, we call them bio boxes for biological samples. And those boxes don't hold up to pressure, but they hold up very well to temperature. So the samples we get as they come to surface, those boxes are able to maintain the temperature of the samples, which help with preservation. And then we have 2 large rock boxes in the very front of D2. One has a lid for some smaller rocks so they don't fall out, and then for the heavier rocks, where we're not concerned of them falling out, it's an open lid. And that's, again, all in the front of D2 on a drawer system, so it can actually extend from D2, which can help with the sampling process.
Great, thank you. So that's just a small introduction to everything that Hercules can do, and D2. Again, they make it, D2. They make it look a lot easier than it is. And I think we're at the southern end of this mound now on
The
near
base
this feature. Okay, but we're seeing some exposure to some of this hard substrate, and right immediately as soon as we have this more tough substrate where these corals can settle upon, we're starting to see the more robust coral features that we're getting in that. So they really do enjoy the fact that they can settle on something really hard where it's not moving, they're guaranteed solidarity and can keep thriving.
Yeah, to the left, it looks like we have more Liopathes. It's off screen now. If you have time, but if not, no worries. Yeah. Yep. So we've got a couple of large gorgonians here. This red one in the background, he has Lyopathes. That's a black coral.
Quite large in size. We'll get a close-up, a better view of it, but Scott had mentioned earlier in the day that some of these collections, one of the more recent ones in the Pacific that was collected, has recently been found to be over 4,000 years old, and it was still living. So,
Yep, they can live a long time.
and it actually has some Lophelia attached to it on the side there, and it looks like some branches fell.
Yeah, it looks like they're dying off. So this one has that same lumping texture on the main stalk, right? What we were talking about earlier. The lumping texture on these is actually the tissue, because when you strip them down, it's like really almost shiny. The black skeleton's almost shiny underneath it. No, we were just looking. It's just a cool species. There's Lophelia on the left, on the right, excuse me.
And are those zoanthids, or is that another coral?
Yeah, it looks like a zoanthid infection. And they're parasitic, yeah.
Yeah. Not sure if the currents is from the thrusters or if that's the natural current, but these guys are just, you know, slowly wafting in the current there, so it gives some indication of what the direction is going in. Okay, yeah, so then that's definitely the current system there. So, you know, just strong enough to deliver food and make everyone happy, but soft enough to continue and promote more coral growth. We're happy if you guys are all ready. There's a little black guy at the base of the screen in the center, if we want to zoom in quick on that. Off over now to the right a little bit. I don't know if that was a hole or if that was some sort of fish. No, it looks like it was just the hole there. Sorry. Oh, there's a little shrimp there, though. There's more Stichiopathes also on the lower end of the screen. And we're also seeing there's still quite a bit of coral growth on this area. So even though we're on a slope, that substrate that they're growing on is still pretty hard. They don't have to worry about really a lot of that tumbling coral rubble coming down.
Looks like another one of those barracks fishes at 6.
Yep.
Maybe. It's flattening out here. We're probably way down at the base of this mound. Oh, we are looking down. Okay. Okay.
Oh, we even have another Leopathes there, a really big one.
Oh, I see, yeah.
Oh, wow.
Yeah.
That's level? Okay. So we are scaling now down that ridge. We're about halfway between, halfway down the ridge. About a fourth of the way to the other pinnacle.
We have a dark spot at 5 o'clock, if you guys can take a peek at that. That's different. Yeah, 5-ish. See the dark spot? All right, closer to 6. Yep, there you go, you got it. Yep, the dark spot, I don't know what that is. It is very dark. It could just be a
It's
hole.
a hole.
Yeah.
It's probably another hole. Can
Jeez.
we get a quick zoom in?
Yeah, it's just a hole. Maybe somebody lives in there. But right next to it, we've got a nice Aphrocallistes.
Yep.
Right to the right, that's a glass sponge. On the right-hand side, I'm sorry, the left-hand side. Yep, we just got a hole. Maybe somebody's house.
Populated with some sediment and coral rubble.
So there's a really cool Aphrocallistes. You can see it's like prop roots have grown really long.
Yeah.
I don't, they're not really prop roots, but, you know.
For the sake of it,
Yeah.
holding it up.
Yeah. Those growths.
And for those of you just joining here, we're starting to see a lot of coral gardens and forests near the crest of that ridge. So we've actually extended this dive by 2 hours. So we'll be wrapping up here approximately 6 o'clock local time. And then we'll have our dive call at 6:10. But we're hoping to see more of these coral garden features at the next 2 little crests that we have on the projection of the ROV dives.
Can we zoom in on this flytrap anemone that's at 10-ish?
What's
we
also
float
at
by?
the base of that? Oh, is that just a sea urchin, a pancake sea urchin at the bottom?
Yeah.
Okay.
Ariosoma. So this is a type of anemone. We call it a flytrap anemone, and it could be many species. I think I know it as Astroshema. Nope, I'm blanking. But it can be many different types of species that we don't really know. You have to physically pick it up and look at it because there's many forms, and some of them are even in different families, I believe. And this is sitting on a black coral, which is called Leiopathes, which we've seen many today. I believe we collected one. They are one of our target species. I think that's one of the first or second things we got today, so we've already collected him. The black coral that it's sitting on. It's also got a zoanthid invasion around it.
And then another octocoral taking advantage of some empty skeleton. And is that another flytrap anemone behind it?
It is right behind it. You can see the lumpy, and that's its mouth margin, right, where the tentacles grow. They're much smaller. Yeah, so they're good. Thank you. And hydroids. Lots of hydroids on the dead part of this Leiopathes. Yeah.
And some barnacles or tube worms in this area too.
Actinoscyphia, that's it. Thank you, Scott. It was there somewhere in the back of my brain, buried amongst all the other genera I have stuffed back there. And you're right, we have only seen them on Leiopathes today. We haven't seen them on any other coral. They've only been associated with the Leiopathes. But we've seen probably 4 or 5 of these. And honestly, Leiopathes is a good place to be. They're nice and tall.
And they grow big and bushy.
It's a good spot to sit on and catch stuff.
Where's the base of this guy? I still haven't found the base of the coral structure.
The base of the mound?
The coral, yeah. I think he's just hanging on inside there, that ledge, right on the edge of it. I think we're good. Thank you. Just got lost in all the bush material.
Yes, Leiopathes is a champion mucus producer. when we pull up a little piece of it, it's going to be super slimy. Sometimes we get these and they're really large. We've pulled up really large ones. We don't do it very often because they're really old. But when we bring them up, they just slime everywhere. It's snot.
Good luck with that.
I know.
I think we're coming across one of those columnar sponges again.
Oceanapia.
Oceanapia.
We collected this one earlier. I think this is the fourth one we've seen today, roughly. We got a piece of this, but we think it might be either undescribed or a totally new species or passing below us right now at 6 o'clock. Only if you want to stop. We've seen it a couple times already today.
Can keep moving. I think there's interest to try to see the other crests of the mounds.
Yeah, you can probably blow through here pretty quick. Sure.
Yeah. There's another one of those ribbon fish that passed through. Potentially a, what was it referred to as?
I forget what he called that.
Barracuda? Yep, Barracuda.
Barracuda. There's another jelly of some kind that just went past. Looks like we might have Lamanema? Nope. A pink lip anemone at 6. Would be Sagartiidae. Would be at 6 o'clock, I think. We have some sparse branched gorgonians. There's sea fans. There's soft corals.
More sponges as well. Not to discount those.
Right. We are at the base of this almost. Are we still heading down slope? We're still heading down slope. We're almost at the bottom.
There's a stocked crinoid.
Oh, nice.
Oh, you're good so far.
We got a rat tail at 3. We're hoping the coral cover will pick back up when we get to mound number 2. Maybe a cutthroat eel there again. It's at 6 o'clock. We got something dark moving in the center. If we can get a quick zoom on him. The jellies, yep. Oh, nice.
A black jelly.
Yeah, it's actually a red. If we get close enough, he'll lighten up in the lights. But that's a perfect example of how red disappears in distance underwater, right? Now it looks black.
Yeah.
When we get close, it'll be bright red with a clear bell. Oh, he looks stuck. Oh no, he's stuck.
That gives us a chance to get a quick look at him.
Right. See, now he's more of a maroon color, right? So they're he keeps getting stuck. So this maroon color disappears with depth. So in the deep sea, red is actually a camouflage.
Oh no! Now he got himself really stuck. And Scott is calling him a helmet jellyfish.
Okay.
Paraphylla.
Paraphylla. That's the one. That's the word I know.
And it looks like he Either there's a parasite attached to him or that's attached to his tentacle. I'm not entirely sure.
I don't know. I don't know what's happening.
He looks
with
very
him.
confused.
He's having a rough day. I mean, it is Saturday. I guess that makes sense. But
It almost looks like a parasite on him, actually. It's attached to
It might
the
be.
top part, the helmet. Can we get a quick zoom in before he gets away? It almost seems to be eating through him. Or if that's part of his internal structure, the jelliness that's
he maybe got bit.
protruding out.
Oh, poor guy.
It's a rough day, isn't it? Yeah, it actually looks like part of his internal structure got ripped out a little bit. Maybe that's why he's behaving
Injured.
a little wonky. And Scott says they're usually pelagic, so they're not usually at the seafloor, and so interactions with them are pretty rare. And he's just getting himself caught left and right on things.
Yeah.
So
Poor buddy.
could be due to his injuries.
Maybe he got poked by something.
Or tried to be eaten by something.
Yep.
Oh.
He does not look happy.
No. Thank you, ROV.
Yeah, leave him alone, huh?
Yeah. Oop, and just to correct myself, that it's not that it's a rare encounter, it's just that they're rare to be part of the actual substrate. They usually don't hang out in the benthic communities. They're more of open water species.
Well, and that makes perfect sense because as soon as he touched the substrate, he was stuck to it.
Yep. Doesn't look like his favorite place.
All right, we're going to start sloping up now. Okay. Yes, that's a mound.
Oh, we have some even more Leopathes. Some more sponges and some octocorals in the background. We have quite a few of those Leopathes actually around. Some more slab-looking features. Here we have a lot more of that coral rubble with some softer sediment, and you can see immediately the difference. And this is a good shot to get the comparison between the hard substrates and some pretty dense corals and large corals versus the more softer sediments where you don't see a lot of that coral growth, and if you do, they're usually not as large or robust as those that are on the harder substrates.
It looks like we've got corals coming back.
Yep, so now we're at pretty much at the base of between those two mounds, and we're going to start to climb now upslope. We have more Leiopathes.
Yeah, that's a big one right there in the middle.
Big one, or is that two?
It might be two. Oh,
Still
maybe
quite
not.
large, some large ones.
And another one right there. There's a cool white anemone at 9.
Now approaching center.
He might just be a fly trap, maybe? He's got a really wide mouth.
It's not very pink in the center. Is that a normal characteristic for fly traps?
Most of the fly traps we see are that really pink and orange. This might be a corallimorph. Yeah, I believe this is like a bubble tip corallimorph and not a fly trap, right? The fly traps have much wider mouths, really distinct margins. There's also 2 Cerianthid anemones. There's 3 of them, 4 of them, 5 of them. Okay, there's a bunch of Cerianthid anemones on this rock, and they are tube dwellers. So they sit down on the substrate and they can suck their heads down into their own tubes. So if we were to get close enough to them, they would pop in. And then we've got Aphroclistes again, which is right in the front at 6 o'clock. And a nice white gorgonia, maybe corallium. And a bamboo coral. My gosh, there's so much stuff here. A bamboo coral is right next to the white anemone.
So do those anemones house a skeletal structure?
The corallimorphs, I believe, do. No. I have to look that up.
Okay. Or maybe someone
Scott.
on the chat room can also chime in if you have time even to come on and dial in.
I thought the corallimorphs could move, but
that doesn't mean they can't have a skeletal structure.
That's true.
No, corallimorphs do not have a skeleton.
Okay. Might be a long fish in the background on the bottom at 9 o'clock.
And Scott says they're normally, corallimorphs are, in quotation, called naked corals.
Naked corals.
Is there, what is the main difference then between a soft coral and a corallimorph?
Right, so corallimorph looks more like an anemone.
Okay. And they're called naked corals because their polyps are much like that of scleractinian corals, but they lack a true skeleton. Really interesting. Thank you, Scott.
I think I see a starfish at 9 o'clock, just to the left of that white gorgonian fan. Looks like our outcrops are increasing too. We've got some overhangs.
And with that, we also have quite a few scleractinian corals, but the last quite a few scleractinian corals we've seemed to be hanging off other coral skeletons and other corals either living or dead. So this is quite interesting, the fact that we're not seeing actually any on the substrate itself, but all hanging out on other corals.
Yeah, there's a star.
Next is some sponges
Yeah, he's near a yellow sponge and a white sponge.
and a stylaster.
Mm-hmm.
If that's above it.
Yeah, this is another one we saw one of these earlier. The nice lumpy skin. And I am unfortunately blanking on the name. And there's a bryozoan right next to him on the, you can see this mesh netting at 7 o'clock. Sure, if you want to zoom in.
Zoom in also on the bryozoan,
The bryozoan, the mesh.
the little mesh.
Yeah.
That sponge around 7 o'clock, kind of massive and tan, looks like a pacastrelide.
Castrellid.
A little more to the right, please. The net-looking
Astrophoid.
piece.
This one is an astrophorid. This white one.
That one in the left that has one oscule on the top. Actually, I can see now two of them. One in the right, and one in the left.
Or astrophorids.
The spiky thing at the bottom, which is another sponge, probably Tetilidae.
Which kind?
Family Tetilidae. So we have something in the bottom that is very spiky, and it possibly could be a Tetilidae. Then you had to the left, you could see a more massive sponge with an oscule with a membrane. That's a Pacastrelide.
Pacastrelid, okay. And this white mesh is, I think, a bryozoan.
With the little hydroid growing out of the stem. Is that a hydroid or a crinoid? Maybe a hydroid.
I think it's a hydroid. He's tiny, tiny, tiny.
With an ophioid hanging around behind him.
All right, we're good if you guys want to back out. We're happy. We got bamboo coral, stylaster. This nice gorgonian on the side. I think is also a bamboo coral.
We have another crinoid also up ahead that seems to be hanging out on the ledge there.
And another one, another stalked one underneath the rock.
Can we swing more to the left, please? I want to make sure we capture some of the footage of the scleractinian that's hanging off the Leopathes. Exactly, right there.
The white coral at 10. Might even be some madrepora underneath it, this pink ball.
Yep. And Scott says maybe that little guy that was coming off the bryozoan is in fact a crinoid, not a the bryozoan is in fact a crinoid, not a hydroid. And this is really interesting. This is now the third or the fourth case that we've seen where we have a scleractinian coral growing off another coral itself, usually a Leopathes. They haven't all been Leopathes, but it's really peculiar to see that such a coral will be growing off another coral. Usually it's vice versa, or the coral itself is just growing flat on the substrate. So this is quite interesting to see that this is kind of a popular case here in this region.
I wonder if the Leopathes is a more stable substrate than anything else it can attach itself to grow, to sit there long enough to grow that large.
Maybe. But then the substrate it's sitting on has to be there pretty long too. So I don't know if there's a general preference or it just really happens to be all coincidental. But these polyps look pretty happy right now. They all seem to be out and trying to graze on the passing current. We're starting to see various hydroids in this area. You can pull out now. Thank you, pilot.
There's a pink one at the bottom.
Oh,
that's
yeah.
madrepora, I think. This is the first madrepora I've seen this dive, I believe.
Looks like it. I'm not sure if it's madrepora.
To get closer. Yeah, it's got the zigzag pattern. It's just that its polyps are out.
Oh, yeah.
Look at that. I'm not sure the species. I know we're after Madrepora aculeata, but I'm not really sure what the species is.
Could be Carolina.
Mm-hmm. Awesome.
Looks generally interesting.
Mm-hmm.
Are the lasers on? Just to get a general size orientation. Thank you. So it's about 15, maybe 20 centimeters long. So a pretty small guy overall, but this is the first one that we've seen of the Madrepora. And I think we're good. Thank you, pilot. Want to get moving? We're going to keep moving up, scaling up on this now different mound. We have quite a large yellow coral off to the right-hand side. I'm not sure if this is the same coral that we were debating on earlier and whether or not we could tell what the species was.
Yeah, lots of bamboo corals though. All these these 6 or so flat planar corals on the left, on the right are.
They seem to have another coral almost intertwined with it. If it's not the same coral,
it might be two.
It is a little interesting. Might be two planar. It's kind of brownish, yellowish. Can we get a tight zoom on the polyps? No problem.
Nice. I get the ophiothrix on at 3, hanging out in the branches. Any ideas, guys?
This looks different than what we saw earlier on the polyp structures. I would not know what this is.
It looks similar to the bubblegum coral in the texture, but the color's all off.
Yeah.
So maybe it's paramyriceid. Oh, plexurid. Yep. This is paramyricea. And then Scott said, I think it's the same plexurid that we collected earlier. So this was of interest. We weren't sure how to identify it. So we collected a piece. So we could see its polyps closing. We blow dust at it. All right, guys, we're good whenever you are. Here's lots of bamboo coral here.
And quite a bit of the coral rubble, but all in relatively good framework still, holding on to that framework position.
So Scott was saying that part of the explanation is the possibility of why Lophelia was using the black coral as a substrate as opposed to the rock was that some octocorals and black corals can produce wide holdfasts, and that helps stabilize the underlying substrate. So basically, they help prevent local erosion around them, or at least where they can grow large enough and to perhaps serve as substrates for others.
Can we actually go back down really quick off to the right-hand side on the lower left of on the lower right of the screen? There seemed to be something weird looking, a little lower. There, now straight ahead. What is that, do you think? Just some...
Is brown?
Yeah, rubble or something decaying?
I'm not sure.
Or?
Can we zoom in on that? It looks like it's like iron. Maybe?
I don't know. Can we get closer?
I think it's definitely a rock formation.
Some form of maybe encrusted skeletons, or I can't tell if it had any structure that may have been man-made. It looks pretty jagged and rigid, but I don't know.
Like that iron, when the iron,
Yeah, something
oxidizes.
that came off of a boat or something that fell and is now all rusted. Not sure.
Weird.
With a little anemone or a strawberry coral?
I think the strawberry coral and a
sitting
dahlia.
on top. Okay, thank you, pilot.
Oh, Chris said it might be a sponge.
Might be a sponge.
Hi.
Hi.
I'm looking at the part below. You could see like yellowish parts of it. So
Yeah, maybe zoom in on that larger area that we're looking at right now,
a
too.
massive sponge. There's more far ahead, with the front around in the middle of the image, like in the center of the clock. There's more coming.
Yeah, we're zooming in. You might be on a little bit of a delay. Are you at home?
No, I'm in the
ECC. Okay, so
Harbor Branch.
Yeah, we're zooming in on a bigger chunk of it now.
The same rusty color here again. So it might be an organism here. And a sponge is a candidate because it just can grow in these amorphous forms. And
yeah.
It could have an internal,
if this
more yellow.
If this is a rock, would it be of
color.
interest to collect?
I don't think it's a rock.
Okay, you think it's probably a sponge?
Either a sponge or some sort of material that's been rusted, but it doesn't look really like a rock. Is there any way we can possibly try to crush a piece of it to see the strength? Not necessarily for a sample collection, but just to crush it to kind of see.
Give it a good squeeze.
If it's more spongy or...
Yes, it seems quite interesting to find out what this is. Because if you can see, there's kind of spread yellowish tissue looking around the area. So it could be something really different, Some type of hemospongia.
Yeah.
I think we're going to pull the arm out and give it a good squish. Figure out if it's brittle or squishy, if it's rock. Now take a close look and see how it responds to this arm.
It's cool.
Huh. It just crumbles.
Yeah.
Oh my God.
You think it's
It's
sponge?
releasing some sort of green thing. Something's decaying.
Yeah.
Oh my God, what is this?
Can we get a zoom now on that piece?
Green part? You think
That
it's
is-
rusted metal? What the heck is going on here?
I don't know now. I honestly now have no
No
idea.
idea what that was. You think it's sponge, Chris?
It could perfectly be a sponge.
Do you think
could.
it's
I mean,
dying
it's a sponge
sponge?
that is releasing mucus, and it has that. But it looks very strange. I agree with you. And, you know, I'm not an expert on materials and minerals to
Yeah, it just
see what
collapses.
this could be. No, it's too breakable. Yeah.
Well, he could suck some up.
No, this looks chemical.
I don't think this is...
Wow.
Could this be sulfur? No. Sulfur doesn't occur in this area, at least not naturally.
You want to suck a piece up? Since we have
No,
no
Scott-
idea what it is.
Scott thinks to think it's just a really big dead sponge that grew and it's now
If it will be
decaying.
there, it will be rottening and it will be more stringy or... It
Be
will
like
different.
Yeah.
snotty? When it turned to like
Behavior that we are
rock.
seeing. As long as it's not kryptonite,
It's not
why don't you take a small piece, you know? Scary looking.
well, we don't have Superman on the boat, so even if we picked up kryptonite, I think we'd be good.
Okay. Let's say with that. But it's very
I don't know about
strange.
that, but these guys are pretty well equipped to be.
There's definitely a outer layer that you saw it in other pieces. You could see kind of plates, almost plates, and there's several sponges that have this kind of internal color and that have sulfur compounds.
Mm-hmm.
But it is a little bit exaggerate the weirdness of this.
Yeah.
We should just grab a piece of it.
I don't know
Yeah.
if it's worth grabbing.
Okay.
I guess if we find it again, maybe we can consider it. I don't know.
Okay.
Just by the rareness of, I will grab it a piece because you're there.
Yeah.
Yeah.
And it's such a strange
Texture.
Item.
that
Yeah.
we
And there was actually another piece, you know, we saw first. It's actually the second one, right?
Yeah, it's too crumbly to really be any sort of substrate that I would associate being down here that maybe wasn't an animal. If we could actually suck some pieces up that
Okay, a little piece, yeah.
that we broke up a bit.
Are we going to call this biological?
I have no idea.
Geological?
What are we calling this?
I would call it kryptonite.
Kryptonite.
There's no unknown box.
Unknown, really. And is Scott or anybody giving us any input on this?
I haven't heard much.
No.
I think everyone is as stumped as we are.
But he's thinking that it's rock with iron. Yeah, it could be something completely chemical. A rock.
Maybe. I just am not familiar with anything that is this crumbly with this kind of texture and color that normally, that I
And
would
that
ever
gives all
associate
these
with
colors,
carbonates.
This exudate, no? And this is an exudate that could come from a sponge. That's why
Yeah.
Okay.
That's why, Scott did say that he thought it was also a dead sponge, so if it starts to smell really bad, we'll let you guys know. I think we'll call it maybe biologic/geologic. I don't know.
I put a question mark.
Yeah, there's question marks all around for this dive. What I'm also interested in is that just on the left there's some column features that look like it was standing at some point. I don't know, attached to some substrate. But let's let these guys do their thing.
I didn't see how much you grabbed. Yeah, we just need a small amount. I don't need a big chunk, so if whatever you guys can grab real quick is good enough. That went in suction bucket 5. Question mark. I guess for now, biology is fine. A biological sample. If it's something different, can we edit it? Or no? Oh, just leave it as B then. Yeah. All right, so we're going to move on. We're headed up slope of the second mound. Give everybody a recap of where we are and what we're doing. We're on dive 2 and what we call Million Mounds East, which is on the Lake Plateau off the eastern coast of Florida in about 850 meters of water. Well, now we're at 750 meters of water at the top of these mound feature. It's a series of mounds that run north and south with a scour mark to the eastern side at the base. The base of this mound features about 830 where we landed this morning. And right now we're at 750 at the top of the mound. We hit maybe a 45-degree slope along this edge as we traveled up it throughout the day. The top of it was mostly covered in slabby rock material and lots of gorgonians and invertebrates. So for that reason, since we had such an abundance of life at the top, we decided to extend this dive by 2 hours. So we'll be in the water today until 6 p.m. Eastern Time. Let's see what else. The dive track we planned was 820 meters long, but if we're in the water for 2 extra hours, we might go further than that. Most of the bottom is covered in Lopelia rubble. But we're only seeing a minimal amount of actual living Lophelia on the top. Most of it's attached to these Lyopathes or black coral species that are pretty common or abundant up here. They're quite large, which means they've been here for many years. The longest living Lyopathes that has ever been pulled out of the oceans was about 4,000 years old. Give you an idea how long they live. We've had many species of gorgonians and anemones and squat lobsters and shrimp, and it's just been a great dive so far. I think we've seen 3 or 4 sharks, 2 chimeras. Yeah, it's been a great day. So we're going to continue on this second mound here we're at the top of. And the ship is making an adjustment so that we can continue to move forward. Yeah, speak of Mound 2. Yeah, just keep going. If you get bored and you want to go like to the eastern or western side and take a peek off the edge, that's fine too. Yeah, slopes the edges usually have a lot of the coral, which is really what we're interested in. Keep it entertaining for the people shoreside, and frankly for me. And since the geologist Kim is out of the room, I get to make those calls, right? Oh, she's back.
Sorry, what's happening? What calls are you making?
I said favor the coral spots over the rock spots.
No. Corals are definitely
Can we look at 6 o'clock off screen, please? Right below screen, what is that? Yeah, that white lumpy thing. There you go. Oh, okay. Ooh, we might have Lophelia. Okay, so looks like a dead Lyopathes with a bunch of stuff on it. Maybe parts of it are still alive, but this has mostly been encrusted with, inhabitants. We got zoanthid, pink zoanthids. Looks like Lophelia, but a dying Lophelia. And there's tons of solitary cup corals at the base.
Yeah.
There's like probably 20 of them.
Can we get a quick pan over the whole thing? It would look interesting to see exactly how many different corals there are in here. Is the top a stylastrid, you think?
I don't think so. I think it's a white gorgonian. This white one up here at noon, I think it's a white gorgonian. I'm not sure which one, but... looks like we have an Ophiothrix ophiuroid, which is a genus of brittle star. You can see his legs moving around in the Lophelia clump over here.
Yep. But that Lophelia looks pretty much dead.
Yeah, he's not having a good day. Probably not having a good couple months.
Yeah.
We do have some hexactinellid sponges at 9 o'clock. Those are the white glass sponges at 9, almost off screen. There is another small piece of Lophelia at 2.
Oh, and I think a starfish also at the base there. Or are my eyes deceiving me?
I'm not sure where you're looking.
Where you just pointed.
Oh, over here? I'm not sure. We'd have to get closer. There is a rat tail, though, down there.
Yeah. But that looks good, Pilot. Thank you.
There's that purple crinoid again. I think Xenometra, maybe? Yeah, on the black coral.
So just to keep an eye for time-wise, we are now into the portions of our extended dive. which we're able to do graciously because of the ship crew. They're working now extra time, as well as the ROV team and everyone. So a big thank you out for extending this time so we can continue exploring.
Now we might have a pakistrellid sponge at 6 o'clock. This white lump. I get a little closer and take a good look at it. Maybe choristidae? Not quite sure. Yeah, that guy.
Oh, with some pencil sea urchin under it.
Yep, Stellosidaris underneath it. Along with Hylanema, it's at 3 o'clock, which is the wedding sponge.
And a shrimp.
And a shrimp. And a stylaster. Chris, are you on the line still? Any interest in this guy?
I'm here. I'm here.
Any interest in
Hi.
this white sponge?
You guys are doing amazing. I'm here hearing you. And yes, the one on the left, I'd say probably a Pacastrelide. So it is a Astrophorida. Family within the Astrophorida. That is Pacastrelide.
Pakistrellid.
In the Astrophorida, we have the Geodias, we have Erilus, we have Pacastrelid, and Corynix. These are more the form, they're not as roundish. The one that we find, these two white ones that look like we just saw before, two round sponges that might be Geodia or Caminos. So we have definitely this large, probably Pacastrelid sponge. And then there's few genera that could be within the family, but they're common and abundant at these depths.
Oh, nice.
They're common in the collection. So good,
no need to
very
collect.
good eye.
I do like the veining in
Nice,
it. That's
you.
really neat.
Yeah, it's not something you see in a sponge every day.
Yeah, look at you can see the osseous with it surrounded by a white membrane that can close like a diaphragm, and then these branching canals that are not that common in this group. So that makes me a little suspicious. So
awesome.
Feel the general shape and the color and the massive amorphous form. It's particular for this group of astrophorids.
Thank
and then
you.
Right below it, two sponges of probably Caminos or Geodia.
Geodia, right. Thank you.
They become very sharp, spongy.
Thanks, Krista. That was a really interesting
Thank
looking
you.
sponge. I've never seen one with veins and whatnot.
So we're facing downslope.
Many sponges have this. These are part of the excellent canal system that end at the oscule. So there's a system of small canals that get together, sort of like small rivers get to main rivers, and they end in the large opening of the oscule.
Right, like
It's not very common to see it in this type of massive sponges.
What, like the veining, right,
Especially
underneath it?
in astrophorids. So that's why I'm a little doubtful now when we look at these surface features. These features are very common among Poecilosclerida, among some what used to be called Hadromerida, that are dispersed in a few orders, like Suberitida and others. So it could be more common in other orders, not in Astrophorida. So we'll have to keep an eye on this.
Absolutely. Thank you.
You're welcome. Thank you.
So we have another pancake sea urchin.
Ariosoma.
And now going downslope again, we're having a lot of the coral rubble, still quite dense, and we're having some small pockets, but still high diversity in corals that are actually alive and present in this area, just not nearly as large as the ones that we saw maybe at the crest or at a dominating current side.
We have another one of these yellow scleractinian corals at dead center. It looks like Lophelia, but it might be Analepsamia. I think if John's on the room, he's good at telling the difference. If we can get a snap zoom on the yellow. It's very bright. I wonder if it's a sponge-covered coral instead of a natural color.
Looks to be the original coral.
Yeah, you can see the polyps are yellow too, I think.
Can we get closer if possible?
The base of it has white.
I think though that tissue then is dead.
Oh, maybe. Yeah, it's definitely yellow polyps.
Is that the max zoom? Oh, perfect.
Oh,
Thank
too
you.
cool.
Wow. That is some bright yellow, highlighter yellow, with some squat lobsters on them.
You can see it's definitely the tissue. See, some of these branches to the at 3 o'clock, you can see how thick the tissue is, and you can actually see the skeleton underneath.
And the polyps also have that yellow tint. And the base of the skeleton there that you see is bare. It's all white. So the skeleton itself is white, but it's a good indication that it's all coming from the tissue. All the colors coming from the tissue.
Right. I got Afaria hexactinellid at 9 o'clock off screen. And then in the background, you can see it looks like Madrepora, really small Madrepora in the background.
Or
right
a
behind it could be a stylaster, but I'm pretty sure I see the coralites, right? The coralite cups like this.
Nice.
Yeah, look at this one here at the bottom. The polyp is still alive, but the tissue next to it isn't. So you get this bright yellow polyp stuck on this white skeletal system. That's pretty fun. It may not be fun for the coral, but it's cool for me to look at.
No, thank you.
No, I'm not.
Are the lasers on? Maybe we can get a quick overview of the size.
Yeah.
Oh, so this thing is about 20 centimeters long-ish, maybe just a tad longer, but I would say robustly 20 centimeters in diameter. With some broken shards on the floor that are still alive. So maybe it was either recently broken or those corals are happy enough to try to keep growing. Maybe form a whole little new colony. Thank you, pilot. Okay, no problem. So in case you guys didn't hear that at home, the pilots are going downslope, and that's a little harder to maneuver. So they kind of need to be able to see what's going on and what's happening. So we might not get the best view of certain things, but we'll definitely keep an eye out for it.
Got a good jellyfish coming in.
Oh. Some of its tentacles are shorter than the others.
Maybe curled up, maybe caught something.
Maybe. And I think what we're looking at is the pinnacle of that second mound there. Sub-pinnacle. Oh, okay.
Okay.
So we're on our way to 3.
Yeah.
Okay. Okay. I got something in the water column.
Oh, waypoint 3. And here there's, even though a lot of those, ooh, cool.
Oh, fun.
Yeah, we'll just let him go, but he looks happy.
Yeah. That last one couldn't detach himself from the bottom. Yeah, whatever you guys need to do, go for it.
And as I was saying just earlier, we're starting to see again that coral rubble on top, but it's maintaining its coral framework structure. So it hasn't been degraded as much as some of the coral rubble that we see more at the basin or at the bottom of these ridges that we normally see. So either these corals recently died off or they've been able to withstand some of the material from degrading on its own, but this is kind of how a lot of mounds either start to form or continue to form, where they have these pulses of these cold water corals that are actively growing at some points, and while at certain times these corals then all die, if they're able to stay in their framework forming form, they trap still and bind sediments all together, and then over a series of hundreds of thousands of years they start to accumulate and grow taller and taller, and maybe at one point reaching their pinnacle at their peak and start maybe eroding, or they stay just as is with corals on them. But as long as there is coral growth and some form of sediment transport, which usually happens in the current systems, we start to see mound development or continued mound development. And this is quite common in many different areas across the Atlantic. I have another little guy swimming in the current.
Yep.
And despite all the coral rubble around, we do still see smaller octocorals sprouting out in all shapes and forms around this entire area here. So it's not that it's barren. Some corals have established in this area. They just maybe not have been here as long, or maybe can't live as long as some of the other corals that we've seen. Either current changes or they die off by some other means.
Yeah, it looks like we might have a Leiopathes in front of us at 6 o'clock off the
Out
edge.
in the distance, yep. Yes.
Yeah. That's a pretty robust one. Another Leiopathes. This is black coral. This is one of our target species we already collected today. Also, guys, at 10 o'clock, there's a white coral at 10 in the background. When you're done with this guy.
And his base seems pretty wide too.
Yeah.
He's got lots of solitary cup corals around him. Now you're at noon. The white one at noon, it looks...
The stony, jagged
yeah,
one.
Yeah.
And now we're right at the waypoint 3, near just past the pinnacle of the next region, which we're starting to see a lot more taller, more robust corals on this side, so that could be indicative of the current systems. Maybe there's more favorable currents transporting food on this side rather than the other side that where we just were.
Yeah, it's not uncommon to have more life on the current facing side of a mound in this area. That's where most of the food comes from. So most of the corals have a tendency to live or to thrive better on the current facing edge. So I'm not really surprised by that. It's also really common on Lophelia reefs, where you have lots of living Lophelia on the southern side, because the current comes from the south.
Yeah.
At least in this area. We might have another Hylanema sponge. 4 o'clock. Some small gorgonians around.
And for those of you just tuning in, we are on our second dive for this cruise. We are on the eastern side of the Million Mounds region on a ridge, scaling from the base on the Scour side of the region to the east up towards the top of that ridge. And this ridge was recently mapped not too long ago in bathymetry, and this is the first time we've seen this structure on any sort of mapping data. And it is the most eastern ridge or mound-like formation before on the Blake Plateau before we hit a general plateau area where there doesn't seem to be any structures. So this was of particular interest as to why this ridge formed here and how. And we were hoping to find some deep-sea coral communities, which if you were joining us and tuning in earlier, you saw that on certain pinnacles of these mound ridges, we start to actually had some wide hotspot diversities for a lot of these different corals, and we scaled from the depths of 130 meters deep, and now we're about at 760 meters, which is the top at one of those second pinnacle mounds, and we'll be then traveling towards maybe a second, and if we can get to even a third one.
Oops.
So stay tuned, and now we have a fish.
Yep.
That was recently discussed. What are they called again?
This one I think is the Darwin slimehead. The one we were looking at before, which had a lot of texture in his face, I think was a different one. So this would be an orange ruffy, I guess. Beryx. If not, it's closely related to it. Feel free to correct me on the chat. But, yep.
Nice. Oh, and it's quite large, too.
Mm-hmm. He's big.
Kind of put some of the corals in perspective a little bit of how big they actually are.
He might actually be a relative of the big eye. That's another option. Again, I am terrible with fish. Terrible. He's a red fish. It's the best I can get you.
Oh, coming straight at us. And we have a bamboo coral in the distance, potentially.
I think likely he is a bamboo.
Some little
Another one?
spocky, sporadic little stylasterids, potentially. Oh, wow. When you find one, you find plenty. Oh, saying hello right
hello.
to the camera. There's another one of those bulbous sponges with silicate stems, or stalks, rather.
Hylanema.
Sounds good.
Wow, what's
Oh,
going
this
on here?
looks interesting. So we're all of a sudden angling this very steep slope, and it looks like it's unconsolidated cements, but it has some slab textures, and it seems to be jutting straight from the mound upward and vertical, and it looks like, oh, that could be part of a tip of this mound. I'm wondering if that slope feature is just on one side or one angle. It looks, almost
Like
90 degrees almost.
Can you swing to 90? I'm sorry, swing to 9 p.m. Yeah, let's look at the top of this.
Wow. Can we look at some of those dark features that are right on the bottom of the field of view? I don't know if those are holes, but those look like they have legs.
This is what I was taught was a bioherm, right? This texture.
Yeah.
Where we have the consolidation of the Lophelia over, and in combination with sand over a lot of time, it actually builds this giant mound. And I'm actually surprised we didn't see live living Lophelia right on the top of this.
Yeah, oh, those are actually holes, and what I thought were feet were little corals. So quite interesting to see here. So it's not that this feature is hollow, it's just likely that the porous structure of the underlying limestone has been slowly eroded away, either by changes in the ocean chemistry, acidification, overall just if there is a change in the chemistry of the ocean, that could very well have change the internal structure of it. We know that the limestone is very porous, so water does travel through here. It is very permeable. And a lot of the associated biota use these internal structures of caverns and tunnels for their protection and also for food. So I think we're good here. Thank you. And then there's a really cool sponge coming out from that roof of underlying there.
That's probably a pacastrelin.
We can get closer.
You want to get close to the
Oh,
sponge?
wait, sorry. If we can get another zoom in on that sponge there that's coming from under it.
That's like volcano sponge, it looks like.
An upside-down volcano.
Before, or it seems it might have the canals at the surface.
Yeah, so if we can get a tighter zoom, if you guys are comfortable with that.
Really neat.
Like pacastrelid.
Okay. Thank you so much. Yeah, it doesn't have the canals,
Okay.
but it does have the pacastrelid look, appearance.
It does or doesn't?
It does.
It does. Yeah.
Kind of a massive sponge.
Yeah.
Thank you very much.
Thank you.
We have more of that wetting glass sponge.
Basket wedding sponge too.
Yep.
Yeah.
No, go ahead.
No, we're good. Thank you.
Thank you.
Thanks, Chris.
Oh, that little shrimp there.
Yep, the shrimp.
Possibly a royal red shrimp? Like we were thinking
maybe a
about
red.
earlier?
Yeah, royal red. I don't know. Royal reds usually have these swimmerettes at the bottom, right? And they, you know, so I can't tell from here.
Yeah, we can't see that.
Yeah, no, we might even be too deep.
Yeah. So to answer your previous question of if this looks like an overall bioherm, a lot of the community has referred to them more as these cold water coral mounds if they're really associated by unconsolidated sediments and then the buildup of corals over time. This would seem more like a combination between a lithoherm and maybe a bioherm.
Okay.
Because the underlying substrate is straight at the surface. It doesn't look like it was built up by a lot of corals and then some substrate or some sediments rather inputting it. So rather than this limestone seems to be that main feature of why these ridges even exist here, and the corals are just happen to colonize upon it and take advantage.
Nice.
Ooh, what is this?
Siphonophore in the water column. Yep. You can see his bells he uses to swim. Look how cool.
Really interesting.
Yeah. I'm surprised how good at swimming they are. They're, they're jellyfish, or, related to jellies. They're gelatinous. All right.
What are those orange?
They're either a feeding organism or a reproductive organ. I'm not really sure. I don't know that much about siphonophores.
And here, right at the bottom of that little pinnacle that we're looking at, again, we have the coral rubble, so the substrates can change quite quickly overall on what we're looking at here. Oh, and there seems to be another little fish hanging out in the background. Oh, just getting out of view now. He's gone. But yeah, so as soon as that hard substrate ends and we're starting to get more of the coral rubble in this feature, despite we still probably will see some coral in this area taking advantage of maybe the coral rubble of colonizing it, but we are seeing a lot of that hard substrate being more of that factor of the preference of these corals, that because not only are they larger, they hold the corals better, they're solid structures so that corals can really settle on them and continue to grow into these larger structures, whereas the coral rubble in itself does move quite a bit still, so using the coral skeletons can begin to colonize on them, but unless they have the ability to stabilize some of them, it seems to move quite a bit. And if you change the focus on, I think there was another one floating in front of us as well.
It's a hatchet fish, I think. Look how cute.
Is it alive?
Yeah, I think so.
Oh.
If we can get a good zoom on him. Oh, a little hatchet fish. He's so cute. Swim, little buddy. He's not very good at swimming, is he?
No, but he's good at reflecting light.
So these are the ones that had the I was talking about, had the light organs on their underside.
Yep. And this actually looks to be like, can we get a zoom in on that pinkish coral off to the left? That seems to be a one of them. Yeah, right in the center. Yep, thank you.
The Bathypathes.
Yeah.
Alternata, probably. We collected a chunk of this
Someone
earlier. Yeah.
Was it the same one?
I'd have to look at it, but likely. There's the hatchet fish again. Yeah, so in order to tell the species, we need to have a full frontal view of it. I think a big fish just swam over us.
Yeah. Or was that just the lighting of
I don't know.
the ROV? I was looking over. Oh, yeah,
I see
there
it.
it is.
I see it in Sirius.
It's off to the right there. I don't know if it's One of those orange
roughies.
roughies.
So this is a black coral, Anipetherian. Yet again. I think this is, yep, so the alternating branching off the main stalk puts this at Bathypathes alternata. That's the genus and species.
Hence the alternating branching.
Yep, that's where it gets its name.
When we see those individual polyps. It seems like part of it was either eaten off or shrubbed off a little bit. One of those branches there.
Like they got stripped.
Yeah.
Right, and squished up.
Some of those polyps look squished. Wonder what could have done that.
Oh, who knows, man.
Maybe the culprit's behind, lurking.
Right there. This little pink guy in the ground. It's like some lobster or squat lobster.
Or shrimp. That was good, thank you. Seeing quite a bit of coral rubble with some spots of a lot of octocoral sprouts happening. We're seeing a lot of these like slab textures, but what's interesting is also that they look like they're laying on top of each other. And it's almost like they were overgrowing on one another. And it would be interesting if potentially ever a core would be extracted from here to see if those slabs are indicative of individual layers that formed over time, or if it just so happened to look like layers because of the overall erosional pattern in this area.
We've got some scleractinian coral, maybe some white. I can't tell from this far away, but it might be either Madrepora or Lophelia at 9 o'clock. And bamboo corals, 2 white bamboo fans. Oh, we have another Liopathes.
Wow,
He's
another
pretty
really big one.
This is quite steep right here.
Yeah.
He's got a little squat lobster living in him.
With a bamboo potentially next to him. Some cool sponges. A variety of them. Everything from that wedding glass sponge that we've seen quite commonly today to some of the more pudgy ones.
Pudgy ones. That's a good name for them. Little pudgy ball sponges. They're probably Pachistrellids or Geodias. It's really hard to tell. Again, a lot of these sponges look exactly the same. What is that?
Ooh, floating by.
Ooh,
shrimp.
Oh, cool. Bye.
Maybe look like a pelagic shrimp
I
of some
know.
sort.
I have no idea. That was cool.
Yeah. We're seeing quite a few of these large octocorals and Liopathes. We're seeing now on the other side another large drop off with a high slope.
Yeah, that's like probably 50, 60 degree slope.
Yeah. Oh, another fish coming into view. Over there. Is that one of those orange?
Orange roughy or
These-
big eye.
Maybe a big eye. I don't know how orange he looks.
Yeah, from a distance, their orange color kind of just disappears.
Fades, yeah.
They just turn shiny.
Guess it's close to the red scheme enough where it can disappear.
All right, well, I think we're heading a little south, going down slope.
So we're looking down slope, which is why we're seeing mostly blue water, just so the pilots can navigate better. Once we settle out, they'll be able to look around.
Yeah, especially going down slope, it's quite difficult for them to navigate.
Looks like we have some more possible bamboo corals ahead. Yeah, go for it. Okay. Mhm. Okay. Yeah. Yep, go for
Sounds
it.
good. Looking a lot at these coral rubble areas. Some underlying sediment. I'm not seeing too many slab structures, limestone that we were looking at before.
Yeah, I wonder if it's probably underneath it. It's just been filled in, you think, by sediment movement?
Maybe.
Because we're still on the top of this huge mountain,
Yep.
right? 100-meter tall, roughly, ridge that we're on. Oop, not that.
Trying to see what that dark feature is off on the distance.
In the middle? Yeah, I don't know if they can snap zoom. At dead center? Yeah.
Maybe it's another hole. Oh no, it's a coral.
No, yeah, I think it's just Lyopathes. It's just half dead.
Yeah, that's why the dark skeletal structure.
Mm-hmm.
We're good, thank you.
Got lots of small primnoids when you get close.
Yep.
Sure, tiny gorgonians.
You're so good at identifications.
Me?
Yeah.
Oh, I don't. I feel like I don't know anything out here. I'm like constantly asking the shoreside team for help. I can see stuff from far away, I think.
Well,
But-
that's good. But then
yeah.
how did you get into marine science, especially marine biology?
So when I was little, I think my first memories really is when my favorite book had a picture of an octopus in it, and I make my mother read it to me all the time. So I know I have an addiction that dates back from when I was a child. And then I remember, I'd be obsessed with the nature shows and watching all the invertebrates, the corals and stuff move their tentacles. I was fascinated by that. And then I saw Edie Witter on a special where they had the Johnson Sea Link submersible out, and she was taking this mesh tray, and she was able to run it. They were diving with a submersible, and they were running it through the water at night, and the bioluminescent light would show up on this tray that she had. And I was like, I saw the Johnson Sea Link, and I was like, I am going to do that one day. I'm going to do it. So in 2005, I started grad school. I went to undergrad for marine science and biology, and then I started grad school with Chuck Messing, who was on talking about crinoids today, right? He was my master's professor. I didn't really have a thesis professor until maybe 2 years into grad school, and then an opportunity opened up for me for someone to work under Chuck, Dr. Messing, I guess is what I had to call him for 5 years. And there was one spot on the boat on a Johnson Sea Link cruise, so I got on the RV Seaward Johnson, and I got to dive the aft chamber of the Johnson Sea Link to 1,280 feet underwater
in a Lophelia-covered sinkhole. I think Chuck was in the front bubble and I was in the back, since I was his graduate student. And that's where I met John Reed. So when I met John, Chuck said to me, "You're going to meet John Reed. He is the leading expert on deep sea corals." And I was like, okay. So I met John Reed. And then I continued my master's degree. And then in 2007, I got to go back on the Johnson Sea Link. So I only got to dive the first time, though. I didn't get to dive the second cruise because we got blown out and spent like 4 days in Key West. So I met John again. And a couple other people, too. I met Grant Gilmore and Cheryl Morrison, who works on Lophelia. Sandra Brooke, I've worked with her a few times. And then I divorced my ex-husband and moved to my uncle's farm in New Jersey.
A bit of a change.
Yeah. I didn't have a job because I was graduating grad school in like 2008. And there was really nothing open, right? Because our economy wasn't doing well. So I shoveled horse manure for 7 months. And then John called. I saw the position on posted online, and John called me, and we had an interview, and he hired me. So I'm working with John since 2010.
Wow.
So he rescued me from the farm. No offense, Uncle Mark. Yeah, so that's how I got here.
Wow.
Yeah, and then I got to dive the submersible a couple more times, and then the submersible went away. So we've basically been doing ROV stuff for the past couple years. Yeah, it's been an adventure, right?
Definitely.
I have a crazy life. I've been
Not
on
your main track.
Right, I've been on the Okeanos. My third time on the Okeanos. I did a mapping internship in 2012 where I did the night shift, which I apologize to all you night shifters. It's brutal to be up from 7 p.m. to 7 a.m. It's brutal, brutal, brutal. So if you ever get invited on a boat, make sure you are quiet and respectful of your night shift people. It's horrible. Yeah. And then I got to go on again in 2014. I was a co-lead in 2014, a biology co-lead in 2014 on the Okeanos, and then they invited me on this one. So yay.
Nice.
Yeah, how about you?
Way to go. I'm less eventful. My whole life currently has been in university. I grew up in Miami, and having the ocean so close was not just convenient, but quite a privilege.
Right.
And so how do you tire out a whole bunch of kids? Well, you take them and go swimming.
Mm-hmm.
And we, oh, look, a little starfish there. We went swimming quite often, first in pools, and then when we got comfortable enough, we went straight to the ocean, and we were barely 5 years old by the time we were tackling those waves, and kind of fell in love with the ocean. Didn't know what I really wanted to do through school. All I know is that I really liked science, and I liked math, and I liked being able to be part of something bigger than myself. And being able to be that person that points something out that might be life-changing or different or a whole new something or other. And so I applied to a lot of undergrad positions. In the end, oh, he looks cool. Is he the same species that we've been seeing before?
I think so.
Yep. And there's quite dense
him,
coral framework in this area, too.
Yeah.
And
What's
I
a
think you
rebel?
have Oceanapia
And
too.
another Oceanapia.
Yep.
Maybe.
And then one
With
of those...
what is that?
the white thing or the
Yeah,
pink thing?
Both.
What's the pink thing? The pink thing, I think, is a nudibranch.
That does look like a nudibranch.
If we can get a good stable... We are in some serious current here, huh?
Yeah.
Yes, that pink thing is a nudibranch. I believe you can see his... what they rhizophores, I think they're called. The two, they're not antenna, they're...
Almost antenna.
Breathing? Yeah, the breathing, the branchia, which they call nudibranch because it has naked bronchia, right, are actually in the back. But it's basically a sea slug.
Oh, nice.
Right.
And then what's that white thing?
I think that's a Gorgonian, a single-star Gorgonian.
Oh, he's
Maybe Unicia
on
or
the
Unicella.
struggle
or-
bus there.
Oh, how cool.
Oh, he is really cool.
And we have the hydroid, the greenish hydroid hanging.
Yep, that was
Ocean
attached to
apple.
onto the Oceanapia. Yep,
Yes.
that's there.
Oh.
Really cool.
If you see to the left of the Gorgonian, the white Gorgonian, there is a little bulb.
Yeah, the
that
white
we've
bulb.
been seeing from from the first day we've seen it, this small size exactinellid, it seems, with lots of ocules.
Nice find.
Yeah.
Go back.
Coming up on some pretty dense framework systems over some soft sediment, but this is definitely a more dense patchiness of broken up coral rubble. There's definitely, I don't really see much of a framework system really, maybe a little bit off to the right there, but for the most part it looks quite broken down and degraded. So it doesn't look like it's withholding a lot of that framework system. We're still seeing quite a bit of some smaller solitary corals, isolated corals. Colonies, there may have passed just a small scleractinian there. There's another one of those sponges with that column that we had been passing earlier.
It's an Oceanapia.
Yep, and then a pancake sea urchin off slightly to the left-hand side there.
And here's a new sponge. This brown guy.
Yeah. Krista, are you there to give any insight to what this is?
Yes, I'm here. I'm glad I'm here.
You can't leave, can you? You're glued.
Look at it a little closer. It's massive, but it seems it has a very different system of attachment.
Yeah, look at all these spicules sticking out. This is crazy.
This is an exactinellid by those very long spicules.
Is it really, even though it's brown?
Here at the base. Or it's not a different organism, is that? Because to the top of the sponge, it looks like these filaments could be something like hydroids or something, but at the bottom, they definitely look like exactinellid spicules.
Oh, crazy weird.
Very distinct morphology. I have no idea, but let's photograph it and let me put it in the notes because it's someone that works with exactinellids probably will recognize it.
You don't have any idea? I've never seen it before either.
No, I don't have, but let me just make a picture. Definitely an exactinellid, and it's a good size one.
Really interesting. It does look like even at the top, those are the same spicules.
Doing a close-up with numerous pores.
Yep.
And let's see.
So it has the same texture of the DMS tea sponge we find in the tan starlet sponge we don't have a name for yet, that we find all over the marine protected areas off the eastern coast. It has that same texture at the top, but I am blown away by these spicules at the bottom. I can't even imagine.
And the pattern of the distribution of these spicules. So it's sort of like what Iolanema has at the base, that are stringy, of course, much less long. I wanted to look closely in case it's another organism coming off, but it actually seemed to have some of these filaments are smooth, but some have certain patterns. So we might have combination of spicules of the sponge and then the other organism that could be a hydroid coming off.
Right. Is there any possibility that the bottom part hexagonellid, and then it's got a demosponge growing over it. Is that a thing? Or you're pretty sure that that's the whole thing's hex?
With sponges, you don't know. You can have chimeras and crazy things. I think it fits with certain patterns among exactinelids.
Okay.
You might have an area it could be a fringe, it could be a base, it could be a crown at the top where you have particular species coming off. Evidently, here I see these filaments being other organisms because I can see some repeated structures that are not sponge-looking.
Okay.
But at the bottom, honestly, I could be mistaken, I could be wrong, but it seems to be an exactinellid with a fringe of basin species.
Do you think it's a new species?
Since I'm not an expert of exactinellids and I haven't been exposed in the past recent years to these communities, these deep water communities, I would prefer to show it, to have Shirley look at this and maybe show it to our colleagues that work in exactinellids.
Okay.
We should do it. Alan Collins, yesterday was looking at our dive. So let's try to send him a picture. If you send it to me, I can send it to them and to Henry Racewig.
Right.
To see what, okay. That's what I would suggest because we might find it again.
Okay.
Is it possibility to collect this? What do you think?
I think we have one box left.
We do have a bio box left.
To change the symmetry of this beautiful specimen, but it could be an opportunity to find something new.
I mean, we only have an hour left, so it would be the last specimen.
It would be.
If it's new. I mean, this is new to me.
Yeah. Pilots, are we in a position to collect?
Before we do that, will this fit into one of the bio box bins without breaking it up too much? Like to keep it whole?
I'd say to take a snip because it's a pretty large specimen, so it will take a big space if you're going to put the whole thing.
Okay.
What do you think?
I would
recommend to take a snip on the side.
You just want a snip of it?
A snip would be possible if it doesn't fit.
Exactly. Very strange.
Right. Because you have to shift the ship backwards? Oh, okay. Then maybe we should skip it if you have to do that.
Oh, if it's possibly a new species, might be worth collecting. We're not too far from the peak of the second or third, rather, mound feature, so I think we could still manage it.
Okay. Yeah, if you can collect it, let's try and collect it. Just because we think it might be new, and I'd hate to leave it there. Sorry, guys. Okay.
Sounds good. And if we do take a snippet, it could be possible to suck it up. Just because that last bio box does have that potential floater for the sample.
Welcome to the
So if you're just joining us, we're trying to make our way back to a possible sample collection of a new species of sponge. We believe is a hexactinellate or a glass sponge. So we're just getting the ship lined up with the 2 ROV system in order to line up and have our ROV pilots pick up a sample. So we're just doing a quick turnaround here. I think that's it right there at 6 o'clock. So it's in view again, right here at 6. Chris, just to ask, you want a morphological specimen and a DNA, ideally? You don't have to tell me on the phone, you can just tell me in the chat room, is fine. I wouldn't worry too much about that floater. My guess is it's probably not going to go anywhere. It's not very, the current's not really bad, is it? Yeah. That being said, if it pops out, I mean, I'm going to feel pretty stupid, so. My guess is too, this is not really stuck to the bottom. It's probably pretty loose. So if you grabbed it with a claw, it should just pop right off. But again, just a guess. I really not 100% sure on that. That's fine. Yeah, that's fine. Okay. Yeah, snippet's fine. I was wanting some of those spicules. But if you, there's some coming out of the top, so if you can get them out of the top, that's, you know, it's like chunk off the top, that's fine too. Oh, that would work. Let me ask Chris. Chris, would you, if you're still on the line, would you, is it okay for us to put this in the rock box? And the
Yes.
you're gonna have to mute
I
your
was just thinking
TV.
on the kryptonite material coming out of it, but I think it should be clear. It shouldn't be leaking too much. And I'm just worrying for DNA in case that this liquid or mucus
Okay.
could inhibit something. But if it's okay if that's the space that is available.
She said just a small piece of DNA is perfectly fine, but if you end up, having to put it somewhere, if you pick the whole thing up and want to take it, the rock box is okay. It's better to cut she's saying it's better to cut a small piece because we only need a little bit of DNA for it. So if you cut a little small piece and suck it up, that's plenty.
Now, the question was if it could be together with the rock, with a piece of rock.
No. So the rock box the question was whether or not it could go in the rock box. The rock box is open to the environment, so it's going to be exposed to warmer temperatures as it comes up out of the ocean. So that was the question, because the bio boxes are not open to the environment.
If you can take a piece from the side bottom.
From the side bottom?
It would include this long.
Yeah.
That are so weird.
So did you hear that, Pilot? If you can get the side of the bottom? Yeah. Yeah. Yeah, that's fine.
So we don't know how this part is going to break, and so, I won't be too picky. We can take a snip of it. It should be able to be identified by an exactly expert. Thank you.
Yeah, thank you.
Thank you. I just sent a photo to Alan Collins and to Shirley.
Okay.
I hear from them.
Oh, look at this. Yeah. If it's easier just to take the whole thing, But, yeah. Little pieces.
Is there space to take the whole thing?
The issue was whether or not they could put it in the rock box, right?
Okay.
Yeah.
Because it's open.
Yeah.
It's such a weird specimen, Steph, that it might be worth.
Yeah, it is weird, isn't it?
The whole thing, really.
Yeah, go for it. Whatever's easiest. Look at the exudate coming out of it. Sliming up.
But if it's a new species, it will be a beautiful specimen for the Museum of Natural History.
Yeah, it would be.
Yeah. Never in the bucket.
So I'm going to go eat dinner, Chris. I'm going to leave you with Kim. Kim, they're just putting it in the portside rock box at the moment.
Oh, we were able to have a sample?
Yeah, we're gonna grab this guy. There's a shrimp in there.
Yeah, he just kind of helped himself right in there.
So I'm gonna go eat dinner, Chris.
Okay.
I'll talk to you. If you leave before I get back, have a wonderful night, and thanks for helping us out.
Well, the same. It's a pleasure to be with you guys.
Yeah,
Girl.
thanks. All right.
Thank you.
All right, bye.
Hi, Kim.
Hi.
Wow. Oh, my God. Well, the good thing of bringing all this is that we probably will have a lot of species associated to that rubble.
Yep. Oh,
That
that
maybe
shrimp just
have
got
never
out.
been studied and found before. So
Yeah, no, absolutely. And once potentially, if anyone were to want some of those core rubble samples, once all the manganese crust is coming off it, you could potentially age date it. We could find out how old this substrate is. I'm guessing it's not terribly old, and speaking in geologic age, probably not millions of years, but maybe a few thousand at most, since these ridges weren't even known. So who knows? This has never been done before.
well, this is very exciting. Thank you for the pilots and everybody involved in having to move the ship. And
I'll definitely let them know. I don't know if you guys in the front heard or anything, but a big thank you for being able to collect these samples. It was a great help and being able to navigate the ship overall.
Mhm.
Looks to be quite exciting. And I don't think it looked like it was another sponge taking over a sponge, but it looked like it had its own pinnacles, or spicules rather.
Yes. Yes.
Really interesting find.
Yes,
Let's just
for
say
the
I'm
last moment or the last time of
the dive, yeah.
It's a great closure.
It definitely is.
I already sent a message with a photo to Alan Collins in the Smithsonian and to Shirley to see if they have seen this, something like this. Now, Stephanie has been in all these cruises and she hasn't seen something like that. So that makes it very exciting.
Yeah, absolutely. And I look forward to working with this a lot more than whatever dead, potentially stinky other material that we pulled earlier.
Will you be able to look at this in the ship or will you do it back in the lab?
So we do have to, if we want any DNA sampling or anything of that, we do have to start to preserve them right when they come on board. And we usually do so by 95% ethanol. And because it's a sponge, we'll probably have to cycle out that ethanol quite a bit to make sure that it's all preserved inside. If you want a side sample for DNA, we can make sure we can take that and make sure that there is always a sample for DNA. I know the structure for this one, given that it might be a new sample, is kind of important, so we'll try to keep the integrity of it as much as possible. But we'll be able to potentially photograph it and maybe do an initial very rough workup of the overall structure. If you want to break it up or cut it into pieces, that would have to be done all at the lab, and you would probably have to request that sample after.
Okay, excellent. Take a look to see if those filaments, if some of those are some other type of organism, like a hydroid or...
Yeah, we'll definitely take those samples, whatever is attached to it. We normally we call them sample associates. So those are secondary samples that would then come up with it. And if there are quite a few of them, we'll take one or two. But if they're very unique, we'll probably pluck out what we find to be quite unique associated with the sponge.
Okay, well... Really exciting to be navigating with you guys.
Yeah, we're excited that you're able to participate this much.
Yes. This is thanks to Shirley Pomponi and Joshua Voss that have allowed me to be part of this from via telepresence. And I will be with you every day, I believe.
Oh, that's great.
Yes.
We look forward to it.
Okay, thank you. And I'm learning a lot about so many things. So,
Hey, I am too.
excellent.
I think that's the goal of this overall. And it's fun to be able to discover all new things and go places, well, no person has gone before.
Exactly. You know, today in the morning I was reading about Alexander von Humboldt and von Plant, two very famous explorers of German origin that around 200, a little bit over 200 years ago, did very important discoveries in South America. And I was thinking of you guys and on this type of explorations, how lucky we are to reach this material that has never been observed. Some of it not collected in this way because the collections were done with trawling before.
Yep.
So this is very, very special.
Yep. No, and I completely agree with you. We are very fortunate to be in a position where not only do we have the funding to achieve this, the technology has caught up to us. So, like you said before, the only way we could discover places like this is trawling and potentially destroying the actual habitat and not fully understanding what we're looking at. And now we get to see them all where they've been growing and where they get to continue to grow. So it's really great.
To hear all your geological stories and very interesting as well. For
Yeah,
us.
thank you. Well, the geology speaks for itself. I'm just trying to translate it.
Uh-huh.
And we're doing a quick shift change for the ROV, so we're gonna take a few moments in this area just to make sure that they're all settled in before we continue on.
Have you been in many of these cruises, Kimberly?
No, this is actually my very first cruise. I was not able to participate on really any cruises during my PhD. Most of the samples and everything that I worked with was collected right before I got there. So I had plenty of data ready to work with, so I didn't get to participate on many of the cruises. And this cruise, Mike kind of reached out to me and suggested that they did need someone that was complementary to Stephanie in the fact that didn't know maybe very much of the biology, but knew about other aspects of it, in this case, the geology of the area. So I jumped at the opportunity, and I was technically supposed to defend this month, but my advisor graciously let me move it to December so that I am able to participate on this cruise. So I'm very excited to be here and happy to say my sea legs are working.
Excellent, excellent. Well, you are acting like a pro at this. Actually.
Well, I'm just happy to be here and happy to give my 2 cents on what we're looking at here. So it's been one of my dreams to be on one of these ships, and I'm glad I get to achieve that. And the next time, the next dream is to be down in one of the submersibles also. So I know ROVs are more popular and more efficient and safer nowadays, but still to be down there with it and to get to observe it firsthand would definitely be one of my dreams.
Wow.
And now we're slowly getting a move on back, and we're approaching that last crest feature that we're seeing on top of this ridge towards waypoint 4. And as you can see now, we're starting to head up more of these carbonate materials, and we're starting to see a lot of that substrate that's been exposed for a while, so it probably has that ferromanganese crusting, giving it that dark color features. But what a lot of this looks like is there's a lot of overhanging of these carbonate features, and underneath it seems to be quite eroded in some areas, and not a lot of sediments that are there. This could be due to many different reasons. One such being that there's different types of carbonate material that are encrusted into it while the carbonate actually precipitates, and in certain areas there's more dense or more robust minerals preventing that section of carbonate to be eroded or acidified or some way, shape, or form degraded, and while other ones still are able to maintain their structure. So maybe some areas underneath those slabs that look like they're overhanging actually were softer material that was able then to then be eroded away in this area. And now as we're reaching the top, we're starting to see quite a bit more coral presence in the area, not as much as we were seeing earlier, but definitely more than we were seeing more towards the basin of this area. We see quite a bit still of that coral rubble, which is indicative that at one time this area was covered in some sort of scleractinian reef system that has since died off and become that new hard substrate, as well as the carbonate material substrate underneath for these corals to colonize. And for any particular reason, these corals died off, and now we're starting to see a new generation of these corals come to life, and being either octocorals, such as bamboo corals, or even we still have some residual minor scleractinians that we have seen through time. And we're just observing overall what is here. And we're starting to see, as we get closer and closer to that pinnacle, a lot of these reef framework structures are holding their shape, meaning that either they haven't been degraded very much, that just because just because they are standing with their framework structure doesn't mean that they are younger than the overall rubble in itself. It means that they've just been able to withstand any current systems or any major degradation that maybe other corals have experienced. We don't like to associate that directly with age, considering that certain areas do have that trapping and binding of the overall sediments, which keep then those structures in those framework forming positions. Whereas maybe in other areas you get more current impacts for rubble areas. And we also are seeing some lovely sponges, more of those wetting glass sponges that we've been seeing consistently through this dive. So it seems to be quite a popular area for this sponge to be in. And we're seeing maybe another paramecia out on that bay there, that little pink coral with some ophiurids.
Did we get that sponge?
We did.
Oh, nice.
It's actually in a rock box.
Sweet. I don't know what kind of jar we're going to put it in, but
we'll figure it out. Maybe one of those Home Depot tubs.
Right. Yeah, this looks like that bubblegum part coral, maybe Paramuricea. We got a little shrimp in our hole. So where are we headed? Upslope on Mound 3?
Yep.
Okay.
No, this is a different guy. He looks red and pale, unless he's molting. I don't think he's molting. Can we get a tighter zoom on him?
He still has that really long rostrum, that area between his eyes.
Yeah.
I guess you could call it a shrimp nose, even though that's not what it is. Oh, and we got that Ophiothrix leg, possibly. It's an Ophioid leg coming up from under the rock next to him.
He looks interesting. I don't think I've seen a shrimp this color.
He's really pale.
Yeah.
I do think they can change color. So.
I thought, though, that was kind of minor.
Yeah. I don't know how indicative it is of the species. You see, he still has these tight hair, these small hairs in his, really close down on his antenna.
What?
Pretty neat. And the spines in between his eyes. So a lot of those, where the spines are and stuff, is how you determine their species. How many spines they have, whether the first one's longer than the second one. There's a whole bunch of stuff that goes into it.
Oh, hefty criteria.
Yeah.
Thank you, pilot. We're good. That looks good. And then if we can get a quick zoom in on that paramecia pink coral that's off to the right-hand side there. And we're seeing quite a bit of these cutthroat eels swimming along here. They do seem to like this area. Oh, little squat lobster on the base there. He looks so cute. Ooh, really nice. Oh, tall, generally small paramecia, about maybe 20 centimeters tall, with a pretty significant ophiuroid on him.
Yep.
Taking full advantage of him.
There's something in the back.
Really
oh, that's
nice.
just his another leg.
Yep.
Ophiuroid arm.
And leftover of, well, a somewhat singular branched-ish. Oh, well, maybe not. Bamboo coral. He does have his branches, although some seem to be eaten. Really nice. I think this is similar to what we were seeing before. None of that Paramecia arborea that is on the species list, so still trying to keep an eye out for it just in case, but I don't think that's that species that we're looking for.
Right. Same genus.
Yep.
Wrong species.
Which is quite important.
Look at this little shrimp. Look how cute he is.
Yeah, right at the leg of the ophiuroid. All you see are the eyes.
Yeah, we think it's considered a bubblegum coral, but it's not the correct species we're looking for. So.
Yep. Paramecia is of that bubblegum-esque coral. The arborea are the ones that can grow really big, and that's the one that's thus far as my knowledge go, has been some of the oldest recording corals, going back at least 4,000 or 5,000 years old. So quite extensive and still living had it not been trawled. But really interesting to see. I haven't really, not too common in this area. Thank you very much for that shot.
There seems to be a different coral here at 3 o'clock.
Yeah. Like
It's
3
brown.
And then also the one under it, that purpley one.
Yeah, that's a different one too.
Yeah, so if we can first go to that brown coral right adjacent to it.
The brown one first. Oh, it's a nymphaeid. It's a soft coral, true soft coral. You've got some cool crabs out underneath it.
Yep. Really cool. And whenever you're ready, we can scale down to more of that purple one that's underneath it. But get a quick close-up view. Look, there's a. It's below and slightly in the more in the front.
Yeah, so there's another nymphaeid right below it. That's a different color that we're seeing in screen now. And then there's another coral that looks a little different that you might have to pan out to get to.
Oh, wait, there it is. Yep, you got it.
Oh, there he is. He's purple. Look how beautiful.
This is the first kind that we've seen here on this dive.
Yeah, I don't know what this is.
Oh, and sorry, we've been misidentifying that pink coral. It's Paragorgia, not Paramecia.
Oh,
we've been saying that wrong. Sorry. Thank you. Thank you, Dr. Atsumoto. I'm sorry if I'm saying your name wrong. But this is a really interesting looking coral. I have not seen something like him, at least not really in this region. I'm not too familiar with all the corals, but he seems to have 2, 4, 6, 8 tentacles, so an octocoral.
Definitely an octocoral. It's purple.
Yeah, if we can get a really quick, a fine zoom in on some of the polyp structures. He does seem to have a black skeleton. If anyone that's still tuned in has any insight into what this coral might be, we're all ears. We haven't seen this coral on this dive. It's got a somewhat of a purple tint to it. Really nice, big, robust polyps on this one.
Yeah, it's quite pretty. The purple.
I'm not seeing any defining characteristics on the coral itself. Yeah, you're right. Can we get a maybe even tighter zoom in on some of those polyps?
That's max zoom.
Okay. Well, yeah, it looks really interesting. There are pinnules on the tentacles, which Scott did say are quite characteristic of certain species. I'm not seeing any true defining characteristics from the skeleton, but there seems to be some like white spiny dots near the base of these polyps and on the skeleton, traveling to the tissue on the skeleton too, in case that means anything to anyone. It could be taking advantage of it, although I can't really see the base of this coral to see. Normally you were able to see if they're growing more on patchy effects of different corals. They wouldn't consume the entire thing.
Yeah, and they're pretty evenly dispersed
yeah.
too, so it's pretty cool.
Quite interesting. Haven't seen that before.
Okay, we only have about 15 minutes left.
Okay.
So the neighboring one is yellow, and I believe that's a bamboo. Yeah, the the yellow one is a bamboo coral.
Could be quite similar. This is a really interesting find.
I think it's a gorgonian as opposed to zoanth that's growing on it because the tissue actually covers the entire
whole thing.
Yeah.
Yeah. Cool. Thank you very much.
There's an oceanapia on the left.
Yep.
I mean, right?
I knew what you meant.
Yeah. It's been a long time.
That caught my eye, too. There are some stichiopathes in the background there. Cutthroat eel. Keeping up with the current-ish. Oh, what was that? Oh, is that an isopod?
Isopod. Can you look at 6 o'clock? Bathanomas.
It looks like a little tiny one.
Yeah, a little tiny guy. He's teeny tiny. Yep, that's a little isopod. Cool.
The first one we've really seen in this dive.
Yeah,
we might have seen one on a coral earlier, but he's all by himself.
Yep.
Swimming along there. Cockroaches of the sea, if you ask me.
They sure are. Some of these species get to be over a foot long down here. That's Bathanomus gigantea. I'm not sure about this guy, unless he's a teeny tiny baby.
Maybe.
It's probably a different species, though. I don't recognize the brown spots on his behind his head. Yeah.
We'll call it a head.
Yeah.
Very cool looking.
First
though.
segment. Yeah. They are cool looking.
Really cool.
A lot of them are parasitic, so they'll attach themselves to fish faces, and
Well, that's a nightmare waiting to happen.
I know, right? And you don't even have hands to get it off you.
Well, if we collect ever a specimen, I know where to put him.
No. They're cool to look at. I don't want it stuck to my face.
Okay. Fair enough. Fair enough. Thank you, pilot. Have another solitary cup coral there. Coming up on that Stichiopathes.
10 minutes, yep.
We see more of that bluish crusting sponge that we saw earlier today.
There's a nice bulbous sponge. It might be a geodid at 7 o'clock. It's a yellow ball.
If we can get a little close up, we might be able to say something else.
Okay. If we can get a snap zoom on that ball sponge, that'd be good.
Oh, there's one of those crinoids also that we talked about earlier.
What do you think, Chris? Geodia?
It really looks like the one that you pointed as Geodia at the beginning. You remember that I said might be a petrocid? But then Shirley talked about Caminos, which is another Geoide in the same family of Geodia, and it really reads that spell. So I would put it as in the family Geoide and question mark Caminos.
Geodia, okay. And then if we pan up, we're going to see a really cool sea spider right behind it.
Interesting.
Do you see the sea spider?
I do.
Awesome. So these are chelates. So they're in the arthropod phylum, but they're pretty closely related to spiders on land. So they're in the chelate class, Chelicerata.
Following that 8-legged theme with some, what are those two kind of underleg things that it has bent clipped towards its body?
I have no idea.
That would be really interesting to see, and it seems to have even more than pan out by its abdomen area.
Yeah, there's 4 there, right? That look modified.
Yep.
Yep, but there are 8 main legs there. So neat.
Wow.
Yeah, it is pretty cool.
Really cool find.
They're pretty rare. We very rarely see them. There is one species we find often on the Okeanos reefs at about 300 feet.
Can we get a quick
100
laser on them?
meters.
And he looks to be just under 10 centimeters from tip to tail. Legs definitely surpass that. So that's really cool. And that sponge also seems to be maybe about 20 centimeters in diameter.
Nice find.
Yeah, really cool find. Great way to sum up the end of this dive. We have some cup corals around it and maybe some either some small octocorals. That blue encrusting sponge we see sporadically around this whole area. Some of that gold coral. Ooh, there's that crinoid.
Yep, that dying Myopathes.
Yep.
We have about 5 minutes left. Anybody shoreside wants to take a peek at anything last minute? There's
And Scott France has an input saying that the long proboscis is used to suck out the juices of cnidarian polyps.
nice.
Interesting. One hell of a spider. Glad the ones on land haven't figured that one out.
Mm-hmm. Looks like we might have another bubblegum coral over here.
Paragorgia,
Paragorgia.
not paramecia, which what we've been saying earlier.
Paramecia.
Looks really nice.
Mhm. Looks like there might be plumarella on the back on the right. But this Paragorgia is quite nice. Looks like, I believe Astrosema would be the genus possibly of these brittle stars. All right, so I guess to wrap up our dive, we started this dive at 830 meters at the base of this ridge and got all the way up to about 730, I think was our lowest point. Or 760 maybe was our lowest point. And we transected over 3 large mounds. We saw lots of coral rubble. Mostly the whole thing is covered in Lophelia rubble, which is cool. Oh, look at these guys moving around.
And some carbonate slabs.
Mm-hmm. And then the very tops are covered in lots and lots of gorgonians and antipatharians.
Came across some great coral gardens in this area, although no serious framework reefs forming. We did see a lot of these coral forests, a lot of these isolated colonies.
Yeah, many species of coral. A pycnogonid, that was a highlight of my day. An isopod. A couple sharks. We saw 3 or 4 sharks today, including chimeras. New species of fish, like 2 or 3 new species of fish we haven't seen yet this cruise. Not new to science, just new to us for the day.
What else? We saw some really great fish also. Got some really great facts about them from the chat room. Thanks again for all those that have participated in that.
We collected 8 specimens. We think at least 2 or 3 of them are new, or at least undescribed.
Yep. We got all 8 great samples to continue the Aspire program in order to determine population genetics with their DNA. So that seems really great.
And I have to write we have to write a summary of these dives at the end of each day, so I don't know why I'm not taking notes right now. I should be. This will make my life easier after 2 hours of sample processing.
Well, we'll cross that bridge when we get to it.
Yeah, I'll get there. No biggie. So thanks for exploring with us today. I hope everybody had a good time. Feel free to contact us on our social media.
We're on Facebook. We have 2 different pages on Facebook at Ocean Exploration Research and Ocean Exploration Research -education.
On Twitter, we're at Ocean Explorer, and anytime you tweet us, you can use the hashtag Okeanos on anything. And if you ask us questions on social media, we will do our best to answer them. And sometimes on air. We answered a question on air today. And then on Instagram, we are at Notion, not Notion, jeez, at NOAA Ocean Exploration. You can reach me, Stephanie Farrington, at Deep Sea Nerdy on Twitter or at Deep Sea Nerd on Facebook or Instagram. And Kim is on Instagram at Deep Sea Kim.
Yep. And if you use any of the hashtags for NOAA Exploration or Okeanos, we'll definitely keep an eye on those as well. So
we are diving again tomorrow. Let's see. Dive 3 is at Steson West. I'd have to go back and look at my paperwork. We're going to have a post-dive call probably here at 6:15, I would think.
6:10, actually.
6:10, okay, for anyone on the line who wants to join in tomorrow to talk about today's dive and tomorrow's dive for our core scientists who are participating.
Yep.
But everyone out there in internet land, we're glad you joined us. We hope you join us again tomorrow. Saturday and Sunday are great days to tune in. I like to put them on the TV, on YouTube, and then sit down and do my other stuff during the day and keep the Okeanos on the background.
You'll never know what you'll find, and frankly, we won't either.
Yep.
Everything you're seeing is what we're seeing live to firsthand, so we're hoping for some new discoveries and new features that we're seeing. A lot of this stuff has never been done before, especially in these areas. We're the first people to actually get firsthand views. So
yeah, thanks for coming along.
Yep, and we'll hear from you guys tomorrow.
And
so
don't forget to share our videos.
All right, we're signing off now. For those core participants, we'll have our online dive call for next dive plan call for the next dive in about 10 minutes. So stay tuned.
Bye, everybody.
Bye.
Bye. Thank you.
Thank you.
So, see you tomorrow.